Thank you for being here today with us. This is one of the two amazing videos that the Biogen crew did for us, and try not to cry when I see it. And the other one you'll see later today. So just want to cover a little bit of housekeeping before we get started. We do have a full day, so I hope you guys are here to celebrate Nano-rare with us. We do have 3 official Q&A sessions, but I know that there's going to be a lot of questions throughout the day. And so those of you who are here in person, we will be able to ask questions during the Q&A with the mic. If you are joining us via Zoom, you can put your questions in the chat room. Those questions will be monitored. And then if you are on the live feed, you can email events@n-Lorem.com and we'll get your questions that way. And that'll be an email that's checked throughout the entire day. So if you do have Zoom problems, you can always pop out of the Zoom, go into the live feed that's accessed through our website. You don't need a code for that. You can just find the link there. And so, yeah, so welcome. We're very excited to have you here today. We're very excited to share n-Lorem with the community. And I'd like to introduce Chris Viehbacher to officially kick off our day. He's the president and CEO of Biogen.
Well, good morning, everybody, and thank you all for coming. Why are we here today? I think if we have a success at the end of this meeting, it is thinking about how do we help Stan Crook and n-Lorem in their incredible mission. And we've got an amazing cast of characters here with us. We have patients, we have caregivers, we have scientific experts, we've got people who are technical experts. And so I'm hoping that we create a dialogue about not only what Stan and n-Lorem are doing for specific diseases, but one of the things I'll talk about a little later on this morning is that Stan is not only innovating medicine, he's actually innovating a care model. And, you know, when Stan started off many years ago at SmithKline, and we have a shared history because I used to work for GlaxoSmithKline as well. But when Stan started off, he was developing medicines for large populations. So if we went back 20, 25 years, all we were thinking about are things like statins for high cholesterol, or we had products for hypertension. And the idea that we would treat people with just a few thousand seemed, impossible to do. How could you possibly justify the investment that goes into a new medicine and then only sell to a few people? And then somewhere in the 1990s, a company that was founded just in this neighborhood called Genzyme started to change that. And they came up with a first cure really for Gaucher's disease. And this was an enzyme replacement therapy. And at the time, it was pretty difficult because they needed to extract an enzyme from human placenta. And the FDA said, well, okay, but if you want to get this drug approved, you're going to need to do over 100 patients. And Genzyme said, we have enough placenta for 12 patients. And they went around the world looking for patients. In fact, at one point, they had one clinical trial center in the United Kingdom, and they brought a baby from Japan. But you can't just bring a baby from Japan to the UK for 6 to 8 months of treatment. Genzyme brought the parents, the siblings, and the grandparents all to the UK during this trial. And then it came to going to the FDA, and the FDA says, we can't approve a drug with only 12 patients of data. And so Genzyme wheeled into an advisory committee a real live patient. And that was had made such an impression on the advisory committee that the advisory committee voted unanimously to approve the drug. And then the drug got on the market. But of course, it wasn't really a drug. It was, they used to, in Chinatown here in Boston, they used to have these things that looked like giant washing machines that would take these human placenta and try to extract the enzyme. But of course, that then led to a further scientific revolution where they could actually make a recombinant form. So we didn't need the placentas anymore. One of the most interesting things, and this is something that's always amazed me as I've gotten more and more interested in rare diseases, is at the time they thought there were only 700 patients in the world with Gaucher's disease. It was basically going to be people with Ashkenazi Jewish descent. And over the years, they discovered that there were 7,000. And this is a lesson that I learned many years ago. I was a CEO of Sanofi. We acquired Genzyme and we actually invested further in rare disease. And we're doing that now with Biogen as well. But the minute you offer a treatment, you increase the incentive for diagnosis. And for however many patients you think there are, once you actually offer treatment, you always find more. And that is actually also an extremely important part of making sure people get benefit for these medicines, because you actually need then a team of people that know how to go find patients. So in companies like Biogen, we actually have people that will look at family trees, will go hunt for people. Last night we were talking about a case where some of these diseases got spread by going back to the early days of explorers, and as ships left Portugal and went to different countries. We've just acquired a company called Reata that has a treatment for Friedreich's ataxia. And you'll find that that's only for people of European descent, but we actually have quite a large population in Brazil and Argentina because of those early day explorers, and they brought all of these genetic mutations then with them, but we don't find any in Asia, for example. So it's in all of this, I guess what I'm saying is, first, it's a labor of love, right? Because this is not the way things get normally done. And the second is that you really have to challenge conventional wisdom. And I think we're still not there. And I'll tell you a little bit about how does a drug classically get developed, only to give you an idea of the amazing challenges that have had to be overcome by being able to bring these treatments for n-Lorem. But, Stan, I'm sure, will be the first to say, for as many people as we're helping, the job is not done. So again, at the end of the day, what I really like everybody to be thinking about, how do we help n-Lorem to scale? How do we help n-Lorem to think about, do we need to lobby to make sure that the FDA thinks about ultra rare diseases differently than other diseases? I can tell you a case of a drug called Qalsody later on. And we're still seeing that, the FDA wants to think about really large populations. And the unmet need and the risk profile is just so much different. And I'm sure there's many parents and caregivers here who can tell those stories. But that's what I think, that's why we've decided to host this. I am just overwhelmed by what Stan and n-Lorem have done. So we're very proud to host this, and we look forward to an engaging discussion today about how do we take what Stan has built and make it even better. So thank you very much, and I'll pass it now over to our hero. Where is Dr. Stan Crook to give his opening remarks? There he is. Oh, hero.
Thank you. I have been called many things before I speak, but I think this may be the first time I've been called a hero before I speak. And I suppose I should begin with a confession of sorts. I didn't want to call this meeting an annual meeting because it has such a corporate sound to it. So I coined the term Nano-rare patient colloquium, but then promptly realized I couldn't pronounce that word. And then I learned that I couldn't remember how to spell it. And then I learned that almost everyone who worked for me at n-Lorem couldn't pronounce that word. And so we've been practicing. And so if there's anyone in the audience who has trouble saying that word, I thought about bringing some marbles, Demosthenes or whatever it was in the Greek order, who used stones, but I thought the liability of that might be just a little much. And so my plan was to have you, thank you, have you practice during lunch, but that got me bopped on the arm by Roseanne for having another dumb, messy idea. So we'll get rid of that idea. So if you want to practice, you just have to do it on your own, okay? So first of all, thank you so much for being here. We're thrilled with the level of interest. We're delighted to see so many people here in person, and even more joining us remotely. And I'm particularly thrilled to see so many patients and families who've made difficult journeys for them to be here today. you're why we come to work every day. And so it's wonderful to see you. We have an informative and I think really exciting day planned for you. But before we start that, I do want to thank Chris and all of our wonderful friends and colleagues at Biogen for all the support they've given us from inception. Biogen was a founding donor, and they've really worked tirelessly. That's a strange expression, isn't it? You work until you're really tired. Actually, talk to anyone about at n-Lorem, we know about being tired. But they've worked very hard to help us do this, and we thank you very much, Chris, and the entire team. So we have a lot that we want to accomplish today. We want to share what our aspirations are, what we hope to accomplish long-term with you, the quality systems that we've put in place that are working to assure that every step is conducted with the maximum in quality that we can generate. The progress that we've made, and we think, we hope that you agree at the end of the day that the progress that we've made is really pretty extraordinary. We're certainly excited about it. And to share what we're learning. And what we're learning already is really important. And what we're going to learn will change the way we think about health and disease altogether. And it's time that we do that. We'll be talking about that as the day goes on. But n-Lorem exists for one reason, and that is our patients. And so we're also here to listen. And we had a wonderful reception last night, and I got to listen to a lot of people. And all of the folks at n-Lorem here today are here to listen. So we're anxious to hear what we're doing well, what you'd like us to do more of, and what you'd like to see from us. So we're here to listen. We're here to celebrate. We think at the end of 4 years, we have a great deal of success that we want to share with you. And we hope that you, at the end of the day, feel as good about how far we've come with this endeavor, beginning with essentially nothing at the beginning of COVID to where we are today. We think it's worth celebrating. We want to especially focus on those first patients who take an experimental medicine. Those of us who do drug discovery and development as a calling, which I think most people in our industry would say they do it because of a need to do it. We understand the extraordinary importance that the first patient who takes an experimental medicine, the position that patient occupies. We want to share, or I want to share with you a bit of why we want to celebrate what I'm thinking of as our pioneer patients and families. We link that this day is another opportunity to bring the community together and to create a truly unified, cohesive, empathetic, knowledgeable community. And we think if we couple a unified patient community that's unified in knowledge, then there's really nothing that we can't accomplish together. And based on what I've already seen, I think we may have already achieved that, but there's more to do. And though we are celebrating our successes, we're celebrating a beginning. And so there's a lot more to do. And so we need to get your support from every single person and every person you know. We can't do this alone. We haven't done it alone to this point, and the challenges ahead of us are larger. So we're asking for your help. We want to recall why we are celebrating. And it's about hope. It's about providing hope to the hopeless in real opportunity for help. So this day is about hope and the creation of futures where futures didn't exist. We'll be talking about futures. Fortunately for me, my job is a little simpler. I do want to give you a very high-level summary of what we're learning and what we're doing. And Lori, Julie, Lori, Amy, and Sarah will give you much more detail. At the end of the day, we want you to walk out of here knowing what we do every day and knowing the challenges we face and having a better understanding of the process that we use so that you can understand why the process that we put together is so important. There are some questions and issues that have arisen. I want to address a few of them. And of course, in Q&A, we'll have more opportunity to do that. I get the pleasure of celebrating our pioneer patients and pioneer families. And I'm going to ask that you join me in that. A little later in the day, I want to talk about reforms. Reforms don't happen easily. And there are some reforms that are desperately needed. And so we're going to want every single person to put your shoulder to the wheel with us to help us drive these reforms. So we'll be talking a lot about that. Really bringing the group together. When I began this, I realized that if you're an N of 1, there is no community. There is no one to turn to, no one to say, what's going to happen next? Where do I go? How do I get help? And one of the things I'm proud of is this room, that there is coming together a community of the Nano-rare, where none existed in 4 years ago. There is today, and there will be a bigger and more cohesive, more knowledgeable community in the coming years. So in the end, this is about a dream that was impossible. Everyone said it was impossible until we showed that it was entirely possible. And dreams are what we do. As a family of human beings, we dream collectively, and collective dreams can be turned to reality. That's what we're about. Everybody knows our mission, and that is to take advantage of the technology that was created under my leadership at Ionis to discover, develop, manufacture, and provide the highest quality possible personalized experimental ASOs to patients with nano-rare disease, and to provide those for free for life. What's really something that's very, very important to me personally and to everyone at n-Lorem is a commitment to equitable distribution of healthcare. Certainly, we want the services we provide to be needs-based, not means-based. So we are categorically uninterested in the financial means of our patients. We don't want to know. No, the application doesn't ask, do you have the money to pay for this? That's our job. We don't think patients and parents should have to be out raising money to be cared for, not in the 21st century in America. That's simply wrong. We do take what we call directed research donations. And these are really important to us, and we thank our directed research donors. They provide an opportunity to do basic work that we couldn't afford to do otherwise. They improve our infrastructure. And we've been tremendously impressed with the generosity of our directed donors, in that we asked them to provide donations to the general fund as well as to their area of interest. And they've all been delighted to do that. And I've been asked, do we prioritize patients? No, we don't. We don't. The only prioritization that we engage in is based on urgency of need. So if a patient is progressing rapidly to succumb to the disease or losing an organ, that patient rises to the top. Otherwise, we think all patients should be treated equally. All patients are suffering. All patients deserve to be treated. And n-Lorem is going to do it that way. We don't prioritize. I do think that we need to better parse patient populations, particularly down at the lower end of the spectrum, because they're different and they require different solutions. And I've defined nano-rare mutations as mutations that have a known prevalence of as many as 30 patients in the world. And there's nothing magic about 30, it could be 25, 35, but I felt that people needed a hard number. What really matters is in that number range, No commercial solution is available today. Also, if you think through it, generally, at the most, right now, 10% of a patient population will make their way to us in a way that we can treat. And we can afford, as a nonprofit, to treat 3, maybe even 4 patients for life with the same ASO. And so there's a logic to that 30. And these patients, and of course, all of you know this, have one unique advantage, and that is they have an identifiable, understandable, single gene mutation that is driving these enormous phenotypic changes from health to terrible disease. And of course, once we know the genetic cause, we make a genetic medicine, we have all the information we need. So many of our patients don't have named diseases, and we don't care. We know the cause of the disease. That's our job to fix it. Beyond that, though, nano-rare patients are extraordinarily disadvantaged. They're by definition the rarest patient population. We didn't know until genomic sequencing that such patients existed. Now we know. They are isolated, they're desperate. The vast majority of patients are never diagnosed. They progress in their disease, succumb without ever knowing why their lives are shorter, why their lives are less robust than the lives they see around them. And those who are fortunate enough to be diagnosed today are in for a long, perilous journey, replete with misreferrals, misdiagnoses, and far too often mistreatments that make things worse. We have to do something about that. And there is no other option today other than ASOs on an industrial scale for these patients. And so we're it. And we are pioneering a new model. And that means that every step that we take is a step into the unknown. But that's the nature of doing something nobody's done before. And I'm very comfortable at the frontier. We are at the frontiers of medicine and science. And that's a great place to be. In January of this year, and you'll be seeing quite a bit of data from this, we did a data cutoff. And at that time, we had processed 173 applications. 173 applications, that's 173 human beings with terrible disease, 173 families that are desperately seeking an answer. And so we have our own data now. And the average time in our patient population in January from symptom onset to diagnosis is 4.5 years, roughly. But what really matters is the range. And the range is 1 month to 36 years. And that tells you that these journeys are idiosyncratic. They're dependent on factors that patients and families have no control over. And that's something that we have to do something about. But those are numbers, and these are personal journeys. And so I will share with you a patient, our very first patient, patient we know very, very well, patient 001, his history. And this patient is typical of the patients we see every day, except that his mother very diligently compiled the most impressive natural history I've seen. Like most of our patients, patient 001 was a product of normal gestation, normal delivery, but very quickly the parents realized something was wrong. Patient was hospitalized the first time when he was a month old for severe gastrointestinal distress, for inability to feed, and then one of those terrible terms that we use in medicine, failure to thrive. When I say that, it sort of makes me cringe to think about what that actually means. He was worked up thoroughly for his gastrointestinal disease. No cause was found, treated with omeprazole, Tagamet, and the like, and sent home. 8 months, he had his first seizure. By age 2, his seizures were so intractable that he was placed in hospice care. And then, in common with a lot of our patients, his seizures declined. We'll talk about that quite a bit in the coming minutes. He's been hospitalized 29 times in 9 institutions. He's had 33 incorrect diagnoses and 1 correct diagnosis. As an infant and toddler, he was treated with as many as 12 to 15 drugs at the same time, almost guaranteeing deleterious drug-drug interactions. And he demonstrates something else that we see a lot, and that is there was an intense push to care for this child as an infant and a toddler. And then it seems that the medical system seems to weary or tire, I don't know. But we see these patients who then are basically not cared for. And he's to a large extent not been cared for by the medical community. He's now 13. His seizures are a minor component of his problems. His major problems are movement disorders, developmental delays, and he continues to have gastrointestinal issues. And we'll come back and talk about that as well. And so we see these things where phenotypes change. And I refer to that as phenotype drift. We'll come back and talk about that. His journey to diagnosis was far too common. His parents found, eventually, knocking on doors, investigator who would sequence him. They got the sequence information, didn't know what it meant. So then they found another investigator to sort out what that actually meant. That investigator turns out to be in Australia. And since we took this patient, it's been very challenging to find an institution or physician who would take care of him on an experimental ASO. And fortunately, Rady Children's at UCSD has agreed to care for this child, and we'll be treating him very soon. And so it's been a long, long trip, but we're thrilled to be in a position where we can begin to see if we can help this child. That's the journey, and we must do something about that journey. Okay. And Antisense is able to do this because of its properties. And I won't walk you through these properties other than to say one of the most important features of Antisense is that within a chemical class, ASOs differ only in genetic zip code that we use to target the site in an RNA. And so what we learn about one applies to all. And that is a critical piece of the puzzle for us. And the fact that at Ionis, we've treated certainly more than 250,000 patients. And so we have a tremendous experience that we call on to do this the right way at n-Lorem. But we couldn't do that were not for the cooperation of the FDA. And in astonishingly rapid order, they issued special guidance for ASOs for the Nano-rare patient population. And during the public comment phase, we made a lot of different recommendations, and many of those were adopted in this final guidance. And so it's that combination of technology and regulatory support that makes this all possible. And we believe that that is an incredibly important model. As we think about other rare patient populations, how can we put together a provider, a technology, and regulatory commitment to that patient population? We think this is a model that should be emulated. We are industrializing the process. That means that we have systems in place that you'll be hearing a lot about that assure highest quality at every step in the process, that establish a process so that we learn maximally from each patient. We owe that to them. And these are extraordinary opportunities to learn in the right model, a human model with a single variable. And that these patients are managed professionally while being treated with an experimental medicine. They are being treated with an experimental medicine. There is a responsibility that comes with that. And we are very committed to that. And of course, we know we can scale this if we can raise the money. Technology is fully capable of doing that. That's industrialization. These are the organs we're willing to treat. Why these organs? Routes of administration. These are the organs we know. We have extremely high potency, long duration, great therapeutic index, and we believe we can do it properly. On the right is the amount of drug per patient. And so making the smallest batch we can make in GMP is about 10 grams. And very often then that means that we can treat a patient for life with one manufacturing run. And that makes it economically feasible for us. This is the process that we have in place. I'm just going to touch on this process at a high level, but in subsequent presentations, we'll break down each step for you so that you understand at the end of the day, all the things that we do, why it takes time, and what the challenges we face, and why it has to be done this way. This is a table that I've shown in many presentations. It's published. it's the current screening process to identify an optimal ASO for central nervous system disease. And it is multi-step, it's complex, it's cumbersome, it's time-consuming, and it's costly. But the points that I want to make on this table is that every one of these steps is a minimum step that's a product of millions of ASO experience. If we thought we could remove any one of these steps, we would. We can't. This is what we need to do today. The number of ASOs at each point are minima that are a product of 30+ years of experience. If you cut a step out, if you reduce the number of ASOs at any step, you increase the risk that you have a suboptimal ASO. And that's something you don't want to do. And finally, over here is what we measure and the minimum criteria. And again, that's experience-based. These are the product of all the experience that we have with this technology. That's what we do. That's what we must do. And that's what we keep encouraging others who may be providing ASOs to do and do a more thorough job. Julie will be sharing with you each step in much more detail so that at the end of the day, you'll really have a much better understanding of that. The demand has greatly exceeded anything I dreamt about. I thought maybe by now we'd have a handful of patients. We just have processed more than 225 patient applications. We have accepted about 100 patients. So the simplest way to think of us is that we're a 4-year-old biotech company with about 90 drug discovery programs, 70 drugs in development. And in the last year, we filed 9 INDs. And we hope to file a couple more before the end of the year. And so put in that context, it's remarkable what's been achieved. And I suppose the only thing that separates us from other biotech companies is we're deliberately non-profitable. There are plenty of non-profitable biotech companies, but not by design. We are. These 9 INDs we filed are with 4 different divisions of the FDA, 2 neurology divisions, the eye and cardiorenal. That's important because guidance issues, but not always do the divisions follow the guidance. Here we have had strong support from all the divisions of the FDA, and that speaks, I think, to the quality of the technology and to the desperation of the need and the fact that the FDA recognizes it. We've had to expand much more rapidly than I expected to meet this need. And one of the things that I was really worried about when I founded n-Lorem is, will we be able to compete for talent? We're competing against biotech companies and pharma who can offer better compensation, RSUs, options, things we can't. But I have to say that the senior team we have at n-Lorem that you've met is as strong and cohesive a senior team as I've ever had. They're here for the same reason I'm here, the reason you're here, the mission. And so the mission recruits. It's pretty easy, actually. We have expanded our laboratory, brought in a new research leader, and that increases our capacity by at least 4-fold. And so as next year unfurls, we hope to work through this backlog that we have and be able to treat more patients. Today, our only limitation is the dollars we raise. And we couldn't have done that without the extraordinary outpouring of support. And this table grows every day. We just added Takeda and Honggenes as supporters and partners. And you can see it comes from a range of organizations and people and individuals. And again, I think that speaks to the mission. People want to see this mission achieved. And what we need at the end of the day is every one of you going out and talking to folks about our mission and how much we need everyone's support. We couldn't do it without you. The other big worry I had was, would we be able to help any of these patients since they're all likely so advanced before we get a chance to treat them today? And today we have resoundingly positive answers for that. And that's extraordinary to me in just such a short time. I'll show you two videos. Lori will spend some time talking with you about other potential examples of benefit that we're already seeing. This is Anna. She's Neil Snyder's patient at Columbia. He's here. Anna has a very aggressive form of ALS called FUS ALS. And she had progressed significantly before we could get the ASO to her. She was largely bedfast, required ventilatory support. She rapidly got better, went home, and then, as ALS patients do, as I've been taught, she had a swallowing accident, actually had to be resuscitated. And so the video that you will see here is of Anna after being resuscitated. Here's Anna. At diagnosis, after her resuscitation, after induction doses. Her golden retriever saying hi. Here she's showing recovery of fine motor skills, which the ALS experts tell me is really impressive. After another dose in the summer, climbing stairs slowly. She has to stop here to catch her breath. Another dose in the fall. Later in the year, ever stronger, able to rise and walk rapidly. And then this year, she's going downstairs. It takes different muscles and different coordination. She, in short, is alive, breathing on her own, walking, talking, enjoying her family. She has a future where none existed. And her family has a future that at worst includes much more time with their daughter, and at best, maybe a future without an unfillable hole in her heart. We think that's worth doing. We think that's really worth doing. So that's Anna. I want to talk to you about what we're learning. As I said, in January, we did a data cutoff. The database is heavily skewed to neurological patients, but we're now getting eye and liver and kidney cases, and so that's going to change. But just remember that as we go through here. Of the 173 applications, we were able to accept 78. We have 14 on hold. That means an application didn't have all the information we needed to make a decision. And so we're working hard to reduce that inefficiency. Over on this slide, side of the slide is an important pie chart. It's not simply diseases of infants and children. We have 48 of our 173 patients over 18, and our oldest patient is 69. So it's a set of diseases that happen at all ages. This is just a set of gene families. Not surprisingly, if there's a gene family that's important, it can have a nano-rare mutation and it can cause severe disease. Notice that we have 26 different ion channel mutations. And that just speaks to the fact that the brain is an electrical organ. And these patients share many things in common. And one of them is that many of them seem to have severe GI and GU issues. And to date, we see no evidence that anyone has asked, could there be a central component that's contributing to that, an autonomic component to that disease? We at least think we should be asking that question. This is the type of mutations that we see. Lots of toxic gain of function, some mixed, but other types of mutations as well. So we need to know what the mutation is, but we also need to know what kind of mutation it is, and we have to be confident the mutation actually is disease causing. This looks at the design of the ASOs, and not surprisingly, we have 53 of the 78 patients we accepted that require allele selective ASOs. That means they have a heterogeneous mutation. One copy of the gene is mutated, the other is not. The gene produces a protein that has an important function. And so we have to design ASOs that can reduce the mutated RNA and the mutated protein and leave the normal alone. That takes some work, but I think next year, I think we can learn how to do this much better thanks to some recent advances. And so next year, I'm hopeful that we'll be able to address that and begin to do a much better job with that. Some basic research that I want to do. We have 16 mutations where we have more than one patient with the same mutation. And we have 8 of those where we have sufficient phenotypic and natural history data that we can ask simple questions. Like if you have the same mutation, do you have the same syndrome? And not surprisingly, the answer is no. People are different. But we have now, for the first time, a genotype, a phenotype, and a patient that we can then begin to ask why. Here's 2 patients about the same age who have mutation, the same missense mutation in a signaling protein, an alpha subunit of the G protein. And you can see that they share a lot of phenotype, but one child is progressing toward blindness, the other is not at all. And so we now can begin to ask in these very patients, the patients who matter, human beings, why? What makes these patients different? This is a pair of patients who have mutation in a histone. Histones regulate a lot of gene functions, so you'd expect a very severe phenotype, and they do have that. But here you can see one of these patients actually has a lot of structural bony deformities. The other has none. Again, multiple examples of that. And we think that that becomes a true treasure of opportunity to begin to learn about health and disease and how health progresses to disease. And particularly as we treat and we have the opportunity to watch the reverse ontogeny of that disease if we produce benefit. And so these are extraordinary opportunities to learn, and our goal is to learn and to share with what we're learning. We believe that if with an optimal ASO dosed appropriately, we should be able to avoid severe adverse events and most drug-related adverse events. To date, as Lori will show you, we have no drug-related adverse events. And that's our goal. We cannot add side effects to the burdens that these patients already live with. So what did we learn? Well, one, we've learned that this journey to diagnosis must be addressed. We can't have this continue. It's wrong. We know that the systems that we put in place to assure quality are working, and we commend them to you, and we'll be teaching you about them as the day progresses. We know that our approach to understanding and learning from these patients is working. That's a challenge in a single patient. How do you actually design an opportunity to learn from that patient in an effective fashion? All age groups are represented. All gene families are represented. And we have, as we'll show you, meaningful evidence of benefit in some patients. We can't guarantee benefit. There's nothing we can guarantee. But to have already evidence of meaningful improvement in patients is a tremendously important step for the field. We know now that different mutations, the same mutation can produce different phenotypes. Different mutations can produce the same phenotype. And what we see is what I describe as phenotype drift, these changing phenotypes over time. We think that's important. And we think that there are things to be learned about why that happens and how it happens and what we do when we intervene. And that managing patients professionally while they're on experimental treatment is critical, particularly if you do have a surprising adverse or serious adverse event, to be sure to understand that and make that aware, make that apparent to the community as rapidly as possible is really critical. We're not simply interested in the patient. We know that patients should be considered holistically and that they're a part of families. And so we've done our best to begin to create a community for the Nano-rare. And that includes sort of the centerpiece is this podcast series we've done that I host, which has been great fun for me. Lots of very interesting interviews. And then these little lectures where I take people from what is a chemical on to the most complicated sort of biological questions. That's in response to what I see as remarkable achievement by people who have no scientific training. What could they achieve if they understood more? I'm told by people who know about this stuff that the response has been very good. Since I don't even text, it's hard for me to judge, but I'm told these numbers are fantastic. Yeah, I shouldn't admit to being such a Luddite, but I am. And we're sharing what we learn. I personally have given, I don't know, hundreds of presentations. We give presentations constantly. And we think the right way to share scientific information is in peer-reviewed journals. Here's our peer-reviewed publications to date. The manuscript on the 173 patients is submitted. And so I'm hopeful that in pretty soon, you should be able to look at that manuscript yourself and learn what you want to learn from the work that we're doing. Last week, we announced a partnership with St. Jude that we think may be transformative, or certainly a partnership model that could be transformative for our patients. All of you know St. Jude as a great research facility that's pioneered the advances in treatment in pediatric cancer. And great news for many of you, St. Jude has now added severe pediatric neurological diseases to its portfolio. This partnership includes us preparing ASOs for 2 patients at St. Jude, but it's open-ended. We could do more. It also includes us training St. Jude to discover and develop ASOs as we do, as we've learned at Ionis. And we think that that's a model that then we can export to others. And that can get even more patients treated with high quality ASOs. And so it's a model that we hope to export to other organizations as a way of leveraging what we know and what we're learning and getting more patients treated. One question that I've been asked is, are you really justified in treating a single patient? Is all this effort really justified? That's a sort of an unsophisticated way of saying, what's the return on investment in what we're doing with patients like Nano-rare patients? I think the return on investment is going to be really quite extraordinary. First, if you think about the number of patients that we suspect exist with Nano-rare diseases, certainly millions, just reducing the burden on the healthcare system of those patients is dollars saved that can be invested in other patients. But I think the single most important contribution will be what we learn. And as the tip of the spear to drive reforms. If you think about these patients, every year we in the industry spend billions of dollars developing and using animal models. And there's not a soul who does that, who doesn't understand that every animal model has a flaw. And the flaw is, of course, it's not a human being. Here we have a group of patients who have the perfect biological experimental system for us. They have a single major variable introduced that's driving these enormous phenotype changes, and it's a human being. And we can fairly rapidly get to that patient with a genetic treatment. This is an extraordinary opportunity to learn. What we're already learning is incredible. What we will learn will change the way we think about health and disease altogether. And that return, just that return alone, is going to be an incalculable victory for humankind. All we need to do is see it to the finish line. And in addition to that, of course, we can be a tip of the spear to drive reform. Treatment is always the tip of the spear that drives reform. As Chris said, there was no genetic testing for SMA until Spinraza came, and you see this over and over again, and then you can genetically test. And I'll come back and talk about that. So what we see from n-Lorem, and what we're going to ask of you, is that you work with us to make n-Lorem the tip of the spear that drives needed reform in funding, in healthcare delivery, so that nano-rare patients are not left behind, not forgotten. So we think that the return on investment for n-Lorem is going to be extraordinary, and we stand behind that. Honoring patients and pioneer patients and families. One thing that the main point that I want to make is that advances in therapeutics are incremental. I mean, they occur in tiny steps over decades, often over centuries. Think about cancer. Cancer has been a terrifying scourge of humankind since human written history, and probably prehistorically as well. But we can pinpoint precisely the date on which cancer therapy began to change. It's 1946, a paper by Goodman and Gilman and colleagues. And in that paper, a gas that was used to kill troops in World War I, mustard gas, these authors noticed that it caused bone marrow suppression, and they made an analog called nitrogen mustard, and reasoned that maybe if they could kill some leukemia cells in the bone marrow, they might make some leukemia patients better. They reported their results. There were a few patients who had transient benefit, really very transient, and most of the patients had terrible toxicities. And over the decades that nitrogen mustard has been used, it's caused incredibly terrible toxicities in many, many patients. But that single step opened the door, opened opportunities for funding, opened eyes, opened brains to think about, well, maybe there's another way we can help here. And what we've witnessed over the last 80 years is incremental progress in cancer treatment and cancer diagnosis and cancer surgery, driven by that first observation, those first pioneer patients who took a risk and took an experimental medicine and really did take one for the cancer team. And even today, after 80 years, it's perfectly reasonable to say that gastric cancer, pancreatic cancer, colon cancer, biliary cancer, bladder cancer that's disseminated, therapy hasn't really changed. And so we have another 80 years of incremental improvement. None of that happens without the pioneer patients who are first. And so pioneer patients hold a very, very special place in the lexicon and the heart of therapeutics, as do their families. Same for cardiovascular disease. You know, the first cholesterol drug was 1960. It's called MER29. Unfortunately, it just caused terrible side effects and made cardiovascular disease worse. But now all of you take your Lipitor. That was a step. And every disease of every size, of every type, improves incrementally. And it will be the same for nano-rare diseases as well. Spinraza is a particularly germane example for this audience because SMA is a genetic disease, and we designed Spinraza as a genetic treatment to correct that disease. And still, Spinraza advanced treatment for SMA incrementally. First, we had to begin by treating the most severe form of the disease, type 1. Untreated type 1 SMA, average time to succumb to the disease or require permanent ventilation, 6 months. I think about the parents who had to make that terrible decision of putting their little sick babies into our first clinical trial with Spinraza. Had they not made those decisions, there would be no treatment for SMA today. Pioneer patients and families. And then we moved on to a slightly less severe, not immediately fatal, but catastrophic type 2. Then, armed with that information, it became obvious that genetic testing was appropriate. The guy got incorporated, and then Biogen was able to begin a study of treating patients before they became symptomatic. And today we know if we treat pre-symptomatically, many of these patients do extraordinarily well. And after that, more drugs to treat SMA. and second-generation Spinraza coming. This is the nature of therapeutic advances. It begins with a single patient, a single idea, a single experiment, a single willingness and courage and conviction to treat. And that's what pioneer patients do in n-Lorem. And so we honor our pioneer patients and families. They open doors. They help us then do better with the next patient, help us understand how to better evaluate what the benefit that we're making. They provide critical information that's going to teach us about that mutation, that disease, but will teach us about disease in general. And so this is a day when I want you to join me in honoring and admiring our pioneer patients and their families. They truly take one for the team. They take one for all patients with all diseases. And thank you. They are the real heroes. They are the real heroes. But in a very real sense, every one of our patients is a pioneer. We're at the frontier of science and medicine here. So every N of one patient, every nano-rare patient who participates with us is a pioneer and is leading the way to advances in understanding disease and health that only these patients can teach us. We are so privileged to be able to serve such courageous patients and families. Heroes. They're the heroes. I promise a second, and of course, many of you have seen this video. This is Susanna. She has a mutation in KIF1A, which is an adaptive protein that's involved in trafficking endosomes down axons. And if you can't do that, then your axons don't get the material they need, and they get sick. Susanna had progressed before treatment to the point where she was largely wheelchair fast and nonverbal.
I'm ready, Suze. Oh, wow, that was. Yeah, and then what?
And then when
Oh,
my goodness.
see?
Look at that.
get up, you know, when
Oh,
you
that was
I
amazing.
like that stuff though, but. Again. Boom, yeah, ooh.
Dad, come on, I'll teach you how
Okay.
to stand
Teach
up.
me how to stand.
She's a girl. I mean, she's going to teach you how to stand up, right? And oh yeah, now she's playing ball. So Susanna is Wendy Chung's patient, formerly of Columbia, now at Harvard. You know, I'm not a pediatric neurologist, but to my untrained eye, what impresses me about Susanna is she seemed to be improving in multiple domains. And, you know, she has a chronic pain syndrome, difficult to measure, but her parents, who are the best observers, are confident that that's improved as well. And Lori will show you other examples, but to have any example in having just treated a half a dozen patients long enough to have evidence of benefit, I think is an extraordinary statement. And it's a statement of hope. It's a statement of futures created where futures did not exist. So that's what n-Lorem is. It's about creating futures. So I think that if we can come together as a unified, knowledgeable, committed community, where our patients and families are united, together with the great people that we have at n-Lorem, our extraordinary partners and donors and all the supporters we have, there's really nothing we can't achieve. And I do believe that we can change the world, one patient, one family at a time. I commend that journey to you. Thank you very much. I think I'm supposed to take questions now, right? Have at me.
If you have a question, please raise your hand.
Be gentle. Roseanne hits me all the time, and so I'm pretty toughened up, but try to be gentle.
Good morning, Dr. Crook. In talking with you, you had mentioned that with some additional base research, or you may be able to benefit and accelerate these programs. Can you give some more detail on that, please?
I think the thing to remember about antisense is that it's still advancing. It's not a static technology. And what we're learning about RNA every day and the various kinds of RNA and how translation is regulated and 1,000 other things are presenting all kinds of new opportunities to do it better. And so one of the examples that I've mentioned is the opportunity to do a better job of responding to the need to have allele selectivity. We've been doing that for a long time at Ionis and n-Lorem, but I think there's now a whole set of new knowledge that if we can incorporate and just have the support to do that basic research, we'll be able to do a better job with that. That will mean better drugs faster for 53 of our 78 patients that we accepted in that first group. But there's a whole bunch of other things that I want to do, a lot of different basic research that I think will advance the technology while we're advancing our understanding of why disease happens. And so we are committed to doing that research as well. And that just takes money. I think we have the ideas, we have the experience at least, and so that's stuff that I hope to be able to afford to do in the coming years. Antisense is still advancing. There will be new opportunities to expand its utility and make it a better technology for all patients, including the nano-rare patient. Did that answer your question, Tom? Thank you.
Hi, Dr. Crook. Thank you so much for everything you and your organization do. So maybe building on that last question, how do you see the kind of spectrum of different ASO approaches that are being developed, and kind of what are you prioritizing? I saw upregulation earlier on a prior page. How do you see some of these new ASO approaches developing?
Well, as you know, a lot of those mechanisms are publications from me. And so I'm very excited about them as a scientist. But what I'm excited about as a physician is the open opportunities to help more patients. And we have a lot to learn to make those more robust and more versatile. But what's happening right now, for example, in just how translation is regulated, it's almost every week there's a new paper with a new advance that opens up opportunities in my head for new designs of ASOs. So I'm very excited about it. And in the background, I want to do some of that work. And then, of course, we're a patient-by-patient organization. We don't treat populations. We treat the next patient who applies. And if that patient needs a new mechanism that we've actually had the opportunity to learn how to do, that's a patient that we can then help. And then we can learn about the technology from that patient because that's the right model. So this technology, I mean, my goodness, if you're not excited about what, you have to be dead, right? I mean, come on, it's an incredible set of advances, but it's still at the beginning. And with n-Lorem, we have the opportunity to advance it in the human condition. And to change the human condition while we're at it. Listen, this is a privilege. This is a privilege.
So Michelle Sloan, Tub 4A. Question, within the nano-rare spectrum, within our condition, our disease, there's like multiple variants within the actual mutation. To qualify or to be part of a 0 to 30, are we looking at the individual variants? And then my next question is, for the longer term, is this something that can be used for the larger population?
There's two important questions. First, we're a mutation-directed drug discovery program. So it's not how many patients have the disease, how many patients have the mutation. And what's being learned in ALS is going to be learned by about every rare disease. And that is, there may be a fairly common mutation, and then there will be a range of ever rarer mutations. That's the way it's going to play. That's the nature of biology. That's just going to happen. And you know that tail end of that curve is our venue. And as Chris said, it is almost always the case that when you have treatment, whatever prevalence you think existed turns out to be larger. And we do have plans for how to handle that. And it's all stuff I've done now for a lifetime. It's stuff I know how to do. But basically, we hope that we'll be able to take what we have and find competent, committed partners who could then develop for the larger patient population commercially. So I think there are many paths to helping more. And this is just the beginning. But it's like any other exploratory process. Is that you move along and then you find new paths and new ways. And so what we're going to learn, I think, is unpredictable, except that we will learn it, and that will create new opportunities for more patients. You know, it's core of discovery. It's core of discovery. I should probably ask if there are questions from the 116 or anywhere else. Lori, any question I should take?
Sure, we can take one question here. The question was, do you envision a library of ASOs in the future that will be available for patients across the world?
I'm sorry, can you speak louder? I'm old, I have a hearing problem.
All right. Do you envision a library of ASOs that will be available for patients across the world eventually?
Yes, I hope. Our goal is to expand outside the US. Again, it just takes time, money, and we're certainly working toward doing that. In many ways, central payer organization countries, which is every country except the US basically, are going to be easier for us to engineer reform in than I think the US will be. So I'll be talking about reforms later in the day, and I'll warn you that reforms don't happen easily. They only happen if you're willing to really work at it. But we'll talk about all that later in the day. Go ahead.
We have a number of patients with gain of function or amenable targetable mutations in muscle or bone or cartilage phenotypes. Do you think it's a near-term future where these are deliverable to those tissues, or will they take a different approach?
No, for n-Lorem, it's not going to happen in the near term. There are many, many programs. One of the areas of work in the oligo industry, as it were, is to identify ligands that can enhance delivery to difficult to tissues that don't want to take up ASOs. One of them is skeletal muscle, the other is heart, and there is real progress there. But that's going to take some years of experience before then n-Lorem would be able or willing to contemplate treating nano-rare patients with those diseases. So that's certainly happening at Ionis. It's happening at every other ASO company that I know of. And with all that quality research, it seems that over the next few years, we should see some real progress there. But it's basic research. You know, our real problem tissues are heart, skeletal muscle, many cancers for some very complicated regions, and some immune cells are places where we don't get ASO at a dose that we're comfortable using. So those are challenges for the technology that are being actively worked in every ASO company.
Hi, Stan. Dan Curran, good to see you. And thanks for all you're doing for patients. I had a question actually about whether nano-rare can actually turn into ultra-rare and maybe even rare someday. Yeah, I think about, Tim Yu's paper over the summer in Nature, where they took a cohort of AT patients, and he identified at least potentially 10 to 15% of those patients would be potentially eligible for an antisense therapy. And I wonder if you've had any findings in any of the patients you've treated to date that would suggest that what was an N of 1 may turn out to be an N of 30 or even N of 50 over time.
Well, it's already happened with FUS-ALS, and that's being developed commercially by Ionis. So yes, we fully expect it to happen. But I think the challenges change as you go up in numbers. And I think there's a lot of confusion out there about what the impact of different numbers of patients actually is. And the real impact, of course, is when the development decision has to be made, because generally you discount at that stage 90%. If you start with even 1,000 patients or whatever, you just end up with a market that's too small to justify investment. So the right solution for that, I believe, is clinical proof of concept, which we hope to do. And then if we have clinical proof of concept that says, in a few patients that you may be able to help this disease, then I think you can probably find commercial partners that will develop it in a responsible way. But that remains to be done. John Maraganore and Lynn Parshall and I invested a lot of time in developing a venture philanthropy model for the next size up. And we really didn't get any traction. We really thought it was a great solution, but we didn't find a lot of investor interest. So I'm not sure why. One thing, of course, was we were all busy and didn't have the time to invest. So at some point, I want to come back to that and consider that. I think one of the reforms that we need to think about driving toward in our country is some form of cost plus model. I think that's where the long-term solution might be, where to reduce the cost of development, that the provider commit to a price that's set, that allows reasonable return on that investment. I think they're just what I think is that reform happens incrementally, just like everything else. And n-Lorem is the beginning of a process of reform that will encourage thought processes, and people, much more financially savvy than I, will come up with models that will work better. We cannot continue with the single model that we have today. We can't continue to raise prices. And so somehow there has to be a solution that will come, and I think that will happen because at least in this patient population, we're showing at least one way. Does that make sense? Chris, I don't know, Chris knows way more about this than I, so ask Chris.
Come back to it when I do my thing.
Okay, he'll give you the real answer. Okay, actually, there are several people in this audience who know more about that than I do. I'm just a poor, humble scientist. Nobody's ever called me humble.
One question.
Sorry, I'm not a scientist, so I think I'm going to ask a more practical question from the heart. And I just am wondering if, and I have the answer to this actually, but I'm wondering if, Stan, you know how much that you and your team at n-Lorem mean to our community and our families, because I can say with complete certainty that our children would not have any treatment without what you do. So I hope you all know that, and that's my question. And thank you.
I do know that. You know, I've said it, this wasn't what I wanted to do at this stage of my life or career. And I've said that, like every other phase of my life, it succeeded in anything I knew to dream of. It's a privilege. It's one of the great privileges I've had in a life full of privileges. But I do know it. And we all know it. And that's what motivates us, makes us come to work. So what better privilege could we ask than to be doing something where we bring the best science and medicine possible to the service of desperate human beings in need? How could I ask for something more? Thanks, everybody. Appreciate your being here.
We're gonna take a quick break. We'll start again at 9:45. All right. Everybody, welcome back from the break. Has everybody been practicing colloquium? Colloquium. We have a whole day of panels and presentations. I'd like to welcome our first panel up for the day. Helping n-Lorem do more as a nonprofit. We get support from various different sources, from various different groups, and it allows us to do what we can do today. And so this is a very special panel. I'd like to welcome Natasha Gassenberg up to lead our discussion today. She's the head of corporate affairs and chief communication officer at Biogen. Thank you.
Thank you very much. Maybe the other panelists can join me.
Maybe up here.
I don't want to stay alone here. So one patient at a time, changing the world. So this is what Stan said yesterday night, and it really resonates with me. We try to also, we are pioneer at Biogen, and we cannot be more honored to host you today. You know, when I'm thinking about, Sloan, Anna, Margot, Connor, Susanna, Lena, so many of those patients, for them n-Lorem is the only hope. And during the panel today, we are going to discuss how we can sustain that hope. How can we continue to contribute? What does it make sense for us as industry to be part of this vision, to be part of this project, but also nonprofit. So I'm very excited. I'm joined by different people with very different background. I will start with the people from the industry. So Dana Crossi, you lead the Nano-rare Foundation, and Dr. Dan Curran, you're at Takeda. You are the head of rare genetics and hematology therapeutics there. And Kurt Anderson, you lead, that is going to be a hard one for me, the genomic medicine at Cytiva.
Yeah.
Okay.
Kind of.
Kind of.
Better than this, but that's okay.
And you will provide us with an industry perspective. We have nonprofit, with the Wolverine Foundation and Amy McCoy. And of course, a patient perspective, but also an entrepreneur perspective. And I think you're going to help us think a little bit out of the box. So my first question to all of you, and I will start with you, Kurt, is what attracted you to n-Lorem as a company? Why did you decide to partner to this organization?
Okay, thank you very much. Very privileged to be here representing Cytiva and Danaher. So it really started early to mid 1990s when Stan Crook's company Ionis and Cytiva formed a very fruitful collaboration. And what we did was to provide instrumentation, DNA RNA synthesizer, which was used by Ionis to make a new type of therapeutics, synthetic oligonucleotides. And over the years, we developed lots of special relationships. So in January 21, Max Moore contacted us, me and actually a colleague from Sweden, Carina Anderson, and wanted to explain the vision of n-Lorem and Stan Crook's dream, which is to provide free-for-life treatment for young patients with nano-rare illnesses. So that kind of came very close to heart for me and Carina, so we felt we must do something or whatever we can. And the next step then was to figure out how can we get funding for all this. So we got in touch with our donation guru, Ryan Walker, and he presented or showed us the procedure that we have in place, which is a very intense procedure. It's 5 minutes of presentation in front of the donation committee, 2 slides, basically cram in very basic what you want to do, but still to make it very impactful. And then there was like a few Q&A from the donation committee, and they were deciding on the spot, and they decided to approve. And this was in May 21. And in June 21, we were happy to share the news with n-Lorem. So that was a great event. The continuation of that, we of course wanted to continue to give. So in 2022, the awareness of n-Lorem grew within Cytiva and Danaher, and very much thanks to Ryan Walker. And we were able to give more donations from different groups within the company. And currently we're working on 2024 donation. So that's kind of what attracted us, and we really appreciate to be part of it. Thank you very much.
Thank you, Kurt. Then I know you're trying to convince the foundation board to partner with n-Lorem. So I'm just curious about what are the arguments you are building and how what n-Lorem does connect to your mission.
Thank you, and thanks for having me here today. I think, expanding on what my colleague Kurt has kind of just touched upon, really the relationship starting with the Cytiva and product donation, it was an easy introduction and relationship to build with n-Lorem, given the mission, right, that we're all familiar with. And Danaher recently undergoing a separation of its water quality and product identification platforms into its own publicly traded company, allowed both Danaher and the new company Veralto to really expand in their areas of interest. This allows Danaher and gives Danaher the opportunity to focus on being a science and technology innovator in the space. And so with that, I think focusing on those underserved populations in a critical need area like the N of 1 and rare disease patients is of critical interest. And so aligning with the business, the areas of interest, and our foundation strategy from a philanthropic standpoint, really excited us to be able to kind of start that conversation and hopefully expand beyond and enhance the current partnership that Cytiva has grown over the past years to be able to do more and have more of an impact.
Thank you. Dan, so you're a scientist, you're leading the business there at Takeda. So what you decided to do, and I know you are a significant partner for n-Lorem, so I think you're called a hero. So can you tell us why you decided to invest in n-Lorem?
Yeah, certainly. So first, thanks for hosting this event here at Biogen. It's wonderful to see so many faces of people, of patients and advocates for rare diseases. So I lead the rare disease efforts at Takeda. Takeda's had a long and storied history developing medicines for patients with rare disease. But note I say rare disease. I don't say ultra rare, and I don't say nano-rare. And we can't do that simply because of the commercial interests in a large organization. However, we see the incredible need that patients have, their families have, and we know the technology exists to in fact treat these patients. So we felt it was our duty, we were compelled to say, how can we help support these patients? You can't work in rare diseases and then look at certain patients who perhaps just because there are too few of them in the world, you can't try to provide some help for these patients. And so we were really excited, obviously, to be supporters of Stan and the n-Lorem Foundation, because you're doing such incredible work. So
I'd like to come back to the point about incremental innovation. And when we prepare the panel, we talk about that. So of course, we're doing that for the foundation, for philanthropy, but there is a benefit to the science. And I'm just curious, I'd like to hear a little bit more about how do you see what n-Lorem is doing, potentially breaking new ground for more research or more advancement that can become them of interest for the business?
Yeah, I think there are a number of ways. And I think it's so important to, Stan and I were chatting, to preserve these samples and be able to have access to these samples broadly for use for discovery purposes and for translational purposes. So in industry, obviously, we look to try to tackle diseases where we believe that we can transform the disease through whatever technology platform, whether it be an ASO or a gene therapy. But we critically need to have validated markers to try to understand why these diseases are progressing, how they're progressing, what's the clinical phenotype. So there's such a treasure trove of information that's going to be found here over the course of the coming years and decades that industry, I'm quite sure, would be very interested in having. So that might be another source of actually raising some capital in the future for the n-Lorem Foundation. But it also, as I think about the approach you're taking with the regulatory authorities, giving them the opportunity to see that ASOs are safe and well tolerated. And if you can give these ASOs to patients for years and years and years, that helps us in the industry say, look, there are opportunities here to have dialogues with a regulatory authority, whether it be the FDA or the EMEA, to say, this platform is a safe platform. So it may lower the barriers for us in industry to tackle ever rarer diseases because we don't have to necessarily do long-term studies. We can use the data that's been established by n-Lorem and potentially others to essentially hopefully ease the way, if you will, to develop these life-saving therapies.
Thank you. Amy, I know you have also perspective from an early research perspective, so maybe you can share, why your organization is partnering with n-Lorem.
Initially, Stan had us at hello, if you can still say that. We we came in, we met Stan, and that was it for us. But it's really interesting. I just want to touch on what you were just saying, because what n-Lorem has been able to do, and I think they do this with a lot of patient family foundations and organizations, is we did have incremental research, and we had this basic research that we didn't really know what to do with. And n-Lorem was really able to help us pull it together and move forward and provide a solution. And that's what we were looking for. So that was one of the key reasons that we went with Stan and n-Lorem, and have been, really, it's been an amazing experience.
You're a patient, family member. I think you have spent the last 2 years taking care of your granddaughter and helping n-Lorem. Can you tell us more about your personal journey with n-Lorem and why you get involved? Because you can just be a passive grandfather, and you certainly are not.
Well, when you get the diagnoses, those emotions are pretty intense. So trying to figure out what to do is where you start. And for me, I have a business background, no science background at all. And so it wasn't really an option to go out and try and learn the science and see if I could help in that way. Once you get into this a little further, you begin to understand that you can't do it yourself, and that what's good for the community is good for your daughter or my grandchild in this case. So figuring out ways to help the community, and that would really revolve around funding. That is something that everybody that's interested or impacted by this can do. And I thought you had to go out and solve the world, and they're like, look, everything helps. So figuring out everything you can do to help generate funds, because every dollar is, somebody else that can be helped. Figuring out what level that you're going to get involved in is really what I've been trying to do, figure out what my lane is, because some people have certain amounts of time and other obligations, and other people have more time. I'm retired now, and I'm able to dedicate more time to this. So figuring out the level that you can participate is that next step, and working on both the bread and butter donations, and then trying to come up with some bigger programs to help is really what I've been focused on. And it's out of gratitude for these incredible scientists and people that are dedicating themselves to helping my granddaughter.
I'd like to stay with you for a minute, because I think you clearly have big ideas, and we talked about that, yesterday with Dr. Gorgon from Biogen, and I'm sure there's people maybe in the room that would like to brainstorm with you. So, can you share your big idea?
Well, these again, no scientific background, and so my immediate thought is you've got to come up with this solution at 30,000 feet, and Dr. Cook is no. Don't do that. Here's where you need to start, and that type of guidance is what is important. To start with that doesn't mean that you can't come up with additional ideas, and I'm trying to move into those ways, because funding has been figuring out adequate funding is challenging for rare disease, and maybe personalized medicine in general, but trying to understand where that can go and understanding the possibilities and who are the downstream benefactors from this, and trying to come up with some type of approach to get those people involved on the front end, as opposed to waiting until this happens, and that benefit to the downstream folks that can use this technology is what I'm starting to think about. But again, my focus right now is on the bread and butter stuff, with looking at trying to do things like create some type of an association or a public benefit entity that where these downstream benefactors can get involved up front to help fund this technology that's going to help potentially a lot of people. But other things, maybe there's the ability, for me, I'm looking at how, who in my community, and trying to define that community, whether it's GIF1A or Nano-rare or Phoenix or whatnot, figuring out how to explain the benefits of what n-Lorem in particular is doing and how that can benefit your community, and getting those folks involved up front. I mean, people need to be chasing Dr. Crook and n-Lorem, not the other way around. And we just need to explain that and come up, let the creative juices flow, and maybe come up with nano-rare insurance policies, or these public benefit fees, or other things like that, to try and come up with the broader dollar base that can help more patients.
Certainly, and I'm sure everyone in the room is potentially an advocate for n-Lorem with their own organization. I'd like to, of course, funding is the essential, and we need to sustain the model of n-Lorem, but there is other ways to help. And I'm just curious if some of the panelists want to share how they're helping beyond funding.
Yeah, I think from from my point, we're continuing to spreading the awareness of n-Lorem within our organization. And like I mentioned, we pushed it all the way up to corporate. And I have lots of discussion with Max Moore when they need some stuff, and we figure out how can we do it. And so that's kind of on my personal level.
Yeah, I do think you donate some equipment.
Yeah, we have the DNA RNA synthesizer, the actual machine that makes the oligonucleotide, and then that gets taken care of with other equipment to purify it and all that kind of stuff. So with the addition of the new lab at n-Lorem last year, that's where we got other groups involved, so they could donate lots more consumables that they would use on a daily basis. And that's where we hope to expand with 2024 donations, and actually give another synthesizer as well to increase the capacity or the development work at n-Lorem.
Dana, do you want to comment also? I think you're thinking also various ways to help.
Yes, exactly. I think this is the ideal situation to for Danaher at the enterprise level, being introduced to n-Lorem. The mission is so closely aligned to the work that we do as a science and tech company and diagnostics. And I think having the opportunity to work so closely with our operating companies, Cytiva and others, to identify where there are opportunities to support n-Lorem and or any other organization right in this space at the enterprise level, now that we are supremely focused in this space, is identifying these more smaller scale local partnerships and expanding them beyond just what Cytiva is doing in product identification, but are there other players in this space within our family of Danaher that can contribute and enhance the work that they're doing. I think from my perspective as working for Danaher and the Danaher Foundation, we're talking about philanthropic dollars essentially from the foundation. And so really what was critical for us, and we've been chatting with n-Lorem for probably the past year, just to identify what are those needs and trying to find that synergy and alignment for where, our areas of interest may align with theirs. And I think that's probably the case across the board, whether you're supporting as a donor or partner in any capacity. And I think that's what's been very rewarding too, is there's lots of needs, which I know have been mentioned, but really identifying potentially, and creative ways and solutions to provide additional support that can have impact that perhaps you're not aware of quite yet. High level, we understand what funding is needed, but funding can come in different forms, product donation, cost for consumables, things like that. So it's a really exciting time to kind of continue to build the partnership and have these conversations with n-Lorem.
Thank you. Dan, I'm sure you have additional input to that.
Yeah, no, I think certainly speaking for Takeda, but I think more generally, broadly throughout the industry, I think many ways that we could help n-Lorem is just the interactions that we have with our organizations, certainly with the regulatory bodies, right? We're constantly speaking. We have relationships, so they're arm's length relationships with many of the regulators within both the rare disease groups as well as more broadly. And to me, it's critical for us as an industry to try to help push the boundaries, because it's not only going to help n-Lorem, but it essentially will help our businesses as well, as we think about just the regulatory interactions. I think the other thing we need to think through is our public policy efforts. How can we, through our government affairs groups and our public affairs groups, say this is a real public priority? I mean, we speak about nano-rare diseases, but as you go through it, every rare disease is completely underdiagnosed. We know this. And so as the n-Lorem organization continues to identify these patients, continues to treat these patients, the numbers are going to grow from tens to hundreds to eventually thousands of patients. And as a matter of course, you think about this at the public policy level and say, we have thousands of patients being impacted by disease. It's a necessity to say, how are we actually going to pay for these patients? And I think we need to get the insurance companies involved as well. I mean, essentially, n-Lorem is providing them free care and saving all of us quite a substantial amount of money, especially the insurance companies. So the question would be, how can they become good partners in this organization trying to help? So I think there are a number of ways that we in the industry can actually use sort of I won't call it our clout, but at least some of our input.
Some kind of collaboration, totally.
Yeah.
And I'm looking forward actually to the presentation about the reform that are needed, but also other advocates, groups like NORDS can potentially maybe also be more active on the nano-rare space, because n-Lorem, you need the community also in DC. So certainly an area where I think the industry can help through our network. Hemi, do you want to add something on that point?
Well, I would just touch on something Stan mentioned this morning. Antisense is improving and the technology is improving. And so we really need to keep steps ahead with the research. And one of the things that we founded as a foundation, and I would think other patient groups and foundations are looking to do, is to really invest in that basic research and help n-Lorem invest in that basic research so you're keeping up with the pace of change.
Yeah.
Do you think there is enough involvement of the academic world? And we talked about that with Dr. Krogan yesterday. Like, do you have enough support from university? Like, not because you have the industry, we have the business side, but like more university can help n-Lorem.
Absolutely, that's a critical line of communication. We've had a great experience, and I think if you can bring everyone together in that kind of communication, then you're bringing up all ships at once. So absolutely.
I mean, and this is like translational research and translational development at its finest, right? So this should be part of the mission, not just of academic centers, but as you think about the National Institutes of Health and NCATs, I mean, this should resonate with them deeply, right? I mean, this is, I think, one of the missions that they should be aspiring to help n-Lorem organization, because it's in essence just basic translational research and basic translational development, which can be utilized beyond the particular disease that you're trying to treat.
So I have a final question: is how are the people in the room or the people that are watching the webcast? You give them a lot of ideas, but how can they help? Like, how much more can we do? Do you have ideas? What would you encourage them to do?
Every dollar helps is what I've come to understand. And you don't have to solve all the world's problems to help n-Lorem. Coming up with ways to get your community, whether that's your local school or your local hospital or folks in the healthcare industry in your community, to help n-Lorem in their mission, which I think the explanation needs to be made that in helping them, you're helping your own community. And I think that's where this needs to go, and not just on even though they're nano-rare, they're very clear about that, but the benefits are dramatically beyond that.
Potential breakthrough that can come.
Yes.
Amy?
Ditto.
Love
it.
Do you want to, other ideas?
What else can we add after that, right?
I think it's just to continue advocating. That's the biggest important thing, I think. Make people aware and get in there and help out as much as you can to whatever capacity. I mean, we're so different people from each other, but we're still helping.
Thank you. Thank you very much. Thank you for your time. I think it was great. I hope everybody is inspired to continue to help, to do more, to spread the voice, and to help n-Lorem to gain more advocates. So thank you very much for your time. It was a pleasure.
Thank you. Thank you.
So many wonderful people coming together to help Nano-rare. So today is the community of care for Nano-rare. So all of these panelists up here are just very committed and engaged with the community. And so I'm appreciative of their support. Our next panel is how do we expand? So we're committed to treating the patients that we have today, but we want to also invest in the long-term solutions that we can reach even more patients. And so I'd like to welcome the next panel up here. It's going to be moderated by Sarah Glass, our Chief Operating Officer.
Great. Well, thank you so much, Amy. We're thrilled to be here. And I'm really excited for each of you to hear from this, fantastic group of individuals. They become friends, colleagues, partners, collaborators. I think I've gotten to know all of you so well over the last couple of years, and just thank you for your partnership. And so really, I think what we've talked about a little bit today is trying to align on missions and goals. So ultimately, the connection here between even though these individuals represent very different organizations and not necessarily connected with one another, we all have in common these very specific goals to help more patients. And so really just wanted to spend this time focusing on how we're working together and sort of what are your ideas about partnering, about expanding, about being able to identify optimal ASOs and help more patients. So I'd like to start off with sort of a general question to each of you. Maybe Annalisa, we'll start with you. Oh, I forgot the introduction. Oh my gosh, sorry. So let me introduce the panelists. So first we have Annalisa. So Annalisa Taylor Tavares, she's the clinical lead of rare disease research at Genomics England. So we're very pleased Annalisa came over last night, so really appreciate that. Kristen Stevenson is the director at St. Jude's newly formed sort of pediatric translational neuroscience initiative and treating pediatric patients with rare neurologic diseases and expanding and just helping many, many pediatric patients. We have Neil Schneider, who's really representing the Silence ALS Partnership. And so Neil is a clinician scientist at Columbia University. And I think a lot of you know Neil from the FUS ALS patient that we saw earlier from Stan, and also just a general great guy and leader in ALS. And Terry Jo Bichelle, thank you so much, Terry Jo, for coming. She flew in this morning as well. Terry Jo is the CEO of Combined Brain and a neuroscientist. I mean, this is an exceptional panel, right? And ultimately represents over 50 advocacy groups, and here to really think about how can we work together to make those groups aware of what we can potentially offer. So now we'll get to the first question. So I think I just wanted to really touch on, given n-Lorem's a small organization, and we fully acknowledge we're doing everything we possibly can to increase our specific capacity. We're partnering. I think in the last panel, we heard a lot of just amazing support that we're getting, how we can internally increase our capacity and continue to grow. And we acknowledge that we are one organization and we are small and we are limited on our resource. And so therefore, how are we really working with each of you? And what are your thoughts on how can we help more nano-rare patients together? So Annalise, if we might start with you, kind of on your thoughts on that.
Yeah, well, thank you so much for having me. So I think the collaborative approach that you're taking is wonderful, and this is a global problem that everybody is trying to crack. And so we really need to work together globally. I thought maybe if I just explain a little bit that the system in the UK is obviously different with the national healthcare system, and Genomics England had helped with the 100,000 Genomes Project, which is a research project, but completely embedded in a national healthcare system. And now within the NHS Genomic Medicine Service, a clinician can order a whole genome sequence, and their patient can consent for that data to also be available to researchers. And crucially, we can then go back to the clinical teams if they find either a new diagnosis or if there's a potential therapeutic option. So for me, these kinds of opportunities where there's a hugely rich national genomic data set that could really, that comment earlier about you can always find more, I think is going to be really key to this. And I think ultimately, you'll treat a lot of patients, you'll pave the way, and actually also shift the thinking about how one treats rare disease, because if there are, since the vast majority have such a low prevalence, it needs a completely transformative approach.
Great, thank you so much. Kristen, we'll turn to you.
Sure. So I think obviously, in addition to collaboration, which would be my number one suggestion as well, I think a lot of what n-Lorem is doing, for example, bringing this group together today is incredibly helpful. I think, helping identify what the collective voice is for this community, realizing there are many different realities based on the information we've just heard this morning and what we all know from our work. Having a collective voice is going to bring a lot more empowerment to this community and allow for easier policy change and allow us to really move into something that becomes much more scalable and realistic from a long-term perspective.
Yeah, Neil?
Well, first, I'm very pleased to be participating in this and so grateful to n-Lorem for all that they've done for the ALS community. We heard, this morning about this extraordinary technology, extraordinary platform that Stan has built, and there's real need, as we've been saying, to build n-Lorem's internal resources. They are very good at doing what they do, and those of us who are patient-facing, can turn to n-Lorem with great trust in the quality of the procedures that lead to a clinical ASO, and that is really meaningful to us as clinicians and to our patients. They do many things well, but n-Lorem doesn't have direct access to patients. They cannot possibly be involved in every disease community in a way I think that is necessary for these programs to succeed. And I think that's where collaboration, important word, with clinician scientists really matters. We as clinician scientists, and I learned this from the FUS ASO program, working with Ionis and n-Lorem in the early days, that we have as clinician scientists the ability to push at the frontiers of these things, push regulatory policy, push the science of these things, and to do human experiments of the kind that Stan described in a way that are, I think scientifically very important, but also, lead to huge therapeutic advances. And so we need as members of these, disease communities to partner in the way that we have with Silence ALS, to partner with n-Lorem, to bring the resources of our specific disease communities to this effort, and to help the program to reach as many patients as possible and scale.
Yeah, thank you so much. And Terry Jo, I'd love to hear from you.
Well, I think that one of the most important things we can do right now is educate the whole world to realize that what we thought was uncurable before is curable, and so that we can actually normalize ASO treatments over time to the point that eventually we'll all get universal newborn sequencing. We'll all have our whole genomes sequenced. We'll early on know about specific gene mutations that we'll have, and we'll know that you'll have to take your ASO vitamin pill treatment at a certain point in life. We need to, knowing that these families and these disorders are the pioneers, like Stan said, this is going to be ushering in a whole new era. So I think that what we really have to do is educate doctors, patients, and the whole healthcare system to accept this as a new way of treating everything.
Yeah, it's a great point, and we'll talk about a little bit more today and this afternoon, but I think that's one of the key components is like, this is really and truly, it's a shift in our mindset about how we're thinking about healthcare, how we're thinking about drug discovery and development, and how we're thinking about collaborations and partnerships. So Annalisa, I'd love to come back to you. So Genomics England is world renowned for your 1,000 genomes project, and really the genotype phenotype data that you've collected in this very robust manner, and now kind of thinking about that next step and moving towards therapeutics. I wonder if you could just touch a little bit more on some of the key challenges that you foresee and that you envision as we try to identify potential patients that you might have diagnosed that we already have ASOs for. What do you see as kind of some of the key next steps and challenges that we need to be tackling head on?
I think that's a really, really important question. I'll try to keep it brief because I think we could talk for a long time. And I think one of the key challenges is how we join up the ecosystem and how it really is truly a collaborative approach. And I think in the UK, there's obviously the NHS, the MHRA, and the regulatory body, and everybody, Genomics England, are all extremely committed to treating patients and getting the right treatments to patients. And so I think that commitment to the mission, I think, is amazing. And I think in a national system, if you get it right, and once you've got it right, you can then really run with it. It can take time to get it right, because everybody is trying to get it right in a way that then is going to work in a scaled up national way. So it's a strength, and it's also really important to work with the whole ecosystem and bring everybody together. I think I made a couple of notes just because I'm very jet lagged. So I wanted to mention, obviously, there was the mention of the newborn whole genome screening pilot that's starting. Again, that's truly working with the NHS. And I think the point earlier about innovating a care model is so important. And actually, so a lot of clinicians are naturally risk averse, at least a lot of the ones that I work with. And I think it's right to put patient safety first. And I think we have to do this in a way that builds confidence, and sort of spearheading the way with the first few patients, and working out that pathway with the whole ecosystem will then suddenly open the gates, I hope.
Yeah, it's a great point. And that's, I think even one of the questions that Stan answered this morning is, we already have identified a number. We have the 9 INDs, and we have many, many more coming behind. Do you have any individuals who could benefit immediately? And then the question is, how quickly and efficiently can we actually get that ASO to that patient? Can it be in a time that's meaningful for their disease prognosis? So thank you for that. Yeah, this is a really exciting sort of budding collaboration that we have started. So Kristen, would love to turn it to you, thinking a little bit about what St. Jude is doing. Can you share a bit about really what you want to do in this collaboration with n-Lorem, and just kind of expand on what you're thinking of what this will look like in the future?
Sure. So for a little context, in 2020, St. Jude launched the Pediatric Translational Neuroscience Initiative, which is a very dedicated long-term focus on applying the infrastructure and the capabilities of the organization to translational neuroscience. So really addressing catastrophic pediatric neurological diseases. And as part of that process, we're really organized with a translational focus, so that we have a preclinical area of focus through our Center of Pediatric Neurological Disease Research. We have a clinical focus through our Center for Experimental Neurotherapeutics, and then the Office of Strategy and Alliances kind of helps tie those things together, which is the part of the organization that I'm involved with. And so as part of that strategy and alliances component, we're really actively looking at and considering folks across the stakeholder landscape that we might be able to work with to have synergies, collaborations, And n-Lorem, of course, came up very early and often in those conversations when we were looking at the landscape for this ultra rare, nano rare kind of community. And as we were thinking about what our genomics program that we've recently launched might look like, we wanted to see who all we could learn from, work with, collaborate with. And n-Lorem came, as I said, up in many conversations. And I think that with Stan Crook's experience and expertise in ASOs, which I think is quite evident to everyone here, and with your really unique process and model for identification all the way through IND filing, that was something that was very appealing to us to work together on, as well as the mission of n-Lorem and your nonprofit organizational structure.
Awesome. Well, thank you. I know we're really excited also about this collaboration. So Neil, when I turn to you, so I think, what we've established with Neil and with Columbia is we've called Silence ALS that Neil sort of coined. And really the goal here is, as we know for all of our patients, is every day matters, every hour matters, and we have to feel this urgency. I think when you meet all of us from n-Lorem, you probably get a sense we sort of operate the way Stan Crook does, right? So we're hurrying because we know, and we just appreciate meeting all the patients here this week. And so what we're trying to do is just then think operationally. So what can we do to work together? As Neil said, we don't interface with the patients. We're actually blinded to the patient's identity. And ultimately, how can we work with Neil very closely through his scientific and clinical expertise to be able to bring the patients to us so we can just hit the ground running? So I guess I wanted to just talk a little bit about your experience and kind of think about what have been the efficiencies that we've put in place. And maybe you could share a little bit about what we're doing now and where we're headed.
All right. So, Silence ALS really grew out of the FUS ASO experience. Again, it helped us to realize the power of this technology and the ability to help our patients. N of one turned into N of many for FUS ALS, and the ASO is now in commercial clinical trial sponsored by Ionis, but it's really at the cusp of commercial viability. Some might say it's below the cusp, but what it made us realize is that there were many ALS patients out there that could benefit from this. People who I think would never be included in a commercial effort. So ALS lends itself, I think, particularly well to this kind of an approach. It is a genetically complex disorder. It's an ultra rare disease, but there are within that there are dozens of mutations, or hundreds really of mutations in dozens of genes. But many of those genes are micro rare, many are nano rare. And so we needed an approach to ALS that was, I think, able to approach this complexity, genetic and phenotypic complexity of patients. And that was what I think Silence ALS is. It's an effort to pair n-Lorem's platform with expertise and infrastructure that already exists within the ALS community. It's a partnership with Columbia, but really, as this grows, this is going to involve ALS clinicians nationwide and hopefully not too long internationally. We need to identify these patients. We need to phenotypically characterize them. We need to follow asymptomatic carriers in advance of disease onset. We need to provide patient data, samples, disease-specific resources to n-Lorem to help them to do what they do so well. And so that is what we're trying to do with Silence. There is a wonderful active ALS community around the world, and we're tapping that for the benefit of Silence. There is a great opportunity not only to treat these nano-rare patients, but to collect them in a group. And as Stan was saying this morning, to do one case, you learn a lot, but to apply the same ASO to multiple patients with the same mutation, or to look at patients with the same gene mutation, same gene mutation, but different varieties of it, and compare those, contrast those, aggregate those data. We're going to learn a lot about the nature of the underlying biology of the disease, which I think will inform our approach to the larger populations of ALS. So again, just to Stan's point and question earlier, why treat the nano-rare? It's because these lessons have, I think, very important implications to our understanding of the diseases that are more common, and we begin small and we grow.
Yeah, thank you. And this is a great example of, we definitely envision this model for other specific disease-focused partnerships as well. It really enables additional funding opportunities that we can really take and think, okay, specifically, how are we able to apply additional resource to this specific disease group? We can introduce, even more efficiencies across the platform, even in the context of understanding each patient's individual phenotype and as it compares to others. And I think also, like you said, is we're already starting to see, so we have, I think, 15 patients with ALS already in our program. And those that come in with mutations, different mutations in the same gene, this enables us to learn and repeat and do it better and learn and be more efficient because we've looked at this gene before. And so that's really incredible. And also the learning about how these patients, even patients with the same mutation, I think what Stan touched on one example this morning, how patients with same mutation still have different phenotypes. And that comes to the importance of really the need for individualized care. So Terry Jo, we'd love to hear a little bit from you. So a little bit more about Combined Brain. I think you touched on in your introduction about knowledge sharing and education, and maybe you could expand on kind of your thoughts around that space and how can we work together to really, educate and inform patients and advocacy groups to come together to collaborate.
So Combined Brain stands for Consortium for Outcome Measures and Biomarkers for Neurodevelopmental Disorders. So we're actually up to 100 disorders now.
Okay.
And we are trying to share ways of measuring treatment effect across all of those disorders. And I think that's how Combined Brain can help n-Lorem. We can come up with ways to make the actual clinical trial aspects of it more efficient. So our clinical advisory board, Liz Berry-Kravis, they have all suggested that we create a closet of outcome measures that the clinicians, the primary care practitioners who we need to empower and educate could open the closet and pull out the outcome measures that are already validated that they can use to see if the treatment's helping. But I think we also have to really systematize ways for the individual to measure what matters to them. So you talked about how you have patients with the same mutation who have a totally different phenotype. You have to measure what matters to each patient. And so you have to do both. You have to have goal attainment scaling or individualized measures, and you have to have these validated measures from the closet. And then hopefully we can share that data across those measures and individualized outcomes and all of that so that we can make the kit an easier task for the next primary care doctor who comes along the pike. I think that's what Combined Brain needs to do. Plus, we need to help you find more patients. And by doing that, I mean, we need to get diagnostics earlier and earlier and earlier and get stem cells and other resources that can be used so that we can get the whole process to move faster.
Yeah, no, that makes a lot of sense. And we're really working to ensure the standardization across data sets. And it's such an important component. We'll hear more from Lori a little bit later about that. So I mean, we've talked a lot about identifying patients. So, really the newborn sequencing, ultimately all of these ways, working with physicians. Kristen, I wanted to turn it back to you just with regards to some of the capacity building that we've talked about. And I wonder if you could speak to kind of some of the goals that St. Jude has as far as just thinking about even learning some of the processes that n-Lorem is doing for ASO discovery and development.
Sure. I think, from our perspective, it's really about collaborative research and thinking about how can we, across the board, with as many collaborations and engagements as possible, participate in research that becomes smarter and faster and scalable by working together. And I think that's part of what drew us to working with n-Lorem, along with a lot of other stakeholders in the space, including some who are in the room here today. So for us, it's really about capacity building in terms of sharing insights, sharing learnings, sharing resources, sharing models, frameworks, processes. And so that really is kind of at the crux of the work that we're doing together and that we're wanting to do also quite broadly in the research space.
Yeah, no, thank you for that. It's really exciting. I think there are many different steps of our process, of course, that will bring different elements of the collaboration. And I think, I feel like even last night, there were a handful of individuals that I spoke with and were like, we're really interested in doing this. And this is not only in the US, but more broadly speaking. So I guess I would love to hear, and maybe Neil, we could start with you, is just like if there's interest from perhaps an advocacy group or from an organization that's interested in trying to discover and develop ASOs, kind of what is your thoughts and what are your advice on even initiating this type of collaboration?
Yeah, well, I mean, there's been extraordinary interest in this. Our efforts in silence ALS are focused really in the United States, but as I go around the world, people are coming to ask how we can do this in their country. We have the advantage, and I think we'll talk about this later, we have a regulatory path here in the US to do this. That path doesn't necessarily exist, but that's part of our mission, I think, which is to export this opportunity to other nations. And people wanted to get involved, and I think in panels been touching on this. I mean, individual physicians, in order for this to succeed, you need dedicated clinicians, who are involved with the patients, involved with the families, doing the clinical phenotyping, not just at the point of nomination to n-Lorem, but in the months that it takes to get to a clinical ASO, and then beyond into treatment. I mean, this has to be a true partnership. You need dedicated clinicians, and what n-Lorem has to do, what Silence ALS is trying to do in our small corner, is to remove the impediments that are in place for clinicians who want to do this, because the interest is great, greater, I think, than we can accommodate, frankly, at the moment. But we need, as this scales, to remove the impediments for individual clinicians to do it, to provide them with the resources, the templates, the platform to make this easy, for them. When we did it with the first ASO, it felt very much like we were inventing the wheel. It's no longer the case, thanks to n-Lorem and thanks to Silence ALS, and I think this can be true for many diseases.
Yeah, it's a great point. And so really, it starts with the patients, it starts with the advocacy group, and having a very strong physician. I think we have a lot of amazing physicians that we're working with as well, but really that's a critical component, is having that really true partnership. So we just have a couple minutes left. I think before moving on to kind of the final question, I guess, Annalisa, would love to hear your perspective on, in let's say 3 years, when we're sitting here at the Colloquium, we will be in a very different place. I would love to hear from each of you, like what would you imagine, and where do you hope that we'll be able to be in that time?
So I would love it if patients in the NHS could have benefited from the drugs that you're developing. I think if we can treat the first patients, that will really help us to link up the ecosystem and drive forward these pathways to open it up. I think that it's really, it's key to show actionability. So linking again in with the newborn whole genome sequencing, the conditions that are included in this first pilot study are those that are actionable in early childhood. And I think having evidence for actionability is really going to be also key, driving forward the earlier and earlier diagnosis and
Mhm.
treatment.
Yeah.
So I've probably leapt a bit further than 3 years there. But I think that the sort of paving the way, and that it's okay to feel uncomfortable if we are at the forefront of trying
Mhm.
to drive change, that's okay. Yeah.
That is okay, right? It's where we want to be.
Yeah.
I actually love that. And I mean, I think, I doubt the pace will be any slower than that. And if anything, I think, even where n-Lorem has evolved to is if we think we would have had 3 to 5 patients in 3 to 5 years, and here we are with 100, the need is so great. And I think it's going to be really exciting to be able to just really pave the way. So really excited about that. So Kristen, would love to hear your perspective on this question.
Sure. So in the immediate term, being able to point to collectively kind of at a community level, a number of effective and measurable treatments that have done what we want them to do and what they hope they can do. And that beyond that have really generated data and insights to be able to apply to more patients and to larger populations, so that we can really think about how does this go from an individual focus to being a population focus of individuals. And I think that you'll hear that a lot today. We've already heard that a lot today, but I do think with a kind of as an institution with a research aim, that's what we're always thinking about. And we're very hopeful and optimistic that what's happening now will land in that place in the immediate term.
Yeah, great. Okay. Thanks, Neil.
Yeah, I mean, I would say that this, I think for most of us involved in this effort, that these efforts, that there's no question, but this approach is going to be successful. In 3 years, this will be sitting in a larger room with more grateful patients, with more stories that Stan's going to be telling of the success of these ASOs. And I think success will beget success, and that will draw, once you've proven this, I think it's already been proven, but once you prove it again and again as an effective therapeutic and research approach, we'll get buy-in from some of the stakeholders that were mentioned in the previous session, and insurers, federal research organizations, people who will understand that this is an effort worthy of investment, and I think that'll help us to scale.
That's awesome. Thank you. Terry Jo.
I think in the next 3 years, what I'd really hope is that we can streamline the whole treatment process. So come up with satellite sites where people can go and get treatment and yet be able to still be treated by their local physician at home, because it's so complicated to be able to, I mean, you're at a big institution that's allowing you to do this, but a lot of other big institutions are really struggling with allowing n-Lorem to treat patients. And so n-Lorem has to work with really local physicians. I think that's complicated for everybody. So I'd really like 3 years from now for there to be a much more streamlined process. So a guy in Kentucky who's got a patient with KCNT1 mutation that would really benefit would be able to treat his patient with the ASO, but take care of his patient in Kentucky day to day, collect the data, put it back in the pile. I think that's what I would like.
That's awesome. Well, I hope we have all these notes taken, because we're going to come back to you in a couple of years. So I think the key takeaway, what I'd love for each of you to hear is just that each partnership is unique. Each collaboration is unique. We're aligned on the mission, but ultimately we're looking to do different things with different types of groups. And so ultimately together we can help many more Nano-rare patients and do it in whatever way. So if there are interested parties, we absolutely would love to chat with you and look forward to the rest of the day. So thank you all. Appreciate it. We'll move on to the next.
I actually get to sit in a seat this time. Very excited about that. Just wondering where my panelists are. So I am prepared for this panel because I have tissues. So at n-Lorem, we are blinded to our patients. We don't know who you are when you come in. The physician actually submits an application, and unless a patient family comes to us, we don't really know who they are. But the ones that have reached out to us have been just amazing. And so I actually started calling them up and said, hey, would you like to come to Boston and do this meeting with us? And like, I'm only going to give you 5 minutes to talk. And they all came. So just very appreciative of it. This is going to be a little bit different. It's not really going to be a panel discussion. Like I said, I gave them all 5 minutes to try to tell their story, which is going to be really hard, I know. But we are just, this is the reason why we're here, right? All of them have different diagnostics journeys, different challenges that they deal with on a daily basis, but they all have something in common, that there's no therapy available right now for their loved one. And so, you know, that's what we're hoping to change, one patient at a time. And so I'm just going to introduce everybody who's not sitting in order. So on the end is Shanna. She is mom to Ireland, and you might have seen Shanna putting Stan on the spot for our first Q&A that we did on our podcast. And so she was great, stuck in some really hard questions, which was good. Beside her is Kelly. Kelly is mom to Connor, who is patient 001 that Stan was referring to this morning. And so very happy to have you here. She came all the way from San Diego. I should mention that Shanna came from Georgia. So like they came from all over. It wasn't just like, oh, come down the street and talk for 5 minutes. It's like, no, I'm going to fly out here to participate. Luke in the middle, father of Susanna. Susanna has actually been on treatment for a year now. And we have a video that we're going to play at the end of this panel. I'm going to try not to cry all the way through it. Yeah. And then Clayton is from Tampa Bay, right?
Yes.
Yes. And so we just started getting these cards. And so we're at the office, and all these like mail starts coming in, and there's these thank you cards, and they're like, thank you for taking Moston as a patient. Thank you for, you know, and we, not just one, not 10, not 20, like almost 100 of these thank you cards started coming in last year around Christmas time. And so we're like, we got to find who this person is, you know? So Clayton is Moston's dad, and they're here today, and I really appreciate it. And if you were here at the reception last night, you heard beautiful violin by Moston's mom. So really appreciate it. And then probably our longest journey here from Poland is Piotr. Piotr, did I butcher it too badly? Almost. Okay. Father of Lena, one of a set of twins. And yeah, so why don't we start with you, Kelly, and you know, turn it over to you.
Sure. Like Amy said, my name's Kelly, and my son, he's 14 years old today. His name's Connor. And, you know, when I walked in this morning, I was so happy to see all of the people here today. And, you know, in 2019, I was at a conference, and I saw Stan, and we were talking, and he told me he had the idea of n-Lorem, and to see what it is today, you know, n-Lorem represents our hope, my family's hope. And, you know, it's our happy ending. And, you know, 14 years ago, my life was very, very different. I was very naive. I thought that when people got sick, they went to the doctor, they got a medicine, and you know, most patients were okay. And Connor's my second son. I was a high school English teacher at the time. And from a couple days old, I noticed something was very off about him. He cried the majority of the time. He had trouble feeding. And even before he had his first seizure at 8 months old, we were in and out of the hospital. And he had very thorough workups, and all of these tests kept coming back normal. And, you know, he had so many different symptoms, seizures being the most debilitating at the time. We were trialing every single medication available. And at one time, you know, he was on a dozen medications for every symptom and still deteriorating. And we were being told a lot of what I consider today just ridiculous statements about from doctors, you know, take your son home and love him while he's still here. We don't know what's wrong with him. And I was obsessed with getting a diagnosis. I thought if we just got a diagnosis, there would be a treatment available. And, you know, I was flying my son all over the country. And when he finally got whole exome sequencing and he got a diagnosis, it was an apology. It was, your son has an SCN2A mutation. I'm sorry, we don't know much about it and we have nothing available to offer you. And it was devastating. And I started with scientists at USD and just starting calling scientists in the area, trying to find someone that knew something about this disease. And, you know, I'm a parent, I have 3 kids at home, 1 in and out of the hospital, and I'm being told that if you want this disease studied, you're going to have to fund it yourself. You're going to have to raise money. You're going to have to find scientists. And, you know, to make a very long story short, I have 5 minutes. I ended up starting a biotech company, and I worked for years on a molecule that I thought was going to help my son. And our scientist a couple years into that called me and said, Kelly, I'm so sorry, but Connor's not a candidate for this molecule. His mutation is very unique. It's mixed function. Connor's going to need allele specific knockdown. He's going to need his own ASO, his own molecule, and who is going to make a drug for one child? And it was the most devastated I'd been in this entire process. And a few months later, I was at the TIDES conference in San Diego, and I was listening to Stan speak on Spinraza, and I went up to say hello. And I asked him what he was doing, and he said he has an idea to start a foundation devoted to N of 1 patients where they have very unique mutations and to use ASOs. And it was like almost the twilight zone for me. It was exactly to a T what I thought my son needed. And I emailed him a few days later, and I went to meet with them, and I brought our scientist and another parent, and we looked at all the science of my son's mutation, and he thought that there was a potential that this could help him. And as soon as it became available, I applied to n-Lorem, and we're patient 001. And you know, it's been a really challenging path, but what n-Lorem represents today, I mean, my son, his life's a roller coaster. It's up and down. He has good days and not so good days, but he's going to be diagnosed or treated with an ASO this year. And I'm so grateful that my son's life, one child, there's a team of scientists and doctors and donors all for my son. So it's a tragic diagnosis, but it's also really exciting times for genetics and the potential of, you know, what's possible. So that's it.
Thank you, Kelly. I think Clayton, you're up.
Thanks for your interest in our son's journey. Mawson is our only child. He was born precisely on his due date by natural birth following a healthy pregnancy. My wife, his mother, is extremely talented. She was a professional violinist and toured with Rod Stewart, Andrea Bocelli, Styx, David Navarro, Tommy Lee, to name a few. Her mother was a model. Her father was a professional football player, played in the NFL, Canadian Football League, and American Football League. And he currently holds the record for the longest punt in the world. My father was a professional golfer for a little while, and then I'm not sure what happened to me, but Mawson clearly has some rare genes, and unfortunately, one of those genes has a severe, very unfortunate misspelling. He's the only known case with his particular variant on a gene called KCNB1, and that variant causes him to have some severe medical challenges. When Mawson was 4, he had floppy wrists, and kids started making fun of him, adults started making fun of him, and we tried everything we could, kinetic tape, TENS units, massage, physical therapy, exercises, nothing seemed to work. And then one day I came home with a baseball glove, we started playing catch, and Mawson has been wearing a glove. Sorry. Mawson's been wearing a glove every day for the past 8 years, and he's fallen in love with baseball, and his wrists are no longer floppy. When he was 5 years old, he had been working really hard. He could say nearly 100 words, and he was able to run, he was able to climb stairs, he was able to swim, and he could dance like Michael Jackson. He was out of control. He was so fast. And then on December 16th, 2031, my gosh, I don't know what's going to happen on 2031. I hope we're going to make it there. But 2016, on December 31st, 2016, he suffered his first very violent tonic-clonic seizure. And within a few weeks, he was having 4 different types of seizures and as many as 30 tonic-clonic seizures a day. He ended up being hospitalized in Orlando, and the third day we were there, a doctor came into his room and wrote LGS on the board, and she explained to us that our son had Lennox-Gastaut syndrome. I asked her what that meant, and I asked her what it meant for his life expectancy, and she just began to cry, and I'm thinking this must be bad, and she left the room before she'd come back in. Unfortunately, baseball gloves cannot cure Lennox-Gastaut syndrome. Since then, our seizures have stripped our son of his ability to walk at times, his ability to eat, his ability to swallow. He relies on a feeding tube. And on tough days like today, he convulses over and over. His oxygen level drops. He turns blue. He chokes. He vomits, and he wakes up with a painful ringing in his ears. And he loses his balance, his ability to walk, and he gets very scared of the next seizure to come for him. We can hold him, or we can try to comfort him, but we can't protect him from the next seizure. And unfortunately, the anticonvulsants have not been able to suppress his seizures fully either, and they come with a lot of side effects. They can suppress his heart rate, his blood pressure, and his breathing. They're so strong, it's almost like he's been in a trance for the past 7 years. And sometimes we look back at videos of things our son was able to do before seizures, and it's really sad watching how much he's declined. Beyond the decline, we know that there's a very high mortality rate associated with Lennox-Gastaut syndrome. Death has come for our son so many times, we don't know when it's going to come for him again. I could give you countless examples of how hard Mawson's worked and his relentless efforts, of our efforts, and of his amazing doctor's efforts. But what matters more is this. For the past 7 years, we've been using medications to manage symptoms, and Mawson has been declining. We're so grateful n-Lorem has accepted his case. The work that n-Lorem is doing gives us hope that Mawson may one day stop declining and have the opportunity to make sustainable forward progress.
Thank you. Clay.
Sorry.
I think you got us all choked up here. Here too.
So basically, I just wanted to start by saying that I want to echo Tom's word about the fact that emotions are very intensive. And this is why basically we decided with my wife to draw the line, and just to show you like the roller coaster we're going through with Lena and with the twins, basically. But before getting there, I have a feeling that the rare disease overall kind of hunted me way before that. So you wouldn't believe that my passport that I got into US with was issued on the 28th of February. As you know, this is Rare Disease Day. That was the first thing, and it happened like a couple of years ago, way before I found out that we were going to have twins. And then 2.5 years ago, when my wife was pregnant, I've joined Biogen, and back then I had no idea that one of my kids is going to have ultra rare genetic condition. So there were like some kind of signs, like happening way before the actual moment that we started to find out that something is wrong with Lena. So basically, as a parent, because our kids, our twins, are our first kids. So before that, people are going through up and downs. But the emotions, the roller coaster, you kind of start to feel and experience the moment you get kids is hard to describe. And that was, you can see all the way up the moment that the twins were born in the September 2021. Everything was going well. We're like happy and just waiting for the kids to, just to look like, just to see them in the world. And then Christmas Eve came. And yeah, I have some kind of, let's say, a strange connection with this kind of dates, like Christmas Eve. I'll tell you, like Rare Disease Day on the 28th of February, my passport. And the moment we started to recover from the sleepless nights, because I can tell you, twins is not an easy thing to manage. The Christmas Eve came, and Lena started to have grand mal status epilepticus, 30 seizures in 48 hours. So we rushed to the hospital, and that was the first massive hit. And something that maybe it's not like easily seen on the slide is that there was a second baby. So like my wife, she was at the hospital, like having the kind of experiencing the seizure like every hour and a half, while there was a second one at home. And I was like kind of single parent before we kind of started to basically take care of the seizures of Lena. And I remember myself, it was during COVID times, I was rushing between home and the hospital to get the milk. So that I was basically kind of entering hospital illegal during COVID times just to get the milk for the second one. So that was like a huge roller coaster. And after a few days, we started to recover slowly because our doctor, she like made all the tests, MRI, spinal fluid, everything looked fine. And she started to have kind of pre-diagnosis of the GLUT1. But then a couple of weeks later, the gene panel ruled it out. Gene panel didn't show anything basically. But then she said that the thing, and I want to basically echo your words, that there's like one last thing we can try, which is called whole exome sequencing. It's available commercial, but obviously thanks to the fact that we both work in the corporate world, we were able to kind of, afford that basically overnight. And we're like, so waiting for the results. Normally back then, it took like 5-6 weeks. For some reasons, after 2 weeks, we already had the result. And another kind of symbolic date, we found out about the PAX diagnosis during Valentine's Day. And that was like the second massive dip you can see on the slide. And that's basically kind of the straight road to the depression, to like there are like many things like going through your head. However, now if I look back, and if you can go to the next one, now if I look back, I had a feeling that that was the kind of the starting point. So looking back, like more than a year ago, it's like at some point you think it's like it's done, it's game over completely. And looking back for us, it was just the beginning. So obviously we were grieving, and on the next slide you can see our emotions going up again. We were grieving, but at the same time we decided to act quickly. I was always open, always vocal, always transparent. So countless people at Biogen found out about my situation. People were teaching me genetics. I have business background, so I was trying to understand all about ASO, how it works, what is the ASO screening and stuff like that. So I discovered n-Lorem through basically two ways. The one way was basically Biogen employees that told me that, hey, there's this n-Lorem thing, and maybe your kid could get there. And also, I very quickly connected with other parents, mainly from US, and that they also told me that there's this thing that Stan decided to set up to help nano-rare patients. So I remember another date, my wife's birthday, where we were on the call with Wendy Chang for the first time. So these dates are just hunting me all the time. And as you can see, we started like slowly to recover. And we also decided to open the research foundation because PAX2 was first described in 2018. Nothing is known about this syndrome almost. We have no idea what is the mechanism of action. But like basically it doesn't really matter because we just want to kind of treat the patient without knowing fully and understanding fully what is causing the disease. However, we wanted to kind of give back something to the community, so we started the foundation. We started to get in touch with many different parents, which are kind of calling us every night, almost every day, trying to kind of learn something from us. And then September 2022 came. So exactly 1 year later, during Lena's first birthday, we got the information that it seems that the PAX2 could be the way to be treated with the ASO, with the allele-specific approach. And when I think about the n-Lorem, it makes me think about the rocket, as you can see here. So the moment that and we know it's like there's no guarantees, right? But I have a feeling that we are basically part of a big group building the rocket, and maybe at some point we'll be able to get in and just take our daughter and just fly to the moon.
Thank you. Shanna.
Good morning. I'm going to stick to my notes because I'm an emotional person anyway. And then I had my daughter, Ireland, and just really set me over the edge. Thank you for the opportunity to share a little bit about my precious 8-year-old daughter, Ireland. She's on the left in the middle picture with her little sister, Brooke, who is 4. The top or the bottom left picture is Ireland shortly after birth when she was taken to the NICU. They called her a jittery baby. And she had a couple of episodes of apnea where she just randomly stopped breathing. And they did 1 million tests, couldn't really find anything wrong. And a veteran NICU nurse, I'll never forget it, she looked at me and said, "There's something wrong, we just don't know what." So we go home with our new baby and trying to enjoy her, and she's a little bit delayed, the tail ends of the developmental milestones. And then at 6 months old, she was sleeping on my chest because she was a terrible sleeper. We now know why. And she started convulsing, and I had never seen anybody have a seizure before, but I knew immediately she's having a seizure. So we call 911, and few more seizures followed from there. And our incredible team at Children's Healthcare of Atlanta immediately did genetic testing. And so we got a pretty quick diagnosis of a CAC NA1A mutation. And so as you've heard, you have this answer, but no treatment options are available. Ireland's particular variant is neurodegenerative. It causes intractable epilepsy, ataxia, global developmental delays, cognitive impairment. She has an autism diagnosis and severe hemiplegic migraines. And her seizures are all or nothing. She's not having daily seizures, but when she does, they're always status epilepticus. She requires multiple rescue drugs. She has a very unconventional rescue protocol because none of the FDA approved rescue drugs have ever stopped her seizures. She's been intubated 9 times for status. That's the right picture. So we spent a lot of time in ICU. We always carry an emergency bag everywhere we go with her little mobile pharmacy, an ambu bag, because she also stops breathing, turns blue, and it's status. So the seizure's not stopping, she's blue. So the responsibility is really on us to support her respiration. And she has a little nasopharyngeal airway, which is a little tube that goes in her nose to lift the tongue off the back of her throat so that the air can get in. So thankfully, thanks to her care team in Atlanta, we do have this very aggressive rescue protocol. I think it's the only reason she's as functional as she is. But we live in constant fear of the next big neurological event being the one that changes her forever. And I think we've had good care and we've been lucky, and God is taking care of her so far, but we never know when we're going to miss one. So when she was first diagnosed, we were told that she was the only known person in the entire world with her particular variant, no treatment options available. She's tried a long list of anti-epileptic drugs and various other therapies. She's had epilepsy surgery. And the cocktail that she's currently on seems to lessen seizures, but not without greatly sacrificing quality of life. She's very sensitive. I think all of our kids are very sensitive she's very sensitive to medication side effects. And so with the biggest, for her, behavioral side effects are really difficult to manage. Ireland, let's see. And none of these drugs address the root cause of her symptoms, which is her CACNA1A variant, and they're really only pitiful attempts at seizure reduction. Her variant affects every single aspect of our lives. When she's not battling seizures and recovering and medication side effects, we do get a glimpse of what I call the real Ireland. And she's bright-eyed, happy, giggly, she's funny, she's got a great sense of humor. And she's really just an angel. And I know I'm her mother, but she is. Let's see. 2.5 years ago, we found out that Ireland's n-Lorem application was accepted. This opportunity is what every Nano-rare patient and family dreams of. In August of that year, we had the opportunity to sit down with Stan and Roseanne in Atlanta. And I don't really remember what was said during that meeting, but I remember how we felt. And we know that n-Lorem is 100% committed to doing the very best they can to find an optimal ASO for Ireland. For years, I felt like the burden of trying to figure this out for her was entirely on my shoulders. I Googled till my eyes hurt. I contacted research. Anybody who had published on CACNA1A got a contact from me. And it's such, I can't tell you the tremendous sigh of relief knowing that somebody else is kind of picking this up for her and carrying the torch to try to fix it for her. Our entire family has been praying since her diagnosis for healing for her, and it is our hope that n-Lorem is the answer to that prayer. I just want just to close, take a minute to acknowledge the initial patient families and their care team and the doctors who have supported their applications and are committed to carrying this through to treatment. I know that the knowledge gained from these pioneer families will greatly impact Ireland's experience, and I'm very grateful to all of you as well.
Thank you.
Can I just say
Yes.
one quick thing?
Yes.
Just thank you all. Adding to what Stan, in the very beginning, you saw a video of Susanna standing up. That was the first time she stood in 3 years. She had lost the ability to stand. And at the time, my father was dying of cancer. And that night, I went to visit him in the hospital and showed him videos of Susanna standing up for the first time in so many years. And he died a few days later. And it was almost as if he, wanted to have this gift that you've given us. So I told Amy that I wouldn't talk because I can't go for 5 minutes without crying, even though I have these. But anyway, I wanted to tell you what that video meant to me.
And thank you for sharing it with us, and we could share it with the community. I think, at the end of this panel, we have another professionally produced video that Biogen has done for us about Susanna's journey through her treatment. And I was really going to play it while we're all up here, but I think maybe what we'll do is we'll come off the stage and we'll play the video, and then we'll move to the next section. Before we do that, Luke, is there anything else that you would like to say before we play Susanna's journey?
I don't think so. I mean, I think that the whole point of what we're doing is to galvanize the community of nano-rare families. And if we all don't put our shoulders behind that, be it with resources or awareness or whatever, then, it's just going to be more difficult. So I wanted to thank everybody in here for putting your shoulder behind that galvanized community and to continue doing that. It means the world to families like ours, and that's all.
Thank you. Thank you. And can we get a hand for our patients with care?
Every bold and real scientific endeavor in the history of the world starts with an N of 1. Bold endeavor is starting with Susanna and n-Lorem and this fleet of people who have come around to power this thing to work for Susanna and other kids. Susanna was 18 months when we started realizing that something was needed to be addressed.
She was in the bath, and instead of actually kicking her little legs, Miss Susanna picked her legs up by the knee herself and kind of made herself do a little kick. Part of it was like Does she think that's what it's supposed to be? Or is this the only way that she can? And we were worried.
We were worried. Susanna started treatment with her ASO that was designed specifically for her in November of 2022. That would be so incredible if things were less severe for Susanna and she could participate in things that she can't. And if all of a sudden those feet that are in horrific pain aren't in that much pain, you know, and she was smiling instead of crying all the time.
Susanna has always loved the water. Susanna had developed this, I call it the mermaid, where she wasn't able to kick her legs independently. And then I had this moment of having a new present, a new what is here that filled me with hope. And that was seeing Susanna swimming and kicking her legs.
In sync.
And like kicking back. Doing it, like really getting her legs going up and down and splashing through the water and making her way from one end of the pool to the other. And it was just
It's no longer therapy in the pool. It was a little girl swimming.
It was a little swimming. I think we have been really lucky and able to see some ways that there were things we thought that maybe we had lost, or skills that she had that were slipping away, that we feel like maybe she's gotten her grasp back on. But I had never seen her kick before. I had never seen that before, and that was a new thing. And really all we want is for her. We want her to live. That's our hope. And I am so happy with her just not getting worse. But to be so overjoyed with something new. Yeah, you know, it's pretty remarkable.
And this is a hard thought to think of, but imagine if we got this therapy when she was 2.
Oh, go Susanna, go.
Let's see you get up the way you got up before. Yes, Susanna. Yeah. Imagine if other people can have this therapy when they're 2. Here's a note from your teacher. Great day for Susanna in reading.
Is I have that.
Yeah, but we'll take it.
We're really lucky. We have her.
Dad, come on, they'll teach you how to stand up.
You gotta stand up too.
Chris, that's going to be a hard act to follow.
Yeah, thanks, Natasha.
Chris is going to talk about driving innovation in rare disease from Biogen.
Yeah, that is a tough one to follow. But actually, when I was thinking about this, I thought what I'd really like to do is say, why has the conventional drug development system failed Susanna and others? And how is it that Stan and n-Lorem have figured out a way to really adapt this system and be effective? And are there things that we can learn from what n-Lorem is doing, potentially for other diseases? And I guess the final thing is there a point in time, and this goes back to Dan Kern's question, where we might be wandering from nano-rare into ultra-rare? And are we going to see some of the barriers of the conventional drug system come back to haunt us? And if so, are there things that we can be doing today to try to alleviate those? Now, I happen to have a deep allergy to PowerPoint, but I do need one slide for this. And is that up? Yeah. So what happens in a classical drug development process? And, you know, you have a graph here, you know, this is time along the horizontal axis, And basically money is on the vertical axis. And in a normal drug development, you first have to start with what is causing a disease. And that's usually work done in an academic institution, but at some point people realize, well, if we could upregulate this or downregulate that or block this, we can actually fix this disease. And then a lot of years of research go into, can we find a molecule of some sort that will actually make that happen? And that's where you have this first phase of preclinical work. And that's what a lot of companies in the Boston, Cambridge area are doing. They're startup biotech companies. It's probably somewhere between $50 and $80 million, and about 5 to 7 years before we actually can test one of those molecules in people. And when we do, we get this thing called an IND, an investigational new drug. And that is basically the assurance to the drug regulators, the FDA in the United States, the EMA in Europe, that this is safe enough to test in humans. And we go into a first phase where we're testing this in healthy volunteers. Then we do this in patients, and we're trying to figure out what's the right dose and are there side effects. And then we go into a third phase, which are usually 2 studies, placebo-controlled, where the FDA is looking really for 2 major things. The first is they really want to make sure as we go into large populations that these drugs are safe. And that's where they use the word rare, because you have to design a study that's large enough to really identify those rare side effects that maybe only 1 or 2% of the population might suffer from. And then, of course, they also want to really make sure that your drug is efficacious and it's working, and you have to test that against placebo. And the point I'm making is all of that costs a massive amount of money, first of all. Just to get to approval is hundreds of millions of dollars. And that's for the one that makes it, because there are probably 5,000 molecules at any one time that are going into human trials for the very first time. But thousands of those will fail. In fact, if there are 5,000 in phase 1, there's only about 30 new drugs every year approved by the FDA and regulatory agencies. So, and that's what really costs an awful lot of money to the system. It's not just the development of the ones that work, it's also the ones that didn't work. And then even then, and the biotech community learned a long time ago, there is something called the valley of death in here. And that is where you get into phase 3 and all of the launch costs, because it's not enough to get a new drug approved. Why is it that Anna had 34 diagnoses, only one of which was correct? Well, part of that is because, it took that long for physicians to actually do all the research to figure out what was wrong with Anna. But I can tell you that even for non-rare diseases, it can be a long time before people get diagnosed. And there's a massive effort that is always needed to educate physicians and even to make patients aware of potential treatments. And that's where you get into the commercial launch phase. And that's why you need people around the world who go and help patients. So in a rare disease case, for instance, I talked about Genzyme earlier. I was in Malaysia one day and visiting Genzyme Malaysia. We had 3 people and 10 patients. And you have to make sure that these very expensive drugs are there in the infusion center when the patients come. But we're in Malaysia, and there isn't an infusion center in every street corner. So you actually have to go and help. In this case, I remember one case was a mother with 4 children. Genzyme was there to actually make sure that that mother was able to get bus tickets for her and all 4 children to come to the infusion center, arrange daycare for the 4 children while she underwent the infusion, and got the enzyme replacement that was basically helping her lead a normal life. And that's what normal drug development looks like. Now, because of the cost of this, hundreds and hundreds of millions of dollars, when you're talking about the N of 1, nobody can actually afford to do that on the regular system. But the brilliance of Stan and n-Lorem was, well, actually, these ASOs have already been through all that process. You heard about the product Spinraza. Spinraza has been all the way through there. And that gave Stan really the idea, okay, you heard him in his talk this morning say, well, actually, I can use one of these ASOs with confidence because they've already been tested for safety. I need to adapt this so that it can actually fit this particular genetic mutation. But that gives the FDA the confidence to allow that to happen. Now, the one issue we have is going to be what happens. We start with the IND. So n-Lorem is really just working in that first slide of the curve. And we don't actually go all the way to drug approval. And that is great under the foundation model. But what was also interesting was Stan said, well, what happens when we get to ultra rare? So what happens when we're in that? This model works down to about 5,000 patients worldwide, I would say. We're in a model where we're 0 to 30. What happens between 30 and 5,000? And that's where it gets a little tricky. We ran into that with Qalsody. This is a SOD1 mutation for people with ALS. It's an ASO, also from Ionis. We are charging about $190,000 a year for that treatment. If we had to do cost plus, we'd have to charge a whole lot more. There's just no way that you could ever recoup all of the costs that have gone into the development of this. Because at some point, I think the risk to the model is that we get so many patients and people don't want to stay at IND. They want to see an approval. Dan Curran asked, well, why can't we get insurance companies to pay? Well, the reality is that even the NHS that was talked about on a previous panel, European governments, insurance companies, they want you to get to here. And then we'll reimburse you maybe. But if you're here, you can't. So we will have an education work necessary to say, we are seeing results. We are seeing Susanna stand. How much more evidence do you need? And do you need to go through all of that cost? Because the more cost you add, the less likely it is we're going to have a model that actually is able to finance that. If Stan and John Maraganore tested the foundation, the philanthropic approach to larger patient populations and it couldn't work, I guarantee you it doesn't work, because Stan is way too much, say he didn't have the time. Stan doesn't do anything except that at 300% energy levels. And that's where I think we'll have to think about and educate the system. We have a platform that actually works. We know it's safe. We know that we can adapt it. But in today's world, while we can do N of 1, maybe N of 30, if we're going to get to N of 100, we will have to work within the system. We'll have to get the FDA. We'll have to get insurance companies. We'll have to get CMS in the United States. We'll have to get European governments to say, we actually have enough evidence. The reason that Qalsody costs so much was there are 350 patients in the United States with this genetic mutation. And I listened to the advisory committee. We had families who show their whole family tree about how many people have died of ALS because of this mutation. And yet the advisory committee is saying, well, we still don't have enough data. And you only get one shot really at doing the clinical trial in something like this, because the minute you show something works, it would be unethical to do another trial. And so I was amazed that the FDA has been hugely supportive. But on the advisory committee, we have people who don't normally see rare diseases. And the mindset of most of the medical establishment, as well as the regulatory establishment, as well as payers, is this model. And that model is the massive evidence base that everybody is looking for. And when you have these small n's, you can't possibly have that. So I just want to put that out there, because as we advocate, I want to first acknowledge really the brilliance of Stan in understanding that he could use this model, and it's his personal credibility as well as the evidence that Ionis and actually Biogen, because Biogen is the one that really did all of the massive investment on Spinraza, for example, but in partnership with Ionis to actually create the safety foundation, and that Stan has been able to go to the FDA and agree to be used. But at some point, as we expand this, do we run back into that? And that's, I think, what we're going to have to prepare for. So that's all I really wanted to say. I really, I'm just admiring you so much, Stan, for what you're doing there. And, you can count on Biogen to help support all your efforts for n-Lorem. And I really want to thank also the Biogen team, Natasha, and our heads of R&D for all the support they're providing in n-Lorem as well. So thanks.
All right, we have one more panel before lunch. We can't do this alone. We have lots of partners. We have lots of partners that are doing a lot of things for us. Biogen's been amazing, and we thank them for all of their help and all of their support and for hosting this event today. So just can't say thank you enough. I'd like to welcome to the stage our next panel, what it means to be a partner with n-Lorem. So our moderator today is Brady Huggett, who has been in the industry for a very long time. Now he is the enterprise editor at Spectrum, which is part of Simon's Foundation.
Well, the first thing is it's difficult to follow that panel that was right before us. I can still hear people sniffling in the room, and that puts everything in perspective. And suddenly the questions that we're talking about almost don't seem important. They are important, but still, that was an incredible panel. So why partner with n-Lorem? What does it mean to be a partner with n-Lorem? That's what we're going to talk about today. I'm going to briefly, for the sake of brevity, just introduce our panel by just their name and affiliation, and they can tell you more about what they do when we get down to questions. So Frank Bennett is CSO at Ionis. Kate Dawson is a senior vice president at Biogen. Seiji Fujioka is the president of Nano-rare, I think I said it right. Yeah. And Jamie Campbell is the CEO of Parexel. So we have two companies, drug development companies, and two CROs on this panel. And so the first question is, can you just sort of tell us, and we'll start with you, Frank, tell the audience what it is that you actually do in this partnership with n-Lorem?
Yeah, so I I've known Stan for over 40 years and have worked for him for most of that time. And one of the things that we did is started Ionis 34 years ago, and with the dream of developing a technology that could revolutionize drug discovery. And we were successful in doing that. And so one of my goals is to make the technology available for all patients that can use it. And, you know, as Chris originally showed in the slide, for patients that are fairly abundant in our population, there's a well-trodden model for providing antisense drugs to those patients. I started looking at rare and rarer diseases and realized that there are patients out there that could benefit from our drugs. And we started a large number of programs that could benefit these, I would call them micro-rare in the slide that Stan showed, and realized that you could also move that down to nano-rare. And so we've been a partner with n-Lorem and helped do a lot of the drug screening, the early drug screening as n-Lorem was getting launched and building their own infrastructure. A lot of the drugs that were being developed came from Ionis as a scientist screening those drugs.
Kate.
Hi, it's a pleasure to be here. I'm Kate Dawson. I lead one of the development units at Biogen. Part of my development unit does look at some of these more rare disorders, and we partner very closely with Frank. Biogen is really, as Chris has said, has been a longtime supporter of n-Lorem, was part of the original financial founders to get this off the ground. And we continue to really participate both in adding our knowledge, both in some of the manufacturing of the ASOs and the specificity there, but also we have clinicians, Toby Ferguson, that sits on the access to treatment and does that assessment when people come in and make recommendations for potentially joining in and having you know, their loved one participate with this foundation.
Hi, Nirodem Kabisha. I am the president of the Nirodem Kabisha, and we are the CMO. We are the oligo-specific contract manufacturing service company. So we make API, and we also do the fill and finish services as well. Nirodem Kabisha is one of the very first company actually do the CMO space of the business in oligonucleotide. So we have lots of specialties and knowledge and experiences and infrastructure as well. But at the same time, we were grown by the industry actually. we got lots of partnership with like Ionis and many oligo company, and they gave us an opportunity. They gave us the lectures and transfer the technology and things like that. So understand and you know, contact us and a couple of years ago, and we believed it is time actually we do some contribution actually back to the industry, and any little things we can do and would be very valuable for us. So that's why.
Okay, I'm going to come back to that, but go ahead, Jamie.
Hi, I'm Jamie McDonald. I'm the CEO of Parexel. We're a clinical research organization, so we help biopharmaceutical companies take drugs through that very traditional process from IND to approval. We do that for hundreds of customers, thousands of drugs, hundreds of trials in rare disease. We've really not had the opportunity to support the nano-rare space. When we heard from Stan, it was pretty much a no-brainer. Stan is pretty inspirational. Sarah is a former Parexel colleague of ours, so the n-Lorem COO is a former colleague. We provide that clinical expertise, medical writing, preparing INDs, preparing regulatory submissions, workflows, plans, clinical design, database build. So we've supported and helped n-Lorem set up that function internally so they can run as efficiently as possible. This is a highly regulated industry, as we all know, and it is critically important that we document everything that we do, because it has to be there for inspection, but for future use as well. And we've helped set up those templates, workflows, processes, and provided consulting and support on the regulatory side. We'll continue to do that as you look to globalize, which I think is a next important step for n-Lorem.
So it's a little different for you, Frank, but for the whole panel, what did it look like, or what did this sound like when Stan came and said, hey, I've got this thing that I want to do. There's not going to be any money involved. In fact, it's going to actually cost you time and resources. We're going to look at one patient at a time, and we're going to provide a drug for free for life. Would you like to join that? I mean, you know, we're kind of laughing, but that's not the way the industry works at all. So tell me how those conversations went, where he sort of got you on board, and we can start with you, Kate.
Well, I wasn't there, but
what have you heard?
No, I mean, but certainly Al Sandrak, someone who I've worked with for many, many years. And obviously we had this very close partnership with Ionis, right?
Yeah.
We are already in the rare. I mean, Chris talked about how we're in the rare space, and he's talked about how we're in SOD1, which is 350 patients. So Biogen already understood that there's an unmet need here, and there are people who, if we would only concentrate on them, could benefit from the therapies and the knowledge that we have. So I think it was a natural thing for Biogen way back when to say, you know, we understand. I'm a neurologist by training. I will, without going into my age, certainly when I was in my training, there were a lot of disorders that we couldn't treat. And I took care of ALS patients, and I've said this to people before, that good care at the time was telling people to put satin sheets on their bed and wear satin pajamas, because that allowed you to turn over in bed, and that prevented bed sores. I mean, and people were very thankful for that. We need to do better than that. This is an opportunity to really show our belief, which is patients deserve care and they deserve to be treated, and not forget these patients, this rare population of patients, or these N of 1 patients, because they also deserve the care.
I mean, let's go back to you, Seji. So when this proposition is presented to you, is that something that you have to turn to your shareholders and say, hey, we're going to start dedicating resources to this project that's probably not going to see any financial gain for us? Is that okay? Or how did you make the decision?
Well, actually, we don't need to talk to the shareholder, but again, I was so inspired by this Stan Kwok, actually, and his initiatives and Laurent's initiatives, and you know, we do the manufacture the oligo drugs from preclinical to the commercial scale, so very small scale to the large scale. So lots of the infrastructure is available and the resources available, and if we do take the let's say 2% of each person individual time actually, we put together, we could do something actually. So we can create some sort of the creative time actually to dedicate it to this specific program. So of course, it is a challenge for the it's all cost associated. But again, if you utilize the little percentage of your time put together, we can do something. And I thought we can contribute it to his initiatives.
Yeah.
It wasn't a difficult discussion. So, I mean, for us as a contract research organization, we'd already set up as part of our mission and values the patient first focus. We have a chief patient officer that's pretty unique in a company that doesn't actually own products. We have a patient advocate who is a mother also of a child with an ultra rare disease, maybe not nano-rare. So it was easy for us to see how we could help. Stan is also very persuasive. And I think all of us in this industry have realized that the science and knowledge existed to be able to treat those super, super rare patients. So wherever on the spectrum of rare disease they were, but the return on investment and commercial model was not going to support it, or the regulatory process. And I think that was probably one of the first questions. Have we figured out a pathway with the regulators and the FDA to make this work in a viable way that will allow us to reach as many patients as possible? And I think the science is fantastic, but I think the work that the community has done in order to sort of bridge that gap and bring in the other stakeholders, the physicians, obviously the families and patients, but also the regulators and others, and obviously the partners here, it has been a huge accomplishment. And I think it sets up a framework for potential expansion.
Any comments anyone wants to add to that? No, silence. So I had this thought. So if partnerships work because both sides of a partnership are getting something out of it, and it's clear what n-Lorem gets from this. So can you tell me what each of your companies gets from being in this relationship? And I'll start with you, Frank.
Yeah, well, I think the biggest thing is, the vision and the mission for what n-Lorem's doing. It's, something that, is sort of in the ground water of Ionis, and really is, something that, patients come to work every day to help patients. And, they're working to help patients with rare diseases, large diseases, and nano-rare diseases. They all deserve help. And I think n-Lorem gives a number of our employees the opportunity to contribute, the full scale and have a big impact on patients' lives. And I guess one of the other things is that with the nano-rare patients, you can see an immediate impact on the patient's lives for what you're doing. Whereas if you're working on a cardiovascular drug that's going to require 20,000 patients to be treated, it's didactic on paper. You see what you're doing, but you really don't connect with the patients. Whereas with the nano-rare situation, the patients in the room, I've gotten to know a number of the patients in the room today, through working through n-Lorem, as a number of our other employees have.
Yeah, Stan has talked about that before, about this is almost like being a physician again. It's very hands-on with the patient as opposed to helping run a trial of 300 patients or something like that. Yeah, go ahead, Kate.
Yeah, I think what I'm going to say is very similar to what Frank just said, but people work at Biogen because they want to make a difference in the lives of patients. And that's not just the physicians or the pharmacists. It's actually even the people that are charged with going out and, marketing and selling our products. We're all here for this mission because we want to make a difference. It's why I moved from Mass General down the road to Biogen almost 20 years ago. I think that this opportunity really presents us a unique opportunity to really say, we're not leaving any patient behind. We would never, as a company, we would never be able to go, Chris spoke to the model doesn't work economically, but it doesn't mean it's not the right thing to do. And I think we feel very strongly that this is important. And our like Frank said, our employees get something out of it. Stan came and gave a talk. We call it an Ideas Cafe, and he shared about what n-Lorem's doing and the work. That was one of the biggest responses we got from our employees, how inspired they were after that talk.
Well, like what kind of responses were you getting? I get the sense that if you worked at a company like that and you realized, oh, we are actually trying to create drugs to give to patients for free, that you would take a certain amount of pride in that.
To be honest, I'm not sure how much the free is. I think there's a pride to know that you're doing something that's not for profit. But again, I don't think we come to work every day to try to make a profit. We come to make a difference.
Yeah.
And so it's the recognition that patients and families who would never be part of a clinical trial, a large clinical trial, the fact that someone was thinking about them and making a difference for them and those patients and those families, that's what's inspiring, that we're not forgetting about them. And then I think the other piece is we enjoy the intellectual part of it, of can we help?
The challenge.
And that's also, I think for the people that are on that side of it, it's that opportunity to give back and know that they can use their experience to actually make a difference.
Yeah, yeah. Saji.
I think the biggest opportunity was the reminding the pride of our team members, employees, and we are the CMO. We make the products. Anybody joined to the pharmaceutical space, they have some, spirits actually to try to contribute to the life and the patient and etc. But if you do the daily work and every day manufacturing, we have to focus on the quality and the compliance and SOPs and all kind of stuff. So you got to focus on that. So if you do that, sometimes, you will be a little bit distance from the real families of the patient and the real disease and etc. So if we do this and we can have the more real voices actually of the real world, so that will help the lots of remind the people they join to our company or our space. To be proud of it. So
yeah. Go ahead, Jamie.
Yeah, I think very similar to Kate and Seji on the impact for employees, because it's very personal. It's stories. We're somewhat removed from patients. So in our world, we work through physicians who act as investigators, who see patients, and patients are essentially anonymous to us. That's part of the blinding process in clinical trials. But I think with n-Lorem, it becomes very personal. The stories are hugely important for our team. And it's that pride and impact. And I don't think it's about providing therapy for free. As Kate said, it's about providing therapeutic options to people that would otherwise be left behind by the system. On the selfish side, we learn more about the rare disease space, the communities, the caregivers, the advocacy, and we run trials across the whole rare spectrum. And that allows us to think about how to better design the trials. Endpoints in rare disease are horrible, as we all know, and it's very difficult to build that clinical evidence. But if we can get others and regulators to really sort of hone in on new and better endpoints, biomarkers, the real measures of clinical success, that's going to help us be a better partner for other pharmaceutical companies that are developing therapies across the rare spectrum. So we do get a business benefit.
Yeah, so today we've been hearing about this patient family. Is that something that you also pass along to your employees? By the way, that thing we're doing with n-Lorem, we just started treating a patient. Her name is Susanna, like that.
Yeah. Oh yeah, absolutely.
Yeah, all of you. Yeah. Wow. So go back to this idea of partnering and collaboration. When the idea first came to you and you agreed to do it, did you set it up for a certain amount of time? Like, what's the future of this partnership as far as the four of you know? Says you're nodding. You can start.
Well, again, this opportunity will give us lots of opportunity to think about, how Stan was mentioning about 9 INDs and the hundreds of applications is waiting for. So we need to have the more improvements of productivities and the productions and kind of stuff. So then we can increase some capacities. And so there's a tons of capacity probably, but with economical models as well, we need to do the more like automations or simplifying the process and things like that. So we try to think about, give us a good opportunity to think about how we can improve what we do today to do better a little bit, just better a little bit, and develop the justification of the costing model for this one. That's
So when I first heard of this idea, I thought this is a really great idea. I almost thought it was almost like a hobby for Stan. He's going to sort of put his industry intelligence to use in this way, and they might get a few patients, and we'll see how long it's going to be. But as quickly as it's grown, we just saw the numbers today, 200 and something applications. Is that, you weren't expecting that when you first signed the partnership, were you?
No, I did not. Well, I did not. He was talking about a couple of things, but so that's why we cannot commit at this point 100 of the application for our manufacturing only, though. But there is many way to think about for the future wise and improving our systems and the flow and just a little bit shrink for those flows that will give us some more speed and capacity, I guess.
Right.
Yeah, well, I think one of the things that we are learning is how to use our technology for a broader range of patients, but also how to make it more efficient. And so, long term, we're committed to be a partner with n-Lorem. As n-Lorem grows its staff, there'll be more day-to-day operations that's being done by n-Lorem. But I think we are still investing in the technology, and all those investments where we make improvements in the technology or increased efficiencies in how to find drugs will transfer to n-Lorem. And so I think, we have a long-term commitment to n-Lorem, and n-Lorem will be the beneficiary of a much larger investment in improving the technology and being able to apply it for the nano-rare patient populations. And I think that's what we're committing to do.
Yeah. How about Biogen?
I think a lot of our collaboration now is cognitive, and that's pretty free. And I think it's easy to scale that as well. It's just a matter of, because we have the commitment, we're willing to put in the time to do the additional assessments, and like Frank says, we are supporting this ASO platform from a manufacturing side, and what we've learned as well with Ionis, and we're happy to continue that knowledge transfer. And I'll just want to come back to the point made around biomarkers and endpoints. That is something that Biogen gets as well.
Yeah.
We're in this space as well, and we are dealing with, not the nano-rare, but rare conditions that have similar endpoints, and we work with regulators on, and many of these are partnered with Ionis. So we will learn, hopefully, it's wonderful that part of the remit is to actually make the information known to the community and publish, because this is how we will all benefit from this.
Yeah. Kim, you want to add?
Yeah, for us, we're evolving in our relationship. So we've done a lot of knowledge transfer, and n-Lorem are very self-sufficient in clinical development. We will likely be providing additional support as they globalize and think about market access and reimbursement. I think there's an important thing. Not for profit doesn't mean not for revenue and not for reimbursement. I think, as Stan has alluded to, and Chris as well, the ability for n-Lorem to look at others that are not the patient and family, I think that's the key element. But the insurance companies, the healthcare systems, and others, how do you build a model that then brings revenue in that can then optimize and maximize the scaling of n-Lorem and what it does and how many patients it can reach? And I think we have some expertise in that space as well. But that's going to be the critical next phase around sustainability and growth.
Yeah, I have one question left, but I wanted to see if there are any questions from the audience. We've got about 5 minutes left in this panel before I throw another question at this group. This is your last chance. No? All right, so the final question I wanted to ask is this idea of n-Lorem replicable? If some other group came forward and said, hey, I know that you worked with n-Lorem. We're a group for Huntington's. Can you help us? We're going to do this nonprofit thing. Is this actually something that can be done by more people in this industry? What do you think? Jamie, let's start with you.
That's an interesting one. I think the platform, the unique elements around n-Lorem are the platform, the regulator's willingness to accept the sort of science, the integrity of the manufacturing and the product and the safety profile. I think that is fairly unique. I'm not saying it's completely unique.
With the long history of antisense behind it, right?
Yes.
Yeah.
That has been hugely helpful. I think other disease areas that may be in and around this space have less proven technology, less proven sort of manufacturing and product integrity, but it's not impossible. But I think the science is so well understood and the product quality is so good. I think it allows that accelerated process from IND to single patient treatment. I think in other areas, as I see it at the moment, most of those would require to go through some staged clinical trial process, particularly focusing on that early safety and even long-term safety as well. But it's not impossible.
Yeah. What is the rest of Frank? What do you think?
I think it will be. I think other platform technologies will be applicable as they get developed. That's still early for them, but I think given what we've been able to do and Stan's leadership, I think other platform technologies will be able to apply it for nano-rare patients as well.
Okay.
I think it's, I was going to say, I think the ASO platform really enables this, right? And I don't know of any other platforms yet, Frank, that might get there, but I hope. I mean, ASOs didn't exist as therapies, not too long ago.
Yeah.
So there could be something out there that could develop this kind of body of knowledge that will enable it to be used in that way. Personally, not yet do I have an idea, but when I do, I'll go to Stan for some advice.
Well, it's two things. It's the platform, but also a person who has great
Yes.
credibility, right?
Yes.
So if I came to you and said, "Oh, I'm going to start this CRISPR thing,"
Yeah.
that's you wouldn't support it, but
Well,
you wouldn't support it. Let's be
Yeah.
honest. Seji, do you have any thoughts?
Oh, I think like James said, that same things. And ASO, this platform is we worked together with Ionis for a long time, so we know the ASO better. So that's why this will fit better at this point. But again, the new type of the platform could be a possibility for the future wise. But again, we got to do step by step as well. So
Yeah. We're down to about a minute. Any closing thoughts from any of you about the day, the patients, partnering?
I just want to say at the beginning, you asked me shareholders.
Yeah.
Shareholders have a different idea, but when I heard this opportunities and I thought any shareholder will agree with us. So this is just a great initiatives, and we're very proud of the part of the Stan's team.
I was just going to say, it takes a village. And I want to just recognize that this village that has been created by Stan and all of you here, that is just amazing. And we're proud to be part of it.
Mm.
Yeah, no, I'd also like to thank the members in the audience and the patients and their families. That's our source of inspiration. And it's what, again, brings us, all of us, to work every day.
Yeah, perfectly said. We do a lot of other work and deal with a lot of other issues. The ability to impact individuals and families and their caregivers, it's why all of us do this work.
Yeah, well said, well said. Okay, that brings us to the close. Do you want to say something, Amy, or no? Yeah. So lunch?
Not quite.
So then
We're dismissed.
Yeah, we're dismissed.
Well, first of all, am I on?
Yes.
Thank you very much to all the moderators of the panels and all the panel members, and especially the patients and folks who came to share their personal stories. And thanks to all of you who said nice things about Stan Crook. He's a really great guy. And so I've written all those down for the bad days. It's been a deeply rewarding morning. I think you all agree with that. And we have, I think, an equally rewarding afternoon for you. And this is the first meeting. And one of the things that is great to do in a first meeting is to establish traditions. Traditions don't happen, you establish them. And so with this, we're starting a new tradition. And that is, we're going to recognize every year what I'm going to call heroes of the Nano-rare. And these are individuals who have no stake in the game other than the empathy that they feel for others, other than the support that they feel for other human beings who have problems, who are all busy, they have full-time jobs, and yet they find the time to contribute to n-Lorem. And I could name 20 or 30 heroes of n-Lorem today, but I think that would probably upset people because you're probably ready for lunch. And so I'm only going to name 3. But they're all very special people who have contributed to n-Lorem in many, many ways. And the first, Frank and I trade awards. I give them, he takes them. And we also trade a variety of other things, communicable diseases on occasion, and a variety of things. I mean, you can't be together for 40 years without, lots of transmissions, right? But our first hero of the Nano-rare is Frank Bennett. Frank has a busy full-time job, as you all know. He has been involved in n-Lorem selflessly and tirelessly from the day I brought it up to him. He helped establish the systems and processes that we use at Access to Treatment Committee, which is a critical thing that you heard a lot more about this afternoon. He served as liaison between n-Lorem and Ionis. And it's like any other collaboration, there's some good days and there's some days when there's some stresses, and he has to deal with all that. And he will continue to contribute. And of course, as a member of the Ionis team, he contributes continuing knowledge and expertise. So Frank, welcome as hero of the Nano-rare. It takes two women to bring these awards up. And can you show that, Frank? Do your job right, Frank. The beautiful awards I think Sean made, which is Kim's husband. Well, we'll give him credit for it anyway. And so, Frank, congratulations and thank you so much.
Thank you. It's been a pleasure. For many of you may not know this story, but when I was applying for graduate school, I walked into the chairman's of the department's office, and this guy in a business suit walks out, and I thought he's some kind of salesman there. And very dapper dressed, and I grew up in the sticks of out in the middle of nowhere. And the chairman, as he was walking out, and I went in to introduce myself to the chairman, he says, "You need to pay attention to that guy, and he's going to go somewhere." So Stan, it's been a wonderful 43 years. Thank you.
Okay. The next hero of the Nano-rare is John Maraganore. John can't be with us today because he's wealthier than I am. He's closing on a new house in some ski resort somewhere. He should be here working, is what I think. But that's chat we'll have on another day. John, of course, was founder and CEO of Alnylam. He's tremendously busy. He's on multiple boards. He's a mentor to many, many young biotech CEOs. He's on the industry associations and so on. He's a busy man. But I wrote John a note and introduced the idea of n-Lorem to him, and he got back in touch with me. And ever since then, anytime I've asked for anything, he's been there to help and to do what's necessary. And of course, he's not the hermit I was during my career, so he knows everybody. And so anytime I need to, meet someone I don't know, I just ask John, and he calls them up, and then we get together. So again, John Maraganori in absentia. Thank you, John. And our final hero is Max Moore. Max has been at Ionis, like most ions, a long time. And I think people in the manufacturing, analytical, and whatnot space know Max. But Max has been, organizations have a hierarchy, but then organizations that are really good anoint their local heroes. They're the people who, when you really ask somebody who they trust, that's what you'll hear. And Max is one of the local heroes of Ionis. He's a person who everyone knows his stuff. Everyone knows is a person of the highest integrity and the highest commitment to doing the right thing for the right reason. And Max has been instrumental in introducing us to all of the manufacturing, analytical, formulation. All of those organizations has brought all those organizations to the table, and we couldn't do it without them. They're all giving us either free goods or tremendous discounts that make it possible for us to do what we do. So Max is another guy who's worked with me for probably way too long, but Max, come on up and accept it. Max. And it's winter, Max. When are you growing your beard? In a few weeks, you won't recognize him. It'll be mostly beard that you see.
As much as I love you, like the patient-centered, demanding dad I never had, there are only certain heroes here, and it's the patients and the families. So thank you for the award, but as far as I'm concerned, it gets transferred to them.
That's the end of the morning. I think Max said it. Here's to the heroes. Here's to our patients. Let's enjoy the rest of the afternoon and everybody get back to work, because we have lots of patients who need us to work. Okay.
So, yes. At one. All right, welcome back. How's everybody doing? Colloquium? Everybody practiced it at lunch? Next year we'll name it something else. It'll be easier to say. So this afternoon we've got some n-Lorem folks who are going to come up here and tell you, really pull back the curtain and tell you what we do in a little bit more detail than what we might have on our website. And so yeah, thanks for joining us. And I'd like to introduce Julie DeVille, who's our Executive Director of Drug Discovery and Development, to really pull back the curtain on how we make these optimized ASOs that we talk about.
Thank you, Amy, for the introduction. Hi. Today I'm going to talk to you about how at n-Lorem we use our ASO experience and scientific excellence to drive our discovery and development platform. I know that not all of you have a scientific background, so hopefully I can do this so that you can follow what I'm saying. So let's start at the very beginning. Genes are made of DNA, and DNA is made of 4 building blocks that we call nucleotides or bases. And we refer to them by their symbols, A, T, C, G. And these nucleotides are organized in a very specific sequence, so very specific order, so that information can be encoded in our genes. And from there, this information is transcribed into messenger RNA, which really bears the instructions to make a protein. And proteins are complex molecules that are really doing all the hard work in the cells, and they're important to the structure, the function, the regulation of the body. So let's talk about mutations now. What's a mutation? A mutation essentially is a change in the normal sequence of nucleotides. I heard someone call it misspelling this morning, and I really like this analogy. And so it can be a deletion or an addition of a single or longer stretch of bases, but it can also be the substitution or replacement of one base for another in the sequence. And more specifically, we refer to these as SNP, SNP, single nucleotide polymorphism. And I'll talk about that a little bit later. It's kind of important when we design ASOs. And most of the time, mutations are either corrected or they're benign or silent, which means that they don't have any consequences. But other time, occasionally, mutations can change the critical part of the instruction to make a protein. And so as you can imagine, different types of mutation will have different consequences. For example, a mutation can cause a protein to become inactive or be degraded by the cell. So the protein is no longer able to do its normal job, and we refer to these as loss of function. Other times, a mutation can cause a protein to acquire a new function, so a job that it wasn't doing before. And these mutations are referred to as gain of function, and often they're toxic. And so when we get an application for treatment at n-Lorem, it's really important that we do a thorough evaluation of the nature of the mutation, its consequences, and so that we're able to come up with an adequate ASO strategy for each patient. As you can see on this chart, a lot of our patients have a gain of function mutation. And so the rest of my talk will focus on this type of mutation. Gain of function mutation can be addressed by RNase H1 ASOs. So those are ASOs that will allow us to downregulate a gene. Many of our patients will require an allele selective strategy. And so Stan talked about that this morning. And we need to use this approach when the function of a gene is essential to the cell. And so we need to be able to knock down the bad copy while maintaining the good copy. And as you can imagine, it's a lot more difficult to come up with an ASO that will have this level of selectivity for basically a difference in sequence that is only one base. And so I'll cover that in more details later in my talk. As we discussed this morning, our platform is based both on ASO technology and FDA guideline. So Stan talked about that, how this technology has been validated. We know a lot about it, and it's also versatile. So we can change the sequence to target a different gene, and we can predict what's the dose that's going to be efficacious. And we can also predict adverse event quite well with this technology because we know so much about it. It's also cost effective, which makes it scalable. And importantly, the FDA, with those 4 guidelines that were issued, really provided a roadmap of what is needed to advance these molecules to the clinic in a very streamlined and efficient manner. This slide here is just to show you a number of articles that our group, STAN, has published recently on how RNA-targeted drug discovery can be applied in the field to address the needs of the patient. And so I encourage you to go and do some reading if you want to learn more about the technology. And now we're going to switch gear and cover our process to discover and develop an optimal RNase H1 for a patient with a CNS disease. And so each of these steps are critical in coming up with the best possible ASO. And as Stan mentioned, we can't skip a step. And the minimum criteria that we use at each of these steps, as well as the number of ASO at each step, is also really important and based on decades of experience, as well as thousands and thousands of ASOs that have been screened over the year. And so now I'm going to cover every step in more details. The first step is, of course, to design the ASOs. And I'm going to focus on allele-specific ASOs here. So for each gene, of course, we have 2 copies, one that come from our mother and one that come from our father. And the pathogenic mutations, the mutation that is causing the disease, can be present on either allele. So for the sake of this example, we're going to put it here, the red X on the top allele. And so the messenger RNA that will be made from that allele will be referred to as the pathogenic mRNA because it bears the pathogenic mutation. And the other messenger RNA will be referred to the wild type mRNA. So in addition to the pathogenic mutation, we can have benign changes in bases. So those SNPs that I mentioned at the beginning, they can be present on either allele. And depending on the gene, there can be many SNPs or there can be fewer. It's really depending on the gene and on the individual. And so they're identified here by the black X's. And these are really important because each of these sites provide us with an opportunity to design allele-specific ASOs, because at these specific sites, the sequence is different on both allele. And so we can use this to design ASOs that will recognize or bind just to one copy of the mRNA. And in addition, we can also use the sites that are opposite. So the site on the pathogenic mRNA that have the normal sequence, but that are opposite to a SNP, because again, at this site, the sequence is different. And so we can design ASOs that will be specific for the pathogenic mRNA. And so ideally at that step, we want to have design between 400 and 600 ASOs. And we like this number because when you start with such a large pool of ASO candidates, then it gives you the best chances to identify the best ASO. And so I'm going to explain to you now how these ASOs are working. So you have the pathogenic mRNA there with the pathogenic mutation, as well as the wild type mRNA. So our ASO will bind here where the sequence is unique to the pathogenic mRNA. And this will create a duplex of ASO mRNA. And this duplex is recognized by an enzyme that we call RNase H1, and this enzyme will cleave the duplex and trigger the degradation of this duplex. And so there won't be any mutant protein made from that mRNA. On the other hand, on the wild type mRNA, the ASO is not binding. There is no recognition by the RNase H1. Therefore, we can maintain production of a healthy protein. So this is how we can make allele-specific ASOs. And so once we have designed these ASOs, the next step is to test them for potency. So potency is how effective these ASOs are at knocking down the expression of the pathogenic mRNA, or reducing basically expression of the pathogenic mRNA. And so in this example, we're looking for an 80% reduction of expression. So I draw that purple line where the threshold is. And on this curve, each point is a different ASO candidate. And so in this example, we would take these ASOs in the orange box and move them forward to the next step. The next step is very similar. We're looking at potency once again, as well as selectivity for the pathogenic mRNA versus the wild type mRNA. And so you have your pathogenic mRNA here in red, and the wild type is in green. And what we're looking for is a dose-dependent reduction of expression, because we want to understand that when we increase the dose of the ASO, we have a dose-responsive effect on the expression. So that allows us to predict, with whatever dose of ASO, the kind of effect we're going to have downstream. And so in this example here, really what we're looking for is, as I mentioned, a reduction of the pathogenic and a maintenance of the signal for the wild type expression. And so, for example, this ASO number 7 here, we would move it forward to the next step. We would probably also move ASO number 3 forward, and maybe even ASO number 6, not 6, 5. Because the effect on the wild type expression is not significant compared to the pathogenic mRNA. And so at this stage, we're now looking at an assay that we call the BJAB assay. So essentially, that's an assay that is designed to measure activation of the innate immune system. And so some ASOs are known to trigger an inflammatory response, and that's not desirable. And so this part of the screening is really designed to identify those ASOs that would trigger this type of response. And basically what we're doing is treating peripheral cells. So the cell line is called BJAB. That's where the assay takes its name from. And we're treating these cells with all of our ASO candidates that we have moved forward. And we're basically comparing them to 4 control ASOs that we know are either highly inflammatory or moderate, or basically not inflammatory. And so those are the green, blue, yellow, and red lines on the graph. And we're really benchmarking our candidate ASOs against these 4 controls. And in this example, ASOs that are in the orange box would move forward to the next step because they're considered safe. The next step is looking at potential off-target effects. So this is really looking at whether there's any non-specific binding of our ASOs to messenger RNA from other genes. And this non-specific binding of ASO could, in theory, reduce expression of genes that are critical for the cell. And although it's extremely rare to have toxicities that are related to off-target effect, we still want to do our due diligence and really look at this in more detail. So it's a two-step process where the first part is a computer-based analysis where really we're going to highlight any genes that would have a partial match with the ASO sequence. And then once we have identified those genes, we will run an in vitro assay where we're going to use cells, treat them with our ASO candidates, and then do PCR to evaluate level of expression of those potential off-target genes. On occasion, we might have a slight effect, minor effect on these genes. And at this point, it's really important to have a really good understanding of our patient condition to really be able to do a risk-benefit analysis here and understand if there's a true liability of this effect. And so once we've completed that step, ideally, we are probably around down to around 20 ASO candidates. And so we'll take these 20 ASO candidates and we'll do a small batch synthesis of research grade material. And we're going to have, even though it's a research grade batch, we still want to have strict criteria for purity and other aspects as well, because the next step will involve animal testing, and it's important that animals are exposed to a batch of material that is comparable to what the patients will receive. And so the first animal study that we're running is a tolerability study. So this study is not designed to look at efficacy, but really designed to identify poorly tolerated ASOs. And we use a route of administration that will be similar to the route used in the clinic. And the animals will receive a single dose at quite a high concentration of those top 20 ASOs from the in vitro screen. And we're going to evaluate the animals over a period of 8 weeks. And we look for any abnormal clinical signs, any effect on neurobehavioral parameters. We look at their body weight. And then once we reach that 8-week period, animals are taken down and we collect tissues of interest. So for example, a CNS target, we would collect spinal cord and brain. And we look for specific markers of inflammation and toxicity. And then from this step, along with all the data that we've accumulated from the very beginning when we started the first, the single dose potency assay, and then the dose response curve and the BJAB and the off target, we take all of this data, look at it in the context of the patient, and identify which ASO has the best potency, the highest selectivity, and is the best the best tolerated ASOs. And that's the one that we're going to move forward to become our clinical candidate, basically. And so at this point, we're going to have 2 steps that will run in parallel, just because that's how we can optimize the timeline. And we'll move into GLP toxicology study. So this study is really important. It's the one that allows the FDA to approve the treatment, and it's designed basically to look at toxicity after repeated dosing over a chronic period of time. So this is designed really to represent a situation of a patient receiving a drug over a long period of time. And the specific design of this study will change based on the organ that we're targeting and the ASO strategy that we're using. But what's important to understand is that this study is strictly regulated. All parameters are controlled. It can be, the conditions in the animal room, the temperature, the humidity. It's the temperature in the freezer where we keep the samples, where we keep the drug. It's how technicians are trained to perform that study. Really, all parameters are controlled, and this ensures that the data that is generated is robust and reproducible. And in this CNS example, animals will be injected once a month by intrathecal injection, and we will look at them over a period of 3 months and Some parameters that are evaluated are general, such as body weight, food consumption, but other parameters will be specific to the organ that we're more interested in. And so it would be neurobehavioral assessment here that we do on a regular basis over the course of the study. And then at the end, animals are taken down. We collect tissues to look for any abnormal pathology finding. And we also look at drug level in those tissues to understand the distribution of the drug. In parallel, we'll trigger GMP manufacturing of our drug. And so similar to GLP tox, GMP manufacturing is a regulated activity. It operates by strict rules to ensure quality, reproducibility at every step. And we're going to have, once again, very strict criteria for purity, salt content, sterility, endotoxin level to make sure that the drug is suitable for human administration. As we mentioned before, single batch of an ASO manufacturing provide enough material to treat a patient for 5 years, 10 years, or even more. So it's very cost effective. And then depending on each institution, so where our patients are going to be treated, we might need an additional manufacturing step, which is basically taking this powder ASO, formulating it in a liquid form, sterile, that will be aliquoted in vials for administrations. This is called sterile fill. So the FDA guidelines have outlined a fairly lean, non-clinical pathway, fairly straightforward, which makes the timeline a lot more interesting for a nano-rare patient. And that's why the rigor and quality are of the utmost importance at every step. As you can see, non-clinical data represents 60 or even more, maybe 75% of the content of the IND, and is really a pivotal part of getting that drug to the patient. The rest of the IND is really focused on the patient. At n-Lorem, we're very patient focused, not population focused. So the rest of the IND is around our patient and a clinical protocol around it. And Lori's going to cover that in more detail right after me. And so we have developed a mutation-driven discovery and development platform that is scalable, while at the same time being tailored to the needs of each patient. As you can imagine, this requires very deep ASO expertise and experience, as well as agility and flexibility in our decision-making process. And I'm missing a slide. Sorry. Okay, and so really what is the most critical is to integrate decision from top to bottom. So each step of this process is extremely difficult, but integration throughout the process is even harder. And we really perform this risk-benefit assessment in real time as our ASO program progresses, but as well as the patient phenotype also potentially progress. And so essentially, my slides are in the wrong order. Essentially, the most important question that we ask ourselves at every step of this process is whether this ASO is adequate for this specific patients. This is really the most important question that we ask ourselves all the time. So thank you for your attention. Thank you to all of our supporters and partners in the industry. We couldn't do that without you. We'll take questions at the end. So right now I'm going to pass it on to Lori.
Thank you. Hi, everyone. It's really my pleasure to be here today and to give you an overview of our clinical infrastructure. So I will cover 3 topics today. Really, mainly, I'll kind of show you how, a deep knowledge of experience in experimental ASOs and experience in managing trials really supports the professional management of our patients, and how we also are able to then customize the treatment of each patient. I'll then give you a little bit more, evidence of clinical benefit, more than what you saw from Susanna's videos already this morning, but I'll give you some more quantitative data. And then I'll also talk about the safety of our ASOs and provide some information on the safety data we have to date from our trials. So we have a bias to treat patients, right? And that bias is really supported by all the expertise that we have. And that expertise allows us to inform the benefit risk equation, really, right? So we all realize there is a risk, an inherent risk of not treating a patient with a progressive debilitating disease, right? But we also know, there could be a risk of treating a patient with an investigational ASO, but we know how to mitigate that risk. And again, not to come back to this, but I think 30 years of safety experience that, Ionis has developed and, is still going on in clinical trials today with those ASOs. So that robust experience in ASO discovery and development really kind of supports all the safety experience that we have to date. We are constantly learning from our own trials, right? So we have a few trials ongoing. I'll talk about those later. We're also learning from the commercial trials run by Ionis, by Biogen, by Roche that use, ASOs. And so we learn from those and we bring those learnings to our physicians as well. One thing that's important to understand is, for all our ASOs, we really start with a low dose, right? And I think that's very important. You start low and you go slow. And so you dose escalate in a very stepwise fashion. At every dose, you can go back, look at the results, and then decide, make a decision on what the next dose should be. So it is very truly individualized treatment. And then we have our, we're creating a safety database for now the trials that are in the clinic. And we have a data safety monitoring board that will be reviewing this data on an ongoing basis. So again, we want to be treating as many patients as we can. But we know that it's very important in order to do this successfully and professionally. So when I talk about successfully here, I mean, we need to treat patients with an optimized, individualized ASO that is of good quality, that is safe, and we need to be able to track quantitative and qualitative outcome measures in each individual that is important to that patient, right? So we have to be very focusing on the patient and what is important to the patient. When I say professionally, I really mean more of the how. How do we do this? Well, we do this by partnering with capable and invested physicians and institutions. We are collecting the data in a very standardized manner using a validated system. It's a REDCap system that n-Lorem houses. And we are also, working with the FDA on an ongoing basis. Every time we submit an IND, we get responses. We work with them, very collaborative environment. And so we learn with every IND that we work on. And so really, to do something successfully professionally, you need experience and infrastructure. One thing, side note, I wanted to say here, when we talk about success, we don't necessarily mean, is the ASO working? Is it efficacious? When we talk about success, did we ask the right question? Did we do everything we could to try and answer a scientific question? And if we get an answer to that question, we reach success, but it might not always be that we cause clinical benefit. So I think it's very important to realize that. So at n-Lorem, we have years and years and years of experience. And we're very lucky to be also collaborating with investigators with a lot of experience. One thing that was very important for us is to build a strong clinical infrastructure. We need to minimize the potential risk of ASOs, maximize the opportunity for benefit, and really learn from one patient to the next. So in order to do this, we assembled a very versatile group of leaders with deep clinical experience. And these are leaders that have experience in research, clinical work, manufacturing, FDA interactions, project management, clinical operations, clinical trial oversight. Basically, every function that you have in a big commercial company, we have at n-Lorem. We might not be many people, but we wear many hats and we have that knowledge. And again, we also obviously partner with, subject matter experts that help us, find the right treatments for our patients, look at the right outcome measures, and then also, check safety with us. And like Stan mentioned this morning, we really quadrupled the size of the organization in order to meet the demand. So when n-Lorem, started, they realized that they needed to have multiple checkpoints where independent peer review would really support the quality of the work that we do. And so it was very important to reach out to people outside of the organization. And so I'll go through various, a few committees that we have at n-Lorem that are really important, and I'll try to tell you what they all do. So the first committee is the Access to Treatment Committee. So when an application comes in that a physician submitted to n-Lorem, the ATTC will look at the application, look at the specific genetic mutation, determine, is this genetic mutation amenable to an ASO treatment that we could develop? This is really made up of 13 members, and I tried to look at everybody's experience, and I think we are more than 200 years of experience in all the fields that we need: research, drug development, ASO technology, ethics, and patient advocacy. We then have a study treatment and assessment review committee. We call it the STAR committee, and half the time I can't remember what STAR stands for. But I know that that is the committee that advises physicians on which outcome measures to use for their patient. Obviously, the physician treating the patient knows best what measure to use, but the STAR committee can really sometimes come up with different ideas or different ways of doing things. And so it's always been a really good interaction when we have a STAR meeting. That committee is made of 4 members of a combined 80 years experience in all the fields that are required for that committee. We also partner with experienced investigators who have robust institutional support and staff. And we call those our early adopters, the one that we've worked with to date. And we've learned a lot from them on how to try and do this at their institutions. And we try to take the learnings from our early adopters, and we'll bring that information to the newer physicians that are working with us. And it's been working pretty well so far. And then the last committee that we actually set up pretty recently, now that we actually have patients in the clinic and we need to start tracking clinical data, is our Data Safety and Monitoring Board. And so that committee will review the safety data across all our n-Lorem programs. We might have to expand that committee because once we get all our programs in the clinic, it's basically we're just going to meet on a weekly basis. But they will be looking at the safety and see if there's safety signals across our programs and help us advise on next steps. Now, besides the committees we set up to make sure that there's checkpoints, there's really one big body out there that actually checks all our work every time, and that's the Food and Drug Administration. So the FDA will review, comment on, and approve every IND before any treatment can be given, right? And n-Lorem has received approval across 4 divisions of the FDA: Neurology 1, Neurology 2, these are 2 different divisions, trust me, cardiology, and ophthalmology. And we've received positive interactions from all these divisions. And I think this emphasizes 2 things. One, the quality of the IND documents that actually n-Lorem puts forth for the FDA to review. And 2, also the fact that the different divisions at the FDA actually read the guidance documents, in a similar way. So they actually interpret them in a similar way, and they, approve these N-of-1 trials. Once the FDA approves our INDs, then we have yet another checkpoint, and these are the institutional review boards. So these are the review boards at the various research institutions or hospitals that will review the protocol, the informed consent, and they're really there to protect the rights and welfare of the patients. And like we said, we've worked with many now, and basically every institution has a different IRB. So, it's really a lot of people looking at our work. So you might say, well, all these committees, it's going to take forever. But, we know prudence and cautious is important. That's why we have the committees. But we also know that time is of essence. And so we try to do things in parallel to be able to move faster to get to a treatment for our patients. So I'll run through you, to show you what that means. So this is really kind of like the workflow. If you look at just like the research and GLP side of things. So at that first star on the left, I hope it's left for you too, I get confused. That's when an ATTC, when an application comes in, then we need whole genome sequencing and patient cells for us to be able to start ASO discovery. So once we have that, ASO discovery starts, and Julie did a great job at telling you everything that's involved there. That star after the ASO discovery is when we decide on an ASO lead. We put it into GLP tox, manufacturing, and then we can submit the papers for patient treatment. So that's one part of the work that's going on at n-Lorem. But in parallel, there's all these clinical activities that have to happen. So pretty much as soon as a program moves into ASO discovery, we start working on treatment goals with the physicians and prepare for them to present at the STAR meeting. So once we have finalized the treatment goals, we can then go on and write the protocol and finalize that individualized protocol and informed consent. Once we have a protocol that's done, we can then go on and develop an electronic clinical database that will be able to capture all the treatment clinical data for the patients. And then again, in parallel to all that work, we also work on the IND documents and on the submission process. We submit to the FDA. Once the FDA approves it, we submit to the IRB. Once the IRB approves it, then we can start collecting, or we can start treat the patient and collecting all the clinical data post-treatment. And so here, the different colored stars show you where all the committees fall. So the ATTC committee is obviously at the very front. The STAR committee will review the treatment goals. External roadblocks, FDA and IRB will be the ones giving us that final okay. And then the DSMB will review the data once the patients are in the clinic. So we've set up this infrastructure. We are very, not optimistic. We know that this infrastructure is scalable and that we can support, the treatment of many more patients to come. We know that because we've, institutionalized standardized protocol language, template informed consent language. We have a streamlined process to support IND filing. It's still a ton of work, but we know the steps to do it and to do it quickly. We've also had really good interactions with the FDA and responding to FDA queries. Again, I think what is really important is our electronic database. It's a REDCap database, an academic one that a lot of our investigators have used for their own clinical trials. We've been able to standardize, some of the forms that we use. We've developed a large library of electronic case report forms that we can use from one program to the other program. And because, for example, for a neuro exam, we've made sure that everything that we think is important is on that neuro exam case report form, and we can use it for one program and another program and another program. And so we can actually, easily afterwards compare the data from one program to the next. And I think that's really a strength of how we've set that entire infrastructure up. So let me give you a little bit of where we are today. And, Stan kind of gave it away this morning, but it's okay. We've submitted 9 INDs in the last 13 months to 4 different divisions. Most of the INDs to date, so 7 of them have gone to the 2 neuro divisions, 1 to the cardiology division, and 1 to the ophthalmology division. n-Lorem pretty much authors 98% of that content. We work with our investigators really to describe the patient, because there's a section in there where we describe every patient and their clinical status and why they should get this N of 1 medication. We've had more than 25 FDA interactions. This is between meetings with the FDA and responses with the FDA, and they've all been very positive. And we are expecting to submit 2 more INDs before the end of the year. So once we're done here, we all better rush back to California, wherever we're from. But yeah, so we're expecting to do 2 more, at least this year. In terms of clinical metrics, 5 patients have been dosed to date, 4 CNS patients, 1 eye patient. If you do the math, you're wondering what's up with the other 4. We still have 2 INDs that are under FDA review, 1 IND is under IRB review, and 1 IND just received IRB review, and the site is planning, is setting up for a visit for the patient. So that gets you to 9. Patients have received between 1 and 6 doses, and I'll show you a table later to tell you what that means. And to date, I think what Stan also mentioned earlier, we have not had any ASO-related adverse event or serious adverse event, which is fantastic. So now let me change gears a little bit. Now I just want to show you or give you an example of how we really customize the needs for each of our patients. So like Stan mentioned this morning, right? 2 patients can have a mutation in the same gene, and it can lead to very different phenotypes. And so it's very important for us when we work on the treatment goals that we really find treatment goals that are important to a specific patient, right? So I'm giving you here an example of 2 patients that, we have in our pipeline. 1 has started treatment, 1 is about to start treatment. Both of them has different mutations in SCN2A gene. So 1 is more of a gain of function, the other one is a mixed gain of function, loss of function. However, there are some symptoms that are similar across both patients. And so we'll track seizures for both patients via seizure diary. We'll also track behavior and communication and development in both patients. But then both patients also have, different symptoms that we'll track. And so for 1 patient, it's really focusing more on the behavior and communication. So we have added measures in that realm. And then for the other patient, it's more motor issues. and gastrointestinal issues. And so we'll have scales that focus on, those symptoms. So this is really just, very high level, obviously, but just trying to show you how we try and individualize treatments for the different patients. Stan, obviously, this morning already showed you great videos from Anna and Susanna. And really, I think especially Anna led us to, understand that even if you start treatment late, you can still have a benefit, or what, some people would consider late. And so it's never too late to try something. What I'm going to try and do here is give you a little bit more quantitative data. You've all seen videos of Susanna, so I think everybody has drank the Kool-Aid, hopefully, on that treatment is working for Susanna, but I'm going to try and give you quantitative data. One thing I want to mention here, because we are really normally blinded to every application that comes in. So we do not know the name of the patients that come in unless the family identifies themselves to us. So obviously the Rosen family has identified themselves, but Susanna is also in our database tracked by a number, which is 255. So just wanted to make that clear. Some patients we know, some patients we don't know. So she has a mutation in KIF1A gene, and as Stan said, this encodes for microtubule motor protein that's responsible for transport along axons. When Susanna started treatment, she was 8 years old. This was just a year ago, had severe epilepsy, intellectual disability, and very, very painful and debilitating neuropathy. She also had to start using a wheelchair because she became non-mobile. This is the protocol that was written, or like timeline protocol that was written for Susanna specifically. So a couple of things here I want you to note. First of all, it's a 12-month study, right? She's where the big red star is. She's received 6 doses to date. The doses are also mentioned in the graph. So you see we go, like I say, we start at 20, we go slow and we increase and kind of wait to see what happens and go to the next dose. You can see all the assessments that are done. So a lot of it is for safety, vitals, EKG, overnight EEG, but then also some, to measure how she's walking, 6-minute walk test and so forth. The one thing I want you to realize is that little red star in the middle. These are assessments that track gross motor function and fine motor function or fine motor skills. And these were actually added in the middle of the trial because these were things that she now was able to do. And we didn't really know to track those at the beginning because she wasn't able to really do some of those assessments before. So I think this is also something that we learn, and it's fine to add that later on and to start tracking it later on. All right, so quantitative data here. So just to orient you, it says parent-reported seizure data on the top, falls in the middle, and quality of life on the bottom. If you look at the y-axis, that, between the first 2 red vertical lines, that's the pre-treatment time. So that's our baseline, pre-treatment baseline. Every vertical line thereafter is a dose, right? So she had a ton of seizures before treatment. And over time, and really actually pretty quickly, the seizures started to come down, right? And so she still has some seizures, but it's definitely much reduced from what it was before. In terms of the falls, I think you can say, see the same, but I think what's important to note here, remember she was really using a wheelchair a lot at the beginning. So she was actually not walking that much and would then fall because of the disease. But now, even though the falls have decreased, she's actually walking a lot more than she did at the beginning. So I think this is really something to kind of point out and something that, one needs to understand if you also understand what the child is actually doing during their day. And then the last one is, quality of life that's increasing. You always want to see that increase over time. And then finally, I think what is really important is that there's no ASO-related adverse events in Susanna. And then additional things, again, you've seen the video this morning, so I feel this slide is a little bit redundant here, but additional comments that we got from the families on things we were not necessarily measuring in the trial, but, things for us to think about whether we should include it in future trials. Her tremor is almost all gone. She can hold a baseball. She made a bracelet for me today, so I'm very happy about that. She can sing, something she hadn't done for a while. And what we heard is also the pain has really gotten much better in her hands and her feet, but the pain does come back towards the end of the dosing interval. So again, this is something that with the physician, the family will have to see and try and fine tune and, maybe increase the dose or reduce the dosing interval. So all things that we'll have to think about. Let's think through. And she's able to make it through the day without a nap, which I think that really talks about the stamina, which is very important. 6-minute walk test supposedly tests a little bit stamina, but not really. It's also, you know, in the clinic. So it's really nice to kind of hear from the parents on that. So another patient, and I'm running over, but that's fine because I think this is very important. Just wanted to give you some baseline or some data on this patient, an SCN2A patient. So SCN2A is a voltage-gated sodium channel, and the mutation leads to hyperexcitability of neurons due to an increase of sodium flux. So when the patient started dosing, was 9 years old, had severe intractable epilepsy, that was uncontrolled by anticonvulsants, had to be on really high levels of phenytoin, cognitive language and adaptive language adaptive impairments, and nonverbal. So for this patient, actually, you will see that the protocol is slightly different. This is a 24-month study now, so 2-year study. And mainly this is due to the fact that if you want to track neurodevelopmental, cognitive, language, adaptive behavior endpoints, you will need more time than just 12 months. You know, it's very unrealistic to think you're going to change, you know, everything in 12 months. So this patient has received 3 doses of the drug to date. And so very preliminary data. I'm happy to share it with you, but I want all of us to think this is very preliminary data. Again, orienting you here, a number of seizures on the y-axis and months pre and post treatment on the x-axis. So the first 3 bars represent the 3 months before treatment. And you see, you don't need to know the number, you can see the height of the bars. And then treatment started at 20 milligrams and 40 milligrams. And you can see that the seizures were coming down from then. So we'll have to track that. You know, obviously there's other medications being given. And I think, you know, we always try to keep the other medications constant, but sometimes you also have to just take care of the patient and do what is, you know, needed at the time. So now moving on to safety quickly. Really, the bottom line is there's no ASO-related adverse event or serious adverse event that we've seen to date. The table below is showing you the number of doses that each of our patients in the clinic have received and what the highest dose level was. First 4 are intrathecal injections. The last one is our intravitreal patient who just got dosed, you know, a couple of months ago. One thing to note is the first 2 patients got 3 doses. They were very advanced when the study started and unfortunately progressed further, making it impossible for them to come back to the clinical site. So technically, you know, treatment has stopped for those patients, but the ASO had no side effects. But in those patients, we probably were a bit too late to be able to, you know, have an effect. One thing I just wanted to also mention is, you know, like we said before, the work that, you know, Stan did at Ionis really, you know, helped us in terms of all the decision-making that we're doing here in the clinic. There's a huge number of papers that they published, integrated safety databases that they published, from non-human primate toxicology studies to controlled clinical trials, both, you know, with drugs given systemically and then also with ASOs of the same chemical class and the ones that we're using, you know, when given intrathecally. So there's really a ton of information out there in terms of the, you know, potency, safety of these ASOs. And, you know, the papers that are listed here are obviously the nusinersen papers, the papers for the AD drug, Huntington's drug, SOD1 ALS. So, you know, there's really a wealth of information that we all have, and especially some of us, you know, came from Ionis, others didn't, but, you know, definitely the technology and the platform, you know, is not new, and we know the safety background. So lastly, before I finish, I just wanted to, you know, say 2 things. It's very important for us to share data. I mean, we wouldn't have organized this meeting here if we weren't about sharing data. And we think it's important to share with the medical and the patient community because, you know, knowledge that we gain when we give it out to you guys, that's how we will really make progress in the future. So we want to do this in presentation. We think it's very important to do this in peer-reviewed journals, case studies that will be presented and so forth. The way that we set up a REDCap system, and like I said, it's because, you know, some of the forms are very standardized. It's going to be easy for us to pool data across our programs and to be able to compare, you know, different data from the programs. Let's say, you know, do seizures, you know, are seizures affected similarly with different ASOs, you know, if the mutation is different? Like, we can easily look at that because our systems allow us for that. Integrating our clinical data sets with other sources is certainly always a discussion, but I think it's very important to realize we have to make sure that the data that would be combined is of quality and, has followed a rigorous process of collection. And I think for me, it's very important to also make sure that we protect our patient information and that, and the use of the data. And so we need to make sure that if data is pooled, everything is done in an ethical manner and everybody knows, especially the patients know how the data is used. So with that, I hope I have shown you that n-Lorem has successfully industrialized the development of optimized ASOs. We have a clinical infrastructure that is solid and that is delivering value to our patients. And we are ready to meet the needs of tomorrow's patients. We know we can do it. We're very secure about that. We appreciate the FDA's collaboration. We've successfully navigated the regulatory landscape across 4 divisions. We're ready to take on the other divisions. n-Lorem is sharing clinical data, and we want our investigators to publish and to present the work that we do, and we're treating patients. So we're very honored by all the families who've put their trust in our hands. And with that, I want to thank every institution that has submitted an AS or a patient, for us to consider, and also, thank the collaborators and pretty much thank everybody who's on the same page than us and who aims to advance the treatment of nano-rare. So thank you very much. And now, Sarah will bring it all together.
Thank you. Great, thank you so much. So they did a really great job. So it's a hard act to follow, but I am really pleased to be able to just talk to you about the how. So we've talked a little bit about the details of the ASO discovery and development. Really, we have a very robust clinical development. We're thrilled to have Lori have joined our team. And I think it's, really trying to think about how do we take this really beautiful mission that all of you are here for, that Stan has inspired us by this morning, thinking about the technology that's at the core of everything we do, with what Julie taught us, essentially. The process is very specific. We have very specific criteria that we follow. Every step is important. And ultimately rounding it all out with kind of the clinical development and really thinking about the regulatory process, the data, and just really wanting to think about the customization of every treatment protocol being so critical. And I think it's really, when I actually first met Stan, I met him with my parent hat on as a nano-rare mom, and then immediately threw that hat out the window and said, how are you doing this? I've been in drug development for a really long time. How are you going to do this? This is so cool. How can we help? And I want to talk to you a little bit more. How are we putting these things together? And actually, without having it planned, Chris so nicely earlier today talked about traditional and drug discovery and development. We know this takes a really long time. It's very, very expensive. And ultimately, what the nano-rare patients need is what we're aiming for. It's a year and a half. It's $1.3 million. We know exactly what this takes. And ultimately, this standard R&D, it's just not suitable for these patients. And we know there's very specific reasons why. So ultimately, standard drug development, you're thinking about a patient population. How do you figure out what are the endpoints? You have to have statistical significance. You're really thinking about this much broader regulatory path and really thinking about kind of the drivers are different, right? They're very different than what we have. Whereas for n-Lorem, we're thinking of one patient. We're thinking of your child. We're thinking of your loved one. And we're trying to understand what is the most important thing. And actually in the regulatory documents, they have some flexibility on what the treatment goal can be. So of course, we want this to be a validated and standardized assessment when we can. And we also like to incorporate very specific patient goals. So what is the goal? What's the goal for you as a family, for your loved one to be able to reach when they're starting treatment? And it really enables us to individualize the programs and to focus on the quality and the efficiency and each patient one at a time. So essentially there's kind of 2 core elements. It seems pretty obvious, right? I mean, it's the people and the process. And I think this is what every organization tries to do, is really putting the 2 together in a very seamless fashion. But we mentioned this morning, I think what's been so interesting, and we continue to encounter this with a lot of our partners who, of course, working in traditional R&D, it's a mindset. So we're not working in a linear fashion. We can't wait to have all of the information. We have to use our best judgment to have as much information as we can at a particular period of time to be able to move a program forward, because that's what the patients need. And so really what we've been able to do is the mindset. Every single person, you've met a few of us here today, but the larger team is still doing a lot of work for all of our loved ones today. Every single person handpicked. We hear, we've talked a lot about the importance of the 30 years of antisense experience, and we've brought to that another however many decades of the drug development experience, and the two of them just have to come together really efficiently. So I think, ultimately the way of working is very biotech-esque, if you will. So we have to all have our core accountabilities, but ultimately, what are you doing each day? It might differ, and we're going to do each other's jobs when we have to. We're going to get this IND done by the last day of the month, and you know what? It's going in regardless, and we're all going to make sure that happens. And those kind of goals, and it's really so fun, and it's really just really rewarding to be able to do that. And again, just every slide is the patient is able to be front of mind, and that's really a gift to be able to have that. And you've already seen this process and kind of where the portfolio is and where each program is. And in order to be able to do this is making sure that we have these quality systems and processes in place. And from a project management perspective, we have to be extremely succinct and concise. And I can tell you right now, if we want to know where every single program is, I know exactly where they are. It's very specific in our process. We know what's the next step. We know how long the next step will take. We know what the roadblocks are, the potential, kind of barriers to moving more efficiently. And that is so important as well. And so our team has really stood up a very precise, and I think that's a really critical piece to be able to manage this volume of programs and to feel confident that as we add 10, 20, however many more, we're able to continue that consistency and ensure that knowledge and understanding. So really what this becomes is this like beautiful, seamless, transparent interaction. It's our processes, it's our team, it's all of you, it's our partners, it's moving from one step to the next, it's, the application process, it's all of these things. It's kind of this nice sort of well-oiled machine. Not to say it's perfect, of course, right? We're not gonna make anything up, but we're really and truly in a place where we have this really nice interaction with all of these different elements. And that allows us to focus on the benefit risk decision, which I think Julie touched on it really nicely earlier. At each checkpoint is we need to think about the data, we need to think about the ASO progress and the data that's leading us towards an optimal ASO in the context of that patient. It's just this constant weighing of the benefit risk, and really then that transfers ultimately to the physician as well. So you already saw this I think it's just important to come back to what the process looks like and highlight the fact that, again, we don't have the luxury of doing anything in a linear fashion. And I think also thinking about how can we impart some of what we're learning back to traditional drug discovery and development. It's like, when do you have the ability to move things concurrently? When do you have the ability to say, you know what, we don't have all this information, but let's move at risk on this GLP tox study because we know we can't wait. And so ultimately that really allows us to focus on every ASO program as a unique program. The ASO strategy is unique. So Julie showed a number of different strategies that we're looking at for each different patient. And then that allows us to kind of flex and move in one direction or another for each program. I think this is really important to highlight. And on the next 3 slides, I was just going to walk through a few specific examples of, I was trying to actually think of a program where, it just went exactly as you might think. Like, let's just go straight from here to there. We have a patient, the physician applied, mutation is a drug target, and then we just went straight to IND, just straight, nothing on, no kind of diversion. And honestly, I think every single program has required that flexibility. And so that's a really interesting element. And I think just a few examples. So if we're first starting in kind of the ASO discovery phase, really thinking about what are some things that we've encountered already, is we need the patient cells to be expressing the target sufficiently. We know that we also need to stay on top of the data. As new data comes out, the ASO strategy may need to change. So it might be that we're going in with a non-allele selective approach, and there's new data that suggests perhaps we need an allele selective strategy. And so that's something that ultimately then you have to pivot, and we're gonna start that ASO discovery with a different lens and a different focus than for allele selective. I think it's also really important to highlight that, when we accept a patient into the program, it's very much that. What that means is now you have a customized, you have all of our team, you have a drug discovery and development program. That doesn't mean you have a treatment, right? And we're going to do everything we possibly can to be able to identify an optimal ASO for every patient. But we're not always going to be successful, and we fully acknowledge that. And so what we have seen is that there may be a path where the path that we were on doesn't end up coming up with an optimal ASO with all the appropriate characteristics. So we might uncover a different alternative, or we might say, you know what, we need to understand this functional consequence of this mutation a little bit more, and that necessitates additional research. So we've definitely had those things happen. I mean, I guess in my mind, I really visualize this as we have this laser focus. We got to go from target to IND as quickly and efficiently, high quality as possible. And we'll have research, we'll have this particular event happen, and then it'll come right back in, and we'll continue to move forward. In the GLP talks and sort of working with our intramural and extramural partners, we've had a couple of different, examples. So we've had one where we know that the GLP tox study is showing us findings that we're not comfortable. This is not an optimal ASO that would be safe for this patient, and that's important for us to impart, and ultimately then we need to either pursue a backup or we have to discontinue a program. I think interestingly, we also know that the IND, the FDA guidance document language, depending on the patient's severity, you're able to submit the document with different amounts of data. So we're very much on top of that, and that's a really critical piece. So some of the patients who are not progressing as rapidly, they're going to require a full tox package that will be 3 months plus 3 months of reporting. Whereas some of the more severe patients, they'll accept 2 weeks of in-life data and then submitting all the rest of the data when we have it. So I think a couple of other really interesting examples is, really having to ensure that we are getting as many ASOs to the CROs. So we need to work with our extramural partners. The scheduling slots, I didn't know anything about this, and I have so much respect for Julie and the team, and just it's juggling of trying to make sure we get the slots and then have something ready to go in that slot at that particular time. And then the last one here is really just thinking about the sterile fill. So Julie mentioned this with regards to 2 potential paths there. So there's the hospital, local hospital compounding, and then the sterile fill and finish. And ultimately, they're both viable paths, and the timelines are different. And so really trying to think about that in the context of each patient. And then the last few examples, really, and we have one more panel. So sorry, you guys have to deal with me one more time, but we have a really exceptional panel of physicians we'll talk about with this afternoon. And we are, now that we have more and more ASOs coming towards the patient treatment, it's becoming more apparent that the institutions, we need to support them better. We really need to think about some of these policy changes. How are we working to be able to move this more efficiently, and be able to make sure that if physicians move, or if an ASO is ready to go, ultimately we need to be able to get this ASO that's ready to go. And that's the most difficult part, is when we have an ASO ready to treat a patient and the inability to do so, sometimes because of institutional barriers. So, I think in conclusion on this section, and then we'll continue to move on to more collaborations, is just really the importance of the antisense and the drug discovery and development, the development experience. We're really focused on the systems, on quality, and on the flexibility, and really are successfully industrializing this process. And I think the really cool thing here is, even though our portfolio is very robust at this point in time, it's going to continue to grow. And because we feel confident that we have these data platforms set up, we'll have the ability to ensure that the knowledge is gained in a very efficient and effective way, and shared in a way that can be meaningful to the community as well. So these last couple of minutes, just wanted to talk, we had our partner, the partner panel this morning, and then also talking more about expanding to be able to identify and help more people with optimal ASOs. And so really what we're thinking about is, how can we try to export this model? And I think we heard from our Ionis partners and others, is how can we make sure all of this is published? But the challenge is, how do you actually put all of these pieces together to be able to ensure that individuals that could benefit from this technology and all the knowledge that's been gained can do so in a very effective way? And so the goal here really is to think about customizing a model evaluating where each partner is, what are the missions, how did they align, meeting them where they are. You heard, I think, 4 of our different collaborations that we spoke to. They're completely different, one from the other, and working with these different individuals and institutions and organizations in different ways. And again, focusing on the integration. So that's really the key piece, is moving from one step to the next. What are the decisions that are being made? Why did these ASOs move forward and these did not? And ultimately, all of these benefit risk decisions that are made constantly as we're working to identify that optimal ASO. And of course, as I will always say, is we would love to chat with any of you guys about interesting collaborations. So Lori mentioned kind of some of the partners in excellence. So we do have 9 that we've sort of from an agreement perspective, continuing to grow. We already touched a little bit on the St. Jude collaboration that Kristen shared, and we have her colleague Frank here as well. We also have, of course, the N of One Collaborative, so thank you. I think I saw Winston and Tim being here. So this is a professional society really focused on bringing together the community of experts in individualized treatments, and also a number of global collaborations. So Sick Kids Toronto, they weren't able to be here today, but that's a future collaboration that we are spending a lot of time really working to foster. Obviously, Genomics England, and there's many more other organizations. We also have Anna Mika, I think, here from the 1M in Europe, and really trying to think, again, if we have ASOs that are already treating patients in the US, we already have an approved research IND, we identify someone in a different country, like, what are we doing about that? We need to do something, because that's important, and it should be a relatively straightforward path. So the goal here is, again, we acknowledge we are a small organization. We're doing everything we possibly can with the capabilities that we have, with the funding that we have, and we need to continue to grow and to partner and grow and partner and grow. And of course, there will be many other ways that we can scale as well, but these partnerships are so critical. I think we already talked a little bit about data collection, and I'm out of time, so so I won't touch on this too much, but I think this is a really important element of what all of us are doing. And we have defined data collection and data sharing in these multiple different ways, and continue to, especially this meeting today, hopefully we've at least taught something to each of you, and would be very happy to hear more feedback on what else you'd like to learn from what we're doing as well. So I started this off by saying, how are we doing this? This is how. I think we've shared, and hopefully you can at least get a sense of, we do know how to do this. And the integration of the team, this fantastic group of experts that we've really pulled together, and having these ability to customize in real time for every single program. And that'll be an important focus that we'll maintain even as we grow and have more programs, that we still will focus every single patient's program. It's not that much different than a commercial drug program. It's going to have that much attention and rigor that's going to go into every single decision. And then ultimately collaborating is really going to help us to be able to help more patients together. So the last 2 here, I just wanted to make sure that everyone that wasn't here with us today is acknowledged. We just have this really fantastic team, and our lab is growing as well. And with that, I think we'll just say thank you. And I think the other 2 will come up for questions.
If you have a question, please raise your hand. We only have time for maybe 1 or 2 questions.
Thanks so much. Those are really great presentations. Could you, you mentioned that for each applicant, you do whole genome sequencing and you have biospecimens. Could you talk about how much of like the tox and other kinds of things are done on patient-specific cells as opposed to just general?
Yes, I can take this one. In general, when we need an allele-specific ASO, will require patient cells, just because we can't work with a commercial cell line, for example. So we really need to have the patient cells so that the ASO will recognize or bind to that specific SNP.
And in general, we have most of our programs are
Yeah.
allele selective, so
A lot of them.
yeah.
Yeah, and the sequencing too, I think you asked about sequencing. We do the long read for each allele selective approach.
Thank you. They were fabulous. And I think my question follows on from the previous one. So you talked about developing an ASO for an individual, specifically for that patient. If we're trying to look across, perhaps globally, to see are there other patients who could benefit from the same ASO, what would be needed for that patient? As in, if you're testing in an individual patient's cells, what sort of
Yeah,
what
so we
needs to
would
be
look
done
at the
to-
sequence of any potential additional patient to see, because as I explained, the ASO is not necessarily going to bind to the pathogenic mutation, can bind to another SNP. And so that is what we would be looking for in any additional patient sequencing data, whether they have this SNP as well or not.
Thank you.
We will take one more question.
Hello, great presentations. I was wondering, what's the typical amount of material that you use as far as grams go for a lifetime supply to the patient?
A single manufacturing batch will yield between 15 and 20 grams. And then depending on the doses that we're using in the patient and how often they're being administered, I think that would be enough material for probably 5 years, maybe more. There's a proportion of material also that needs to be set aside for stability testing and that sort of thing. So that's using some of the drug, but we're really trying to streamline that stability testing so that the majority of the material is going to the patient.
Thank you.
I think we'll break now. We'll take a 10-minute break and then resume at 2:40. There's been a lot of questions that have come in online, so we'll try to address those in the final Q&A. If not, we'll probably do a podcast in a couple of weeks and try to get all those questions answered. So thank you. All right, I think we're going to go ahead and get started. I know we're just like cramming information in. We had one day and we really want to get a lot of information out to the community. So let everybody kind of settle back down in their seats. I think from Lori and Julie and Sarah's presentation, it's vitally important for us to work with physicians through the entire process. So physicians submit the initial application. That's really important for us. And then we work hand in hand with them. And so I'm really excited to announce this panel. It's a physician panel. These are physicians that have been working with us for some time. I'm going to invite the panel and Sarah to come back up on stage. Maybe, where's Sarah? Sarah's in the room. Okay. Your moderator will be with you in a minute. Or I could step in, yes. While we're waiting on Sarah, do you guys want to go ahead and introduce yourselves so we're not like, kind of wasting time here.
Yeah, sorry.
Okay.
Yeah.
Sorry about that.
Liz, do you want to start?
Okay, I'm Liz Berry-Kravis. I'm a pediatric neurologist at Rush in Chicago. Have been in the field of translating disease-targeted medications to rare diseases now for about 20 years, and have moved into the N of 1 space in the last couple years. So very exciting.
My name is Sessions Cole. I'm a neonatologist from Washington University in St. Louis. And I am one of the participants in the Undiagnosed Diseases Network's Therapeutic Matching Committee, where we try to leverage the mechanistic characterization of diagnosed UDN participants to be able to identify and prioritize therapeutic strategies.
My name is Olivia Kim McManus. I'm a associate clinical professor of neurosciences at UCSD and a pediatric neurologist, epileptologist. So I specialize in treating kids with intractable epilepsy, and that's often due to rare genetic etiologies. And so I've been working with n-Lorem and also treat kids with Batten's disease for neurodegenerative enzyme replacement.
Hi, good afternoon. I'm Neil Schneider. I'm a neurologist, neuroscientist at Columbia University. I have a laboratory that focuses on the study of mechanisms of neurodegeneration in ALS, and spent a lot of time working on a juvenile form of the disease caused by mutations in a gene called FUS, and that led me to develop an antisense therapeutic with Ionis for that particular form of the disease, and subsequently that led to, as we spoke about earlier today, a partnership with n-Lorem called Silence ALS, focused on nano-rare forms of the disease.
Okay, well, I'm really sorry. I was standing in there, so I don't know why I wasn't aware of the time. So my sincere apologies for that. But thank you all for being here. We're just really, really thrilled to have each of you. So I guess it would be great to start off with kind of hearing from you about your sort of journey to working with n-Lorem, and sort of what do you feel are the important things that we bring and that we need to make sure that we maintain in our relationship with working with you. And Sesh, if we might start with you.
Yeah, thanks, Sarah. Well, first, I'd like to thank Stan for his vision and leadership. And I'd also like to acknowledge and thank both Dr. Stan and Dr. Roseanne Crook for putting the wind in the sails of the n-Lorem initiative. So I think that's been terrific for all of us and especially for our patients. So as a baby doctor, I get to take care of children and families who don't get to choose me. They have to sort of accept that I'm on call that night. And consequently, I have to earn their trust. And that earning process has, for me, really informed how I then manage and help them manage their hope. Because they then rely on me, as do the UDN participants who get diagnosed rely on those of us who participate in their diagnostic odyssey. They rely on me and us to help them hope. And so in order for me to respect that hope, I need to be able to trust the processes, opportunities that I refer the patients to. And n-Lorem is, for me, the sort of benchmark of trust. n-Lorem has taken extraordinary measures to be sure that when a patient is referred, that patient receives absolutely transparent, rigorous, and informed by experience consideration. And I know that if the answer is yes, n-Lorem will take that patient, that patient will get extraordinary care and treatment from n-Lorem. But I also know that if n-Lorem decides that for whatever reason, because of the biology of the variant, because of the progression of the disease, or for other reasons, that patient is not a candidate for an ASO developed by n-Lorem, that I can trust that opinion and work with that patient and family to move on to the next source of hope. So I think trust for me is a big deal for n-Lorem.
Yeah. Thank you, Sesh. Liz, what about you?
So I want to thank Stan also for developing this program and all the people who work for the program, because we've had great relationships with them. But I think when I went into neurology a long time ago, I went into neurology because I felt like it was a completely open field where we didn't have really any treatments and any treatments targeted to the diseases that we actually treat. And I thought, well, there's going to be research and I'm going to be there for the happening of those treatments. So I thought that was going to happen about 20 years ago, but now after 30 years, it's actually happening. So this is extremely exciting. And over the years, I've kind of gotten to be known as the rare genetic disease doctor of neurology patients in Chicago. And so I get self-referred patients, and so my patients do often choose me, but unlike Sesh, but you know, doctors choose to send the patients to me. And I look at this and okay, there's a gene mutation and it's in a gene that I've never seen a patient before, because that's getting really common these days. And I immediately go and dig into the internet and papers and everywhere and try to find out, what does the protein do? what does the gene do? What's the mutation doing to the gene? And then I meet with the family and try to figure out how big of an impact is this disease having on the family. And then I ask some questions. Are there trials for this disease? Often not, because there's only 50 patients in the world. Or is there a foundation? Sometimes there's a small foundation that a family has created online. What work is going on the disease? And then I ask, is this a mutation that could be targeted by an ASO? Because that really has been like one of my frontline questions when I see a patient in the past couple of years. And then one can go through a process of thinking through the types of mutations and even submitting to n-Lorem, which I have also done. And then even in the back of my mind, if I'm thinking, ooh, this is an intronic mutation, maybe it would be targetable by an ASO. I'm thinking, what is the disease concept model for this disease? What are the things that are impacting the family most about this disease? What would I want to try to improve on, and how would I measure that? And I think those are the things that go through your head as you see these patients. And the fact that we even have the opportunity to do that is frankly amazing. So really every rare disease patient I'm looking at, I'm thinking, what could we actually do to treat this person's underlying biology? And I think by doing that, we have the opportunity in this generation to understand basic concepts like what can we actually reverse if we treat patients after birth, when they have neurological diseases. How much havoc has already been wreaked on the brain by the time you get to the patient at 5, at 6, at 30. Those are concepts that we need to learn, and we're only going to learn that by collecting all the information, sharing the information, working together, and then that will circle back and help us understand the clinical responses to these drugs is going to help us understand the disease mechanisms somewhat, even the basic science disease mechanism somewhat better. And it's going to help us understand how to do the whole process better and what measures can work to measure changes in what patients. And just being a part of that process is extremely exciting. And n-Lorem has certainly facilitated that.
That's awesome. Thanks, Liz. Olivia, what about you?
I mean, for me, as my perspective from a pediatric epileptologist, I mean, you can see the amazing families that were up here this morning. That's very representative of my clinic. It's different variants, but often manifesting with similar symptoms which are very intractable and severe at onset. And so the fact that there is even a potential for a targeted treatment, this is very important because it's not just about, seizures and brain, but it affects the whole body in a genetic mutation. And so the opportunity to even provide patients with a targeted therapy, this is so important to even put out there that it's a possibility. And then that will also, continue to drive earlier genetic diagnosis and interventions. This is very important and will change, I think, landscape of neurologic potential, as Liz mentioned already. And so this is why I'm working with n-Lorem and the amazing team that they have.
That's awesome. Neil, what about yourself?
Well, I think my relationship to n-Lorem is a little bit different than a lot of panelists and other doctors that work with this wonderful group. I'm an ALS guy. I don't work on many diseases. I work on a disease. ALS is a devastating disorder still. And despite a lot of great work from a lot of people around the world over 80 plus years since Lou Gehrig had this disease, we still have very little to offer our patients, except those rare patients who have genetic mutations, whom I think we can now offer a gene-based therapy that is attacking the most proximal mechanisms in the development and genesis of the disease. I said I came to this out of my experience with FUS ALS, and I can say I've been doing this, for 25 years. It's the single most satisfying thing I've done is the work I've done with JC and Anna, Ionis and n-Lorem. I just see the potential to do more of this for more patients with even more rare mutations than the FUS mutations. And n-Lorem provides, for the reasons you've heard, today, a platform to do it. Wonderful people that you've seen on stage, that are trying to enact Stan's vision for this. You heard the words hope, rigor. and dedication. This is what we offer patients with, I think, a potential to help lots of people. And even though I'm a single disease guy, I think that the things that we're doing through Silence ALS and for ALS patients and families have large implications for other diseases as we hone our processes and our approach to these cases.
Awesome. Thanks, Neil. So I just wanted to have a couple of questions about kind of the patient identification and sort of movement into n-Lorem, determining whether we can help these patients or not. And Sesh, maybe we could start with you. I know you spoke about your leadership in the UDN and what an important element that is to potentially be able to then the therapeutic matching committee that you lead. And I was just wondering if you could share a little bit with this group is about the considerations from even a technical or a functional genetics perspective that you're putting in your mind, and also as a member of the ATTC. So when you're thinking about these recommendations, this is such an important decision, and it's the gravity of the decision we fully acknowledge is significant for different patients. I wonder if you could speak a little bit to that.
Yes, well, the Undiagnosed Diseases Network does focus on genomic diagnosis, but then functional confirmation and mechanistic confirmation of what is encoded by the specific variant that the patient carries. And so the UDN participants have an unusual leverage in that we understand something about how the misspelling in the gene disrupts the patient's body. And so that knowledge is critical as we think about who would be best served by which kind of therapeutic intervention. And having that kind of understanding really helps the Undiagnosed Diseases Network cooperate in a very open and transparent way with n-Lorem. In addition, the UDN does very deep clinical phenotyping. So we know what current status of each UDN participant is from a biomarker standpoint, from an imaging standpoint, from an operational standpoint for activities of daily living. And so we can share with n-Lorem what at least our opinion is about what amount of that phenotype is reversible and what amount of that phenotype is not going to be reversible. And that's so important in informing the thoughts about who is really going to benefit most from an ASO. It also is critically important to share with family members or the patient to be sure that they all understand that given this phenotype, this is at least one, this is what we can expect might be reversed by the ASO treatment.
Yeah, and that's a really, I think the point about the depth of the understanding that the UDN gains on these mutations is really important, because not all patients are being diagnosed through UDN, of course. And we know many of the patients that actually apply to n-Lorem may have a mutation that has some knowledge around the functional consequence, but if it's not sufficient information and sufficient research on that consequence, our team, we are not able to identify and feel confident in an ASO strategy. And that's definitely a component, that sort of proof of concept element, is how can we support even from the diagnosis to really and truly having an understanding of the functional consequence, so that then our team can get to work on what the ASO strategy might be. Olivia, I wonder if we could turn to you and just, love to get your perspective as kind of you continue to grow your practice and at Rady, and thinking about the patients that you would nominate for treatment with n-Lorem. From a clinical perspective, what are some of the things that you're thinking about or that you're looking for, that, even building on what Liz said, is how you're thinking at the very beginning about how you would be able to assess the benefit of a treatment or not?
I mean, of course, the basis is going to be the genetic testing is going to be the main thing first. And then in terms of clinical symptoms, what is most bothersome to the patient? Also, what is the disease-specific clinical symptoms that we can potentially follow along a treatment regimen or a clinical trial and assess for change or interventional effect
Mhm.
is very important to really streamline, because everybody is different. Every variant is, you know, as much variability as there is with genetic variants, there is with phenotype as well. And so it really has to be tailored to each individual. And also, it's not an easy process to even consider doing an ASO trial in your child. It's a big decision as well. And so that comes into play. And so those are some of the main features that really have to be addressed.
Yeah, thank you. That's very helpful. So I wanted to, in the next couple of questions, talk to Liz and Neil. So both of you have been treating patients with the ASOs, and just speaking to your experience, you know, in Liz, really thinking again, wanted to come back to the fact that you are the expert here on many of these outcomes, and I think this closet concept, that's so cool. We continue to hear about that, and from a neurodevelopmental perspective, and just really thinking about, from your perspective, what are some of the key kind of challenges that you have had, or things that you've been thinking about in the context of continuing to make your treatment decision, as far as understanding the benefit risk and whether this ASO seems to be helping a patient?
Well, you know, there's a whole list of things that goes into consideration. Obviously, you know, we can diagnose a patient with a super rare disease that's a degenerative disorder, but if they really have limited function left, then there may be no way for us to actually stop the disease, even if we had something that worked. So that's a consideration. A consideration is whether the family is ready to take a jump into experimental treatments. And, you know, then the consideration is, do they have something that we can actually target? And in terms of trying to get the whole thing together, if we decide we can treat a patient, there's really, these patients come from all over, and they often have clinical information here and there. And so you really have to try to gather. We've been fortunate with the patient that we've treated. We got all of his information from Boston very nicely. We've been able to coordinate with a neurologist in Florida for things like EEGs. We do have to coordinate our institution, actually. And we have a process that we developed for that, actually, through our Angelman syndrome work, where we, you know, even when we admit patients who are getting ASOs after their first doses overnight, we have a whole system with the nursing services there. And, you know, you have multiple professionals that do things that just kind of do work on our trials, like nerve conductions and MRI. And so there are really many things to consider and coordinate in an individual hospital. It is quite an undertaking, but worth it.
Worth it. Yeah, and Neil, I mean, that's exactly, I feel like, a lot of your experience as well. I mean, of course, patients come from all over to see you at Columbia, and I wonder if you could just comment, you know, a little bit on how you envision that working from a coordinated care perspective and data collection, and even patients who are at other sites outside of New York City, perhaps, or even outside of the US, but still want to get this treatment and perhaps have the dose administered by you.
Yeah, so again, a lot of what we're doing with n-Lorem for silent ALS is informed by or based on our experience with the FUS ASO, where, you know, which began as an N of 1, and then 2, and then 5, and then 10. And, you know, I think this is going to be the experience of a lot of people, and then people come to you from, you know, many parts of the country and different countries, and you have to, you know, figure out a way of adapting your process, you know, to accommodate these people. And this is what we're, you know, doing and going to have to do more of with n-Lorem. You know, I think that it's hard to standardize, because institutions are so different, and you know, local specifics are different, but we're trying through silence, and I think n-Lorem more generally to standardize these things in a way that would make it, you know, easier to involve other patients, other genes, other, you know, mutations, and I think that's coming. You know, and I just also, I think, wanted to say in terms of processes that, you know, and Stan said it this morning, that these are human experiments, right? So you're dealing with a lot of uncertainty, and, you know, you can be as careful as you want to be with, you know, in advance of delivering an ASO, with phenotyping and trying to predict phenotypes and endpoints, and, you know, but you have to be prepared to accept the unexpected.
Stan presented the case of Anna today. Anna bottomed out. We thought that she was dying, and then began this absolutely remarkable, unprecedented functional recovery. A time when people had given up on her, she hadn't given up, and her family hadn't given up. So we're learning what success looks like in these diseases where there's never been success. We're learning to how to deal with patients at the time of nomination until through the period of ASO development and delivery, and then through the treatment period. So these are very early days, I think, and I just think we have a lot to learn.
We do have a lot to learn. And, I think so even the standardization, I mean, the red cap that Lori spoke about earlier, in theory, the idea would be that the same red cap CRFs could be leveraged for any physician. So for example, if Liz's patient is being seen in Florida and, all these other places, then anybody who sees that physician, the idea would potentially be to standardize in that way. But there are many, many other ways, and the standardization is so critical. So we only have a couple minutes left, and I was going to ask a question about funding and then a question about challenges, but I imagine those two things might go together. So I wonder if I could just turn to each of you and sort of in the way that we're going to be working together in the future. Obviously we have a number of ideas for strategic solutions to help you all, to help the physicians that are in the diagnosis and in the treatment space, and institutions that we want to try to work on together. And Stan will talk about some of those others later today. But I wonder if each of you could kind of just comment a little bit on kind of what are the key challenges that you hope that we can tackle together here in the short term. Now, Liz, we'll start with you.
Okay, so the institutions are not a small barrier. You basically have to really push your institution into doing this because they don't want to, because they think it's going to cost them money. So if you can convince them it's not going to cost them money, then sometimes you can go forward. And that's basically what I've had to do. And then, so I've had a few kind of novel solutions to some of it. We've recently actually created an N of 1 fund in philanthropy to work on finding donors that want to give money to Rush to potentially support this exciting treatment of individual patients with personalized medicine. So that's one of the solutions we've had to the institutional issues. I think one of our big challenges in the field, of course, because I'm in neurology, is outcome measures, which is a challenge for everybody in neurology. And I think that I was the originator of the closet idea. And I think what we need is to add all this work together, share it among other groups like N1C and like Combined Brain, which wants to do outcome measure standardization as well. And, get outcome measures for each type of thing like cognitive or motor function, and for each severity, because you would use different measures for someone mild, moderate, or severe, validate those, get those, sort of that you have them on the shelf, and people can come in, well, I have a patient like this, and so these are things that I could potentially consider using.
Yeah, that's great. And Liz has been I think you've been a great mentor also for other physicians, have often learned a lot from your experience. Sesh, what about you?
So not being a neurologist, because I wasn't smart enough. I wasn't smart enough to be a neurologist. I take care of babies, and the biggest problem for babies is breathing. And misspelling in various rare misspellings in various genes disrupt the lung transition from womb to world. And some of these rare misspellings result in irreversible and progressive lung failure, which currently is only addressable with lung transplantation. So we transplant half dozen babies a year with these genetic misspellings for whom nothing else is available. And I am hopeful, thanks to Stan's support, that we are going to make some progress on ASO development for helping these babies to breathe without transplanting their lungs.
That's amazing. Yeah, thank you for that. Olivia?
I think you know, in terms of challenges, there's very many bringing an ASO forward in an institution. I mean, funding, regulatory, lots of different aspects that need to move in parallel to really deliver it at the same time. And that's why the funding mechanisms, like Liz said, with philanthropy is very important, outside of grants and things like that as well. And then the institutional buy-in, I mean, it's a different model from regular clinical trials, even if it was for rare disease clinical trials. And so that takes a lot of effort in explaining that, and then changing the regulatory model and even IRB processes. And so that takes time. I think it's going to be an issue across all institutions, and that's why it's important to partner with other groups like N1C or n-Lorem and bringing these things together so that we reduce redundancy and effort and just kind of streamline things for everybody to move things forward.
Well, the first time will always be the hardest, right? So it will never be this hard again for you. Let's hope, right? Neil, how about you?
Well, I mean, I think many of the challenges have been mentioned. I would say one of the greatest areas of challenge is going to come from our success. That as these things start working, I think we're going to be seeing more and more cases. Dealing with that volume is going to be tricky. And in the case of neurodegenerative diseases, and this may be a little bit different from some of the developmental disorders, you know, we have the ability to identify and track individuals in advance of symptom onset. And I think that we will have challenges related to the implementation of these therapeutics earlier in patients' course, and ideally even before disease onset, and dealing with the regulatory and logistical challenges of that, I think is a happy challenge.
Yeah. Well, that's a great way to end the conversation. And just thank you so much. I'd like to thank all our panelists for being here today. Appreciate you guys. Well, I'm really happy to introduce the next speaker, me. I'm just going to take just a few minutes to, and I'm going to sit down because there's a chair here, to kind of talk about our program, our Empowering Nano-rare Patient Program. And I think I can use the clicker here. Okay. When I started thinking about, you know, what I want to say today, I pulled out our guiding principles, and I thought this might be really important to share with the group, just so that you really understand that patients are at the heart of everything that we do. If you haven't already figured that out today, through the presentations that we've given, we really know that sick people depend on us. Everybody that comes in to work has that in the back of their mind. We know that a lot of our patients don't have a lot of time. We have to juggle, you know, that with making sure that we're doing the very best that we can for our patients, and developing and discovering and providing these optimized, personalized ASO medicines. And so patients really are embedded in our guiding principles and drive all of our decision making. The goals of our patient empowerment program are really lofty. We wanted to create a community for Nano-rare, and so this is a big step in that today, and I'm very appreciative of the efforts of everybody that has come together today, that has agreed to fly. We've had people fly from the UK over here just for this meeting, and so this community is really important to us, and I know it's really important to our Nano-rare patients. As Stan said, we really think it's important to honor the bravery of the patients who are going first and accepting those treatments and taking that big step. And so we really want to honor those patients. We want to give patients a voice. You know, I think Nano-rare patients, they don't have a disease name. You know, they can't identify with, you know, a disease that people understand. And I can't imagine a parent saying, well, you know, I have my child has this disease, and well, it doesn't have a name, but we're going to call it by this gene, but it's not going to make any sense to you because no one understands what these gene names are. And so really wanted to give a patients a voice. And so we've done that through many various ways, and also today in this meeting. And we want to just give people knowledge, you know, so today is all about conveying knowledge to you, so you can take that knowledge home and you can make informed decisions about
Do I want to work with n-Lorem? You know, what does that look like? What if my physician submits an application? What should I expect to come next? You know, and so what that looks like, and just trying to give you as much knowledge as we can. And then obviously we want to share our learnings. We're sharing today. We share through publications, peer-reviewed journals, all kinds of avenues to share. And we're going to advocate. We're going to advocate on your behalf. We're going to advocate as a community, right? We do this in many different ways, and I'm just going to skip through this slide really quickly because I'm going to go through each of these individually. Obviously, today we're here at the Colloquium. And this is a big step for us. This is our first. And so I hope that everybody has had a great day today. I know we had a lot of fun putting this on. And as the numbers kept creeping up in terms of registration, we were like, wow, this is just amazing. And so just very happy with the you know, the way that this day has gone. So just really appreciate everybody. Stan mentioned our podcast. This is a shameless plug for it as well. We started this a little over a year ago. We released 2 podcasts a month, and you know, I don't think any of us really understood like the work that goes into podcasting, but it's a lot. And I think we thought maybe by the end of this year we'd have about 4,000 downloads, and we've been happy with that. We've doubled that. I think we might hit 10,000 before the end of the year. We've had our partners come in and support and sponsor our podcast. So we're actually you know, getting money coming in to help us offset the production of those podcasts. But really the main point is just to provide more knowledge, more information. We've had a lot of Q&As that have come in, well, Q's, no A's, but we've had a lot of questions that have come in today that we're probably not going to be able to get to. We are going to do a podcast in the next couple of weeks to address those questions. And so I would say if you haven't subscribed to our podcast yet, please do. You can find it anywhere where you can stream your podcasts. I sound like an announcer here. Social media. So this year we were like, we're going to really be strategic in social media outreach, and we have really done a lot. And I think a lot of people have kind of found us through social media. And so I'm also really not on social media that much, but I'm told that these numbers are good too. So yeah, so I would say if you want to help, you want to spread the word, you know, share, comment, you know, subscribe to our social media channels and engage with us that way. Our website was an 8-month project. It launched, our new website launched in March. And one of the things we really wanted to do with that website is to allow patients who wanted to, who we knew, because we're blinded to patients, to share their story. And so we have, I think, 14 patient stories on our website. We'd love to add more. We just, you know, want to recognize the bravery as well as the journey and how difficult it is, and just allow people to understand that these are real people, right? This isn't just a corporate website that we've slapped some stuff on, you know. We wanted to have our website create a community, and we want to share your story. So if you want to be part of the n-Lorem website, we'd love to have you. I'm just going to kind of skip through these a little bit. These are some of the things, oops, went too far. These are some of the things that we also wanted to do with our website, is just provide a lot of information. So today we kind of went in a little bit more depth, but we have our pathway to treatment. So what all those steps are, define kind of a little bit more information about those steps. And so I would commend you, if you're an institution and you're like, I wonder what it's going to really take for me to work with n-Lorem. You have a pathway on the website. It'll give you a lot of information. Potential physicians out there, we love to have you. There's information for you on the website. And then obviously the team here is more than happy to talk with anybody that wants to work with us. Our patients need you. And we are really, as a nonprofit, we don't have a lot of resources to be able to kind of go everywhere we want to go, but we really try. And so we've done scientific and therapeutic focused conferences, patient advocacy, patient meetings, you know, everywhere that we can go. We're always going to ask you, can we go for free? Can you register us? Can we have a booth for free? So yeah. But we love to talk to people, and we're always here to share what we're learning with the community. And we have publications. We have a lot of publications that we've pushed in high quality journals, and a lot of these kind of talk about what Stan said today, you know, the learnings that we're having now. We're going to be able to start publishing those papers. You know, we have published our processes and our methods, and want to share with our community. And I really want to stress this, recognizing our pioneer and patients. So in our offices, we have a patient wall that has all the pictures of the patients that we know that have allowed us to, and we have that there so we can remind people when they come in, like, these are the real people that we're here working for. And we actually had a patient family come in, and they were like kind of, you know, hanging out, and they turned around and they had their child, and he saw his picture on the wall, and he was just like enamored with it. And so we actually got to capture that. So, you know, I think, you know, just these families are so brave, and they're really the heroes of the day. And just want to recognize that. And I hope we're doing well. We'd love to have feedback on sort of the things that we're doing and what we should be doing, what we aren't doing. We'd love to hear from the community, and because we want to grow and we want to provide the Nano-rare community with what they need. And we need your help to do more. So you might be asking, like, what can I do? You could become a sponsor. I'm thinking there's a 2024 Nano-rare patient colloquium in our future. I don't know if I'll survive it or not. We have podcasts that, you know, like I said, we do podcast sponsorships. Social media is great. Donate $1, $1,000, $1 million, you know, we'll take it all. If your employee has an employee giving program, those are great. And we're also very happy to come and talk to your employees about n-Lorem, so they know, you know, a little bit about us, and then just spreading the word in the community. And I also want to thank you for everything that you're doing for us. And I want to introduce our last panel of the day, and we're kind of circling back to all these learnings that we're getting from the community, the incremental data that we're coming in, and how really these learnings that we're getting from our Nano-rare patients can really change the way we're thinking about disease. And I'll introduce Andy Marshall, who's going to chair this panel. He used to be the head of Nature Biotech and now is venture partner at Scion Life Sciences.
Can you all hear me? Is it on? Yeah. So bear with us. This is the last panel of the day. We've got like 40 minutes, and I would just kind of, from a personal view, I've just found this meeting truly inspiring as well as informative, I think. But I really had no idea how much progress n-Lorem had made in such a short period of time. And I was just listening to some of the previous presentations that the n-Lorem team was giving. And I mean, just to give you some context, in terms of INDs, 9 INDs, is that right, Stan? And so I quickly went to the Pfizer 10K and I looked at their 10K. So Pfizer has like 83,000 employees, okay? So they have somewhere over 30 INDs, but that's essentially 3 times what you've done. And this is not only Stan, this is the constellation of people around Roseanne and Frank and everybody else who works with Stan and Amy and the rest of the team. This is an incredible achievement in the amount of time that we've seen. And I just think it's like truly inspiring to see, you know, how much progress has been made. But I wanted to just give you that kind of context for what we've seen. So in this final panel, well, 40 minutes or so, and I hope we'll have some time for some comments at the end. We wanted to explore how we think about expanding what's going on at n-Lorem into the research community, how do we amplify what's going on here, and how do we engage the research community with what n-Lorem is doing? And so we've got like 4 panelists for you here. I'm not actually going to introduce them myself because I want them to talk about what they're doing. But just, you know, we have chief editor of one of the premier journals. We have 2 researchers who are either at the clinical end and at the kind of basic and clinical interface. We also have an example of how somebody who is who at some point in time, Jeff, is going to have to think about how this is going to affect you. And we also have John here, who's from one of the premier foundations that's in autism and neuroscience. So, Jeff, do you want to kind of kick things off and talk a little bit about your interaction with n-Lorem?
Sure. Thanks, everybody. Hi. Yeah, my name is Jeff Carroll. I think maybe the first way to introduce myself to this audience is to say that, as Sarah mentioned earlier, as well as being a professional in this field, I'm also a family member. So myself and 3 of my siblings carry the mutation, which will cause inevitably Huntington's disease, a really terrible neurodegenerative disorder. Although every time I come to this meeting, I'm reminded that as terrible as HD is, seeing all these sick kids reminds me that, you know, there's worse things in the world for now, until we get them all fixed. So I'm a rare disease family member. I'm a scientist. I worked with Frank Bennett for many years on the Huntington's disease program, which is really the great hope of the Huntington's disease community. You know, we're deep into clinical studies for a Huntington ASO that I think is the best hope that everybody in the HD community has ever had. And it's in clinical trials right now, and I'm very excited about it. And when that went to the clinic, I sort of thought, well, you know, what do I do now with, you know, my exposure to ASOs? And I run a mouse lab and I know some genetics. And I met Frank after he had just, and with Stan, kicked off this amazing organization. And Frank said, hey, you know, a little bit about ASOs and a little bit about genetics and some basic science. You seem like the perfect person to rope into helping vet cases. And so I started doing that. So all those 100 and whatever cases, many of those are work that I've done, and now with lots of help from other people because we're way past Jeff's capacity. So I help vet the cases. And so I'm kind of on that early part of the funnel, and it's just been incredibly fun, rewarding, interesting in a weird scientific way, but also motivated by the fact that each of these is just a really sick family. And so just incredibly motivating and fun. And it's been a great honor to be playing that role since the beginning of n-Lorem.
Great. John.
Hi, everybody. I'm John Spiro. I'm a neuroscientist and I work at the Simons Foundation in New York. It's a private nonprofit founded by Jim and Marilyn Simons. Jim was a very influential mathematician turned investor. And instead of buying like a sports team or something, he decided to start a foundation to give away money to advance the frontiers of science. And there's been a special focus of one part of the foundation that I'm involved in called Safari on autism research. And we made a decision about 17 years ago to take a really big bet on genetics. So this was in the early days of autism research where really, other than things like Fragile X or Rett, there really wasn't much understood about the genetic architecture of autism. But so we spent a lot of time putting together large cohorts of individuals with autism, and then at the time doing deep exome sequencing. You know, early on that was a big deal. Now this is becoming routine, and so and when I say we were funding researchers all over the world to do this research. And we basically uncovered that there's this enormous, you know, autism, which is often considered a sort of common disorder. I think one way to look at autism, especially on the sort of more profound serious sides, is really a collection of rare disorders. And so we've upped the ante, so to speak, in the last few years and created a cohort called SPARK that aims to be the largest effort to collect families all over the United States. They either participate remotely online or through a series of clinical centers that we have relationships with. And we're trying to do phenotyping of these families, but also doing very
significant genomics research that we've sequenced something on the order of 50,000 individuals with autism, something like 150,000 in this cohort that haven't been sequenced, but that are sort of involved in the research. And the idea is to sort of create this research-ready cohort. There's a lot to learn. I think there's an enormous amount that interfaces with what n-Lorem is doing. There's a lot more to say. I might stop there, but just want to say one more thing is, in addition to creating these cohorts, Safari has put an enormous amount of effort into sort of doing the behind the scenes analysis of what these genes are doing, where they're expressed in the brain, making iPS cells and things like that. And one thing, and so we've spent a lot of money on animal models, and I still think there's an enormous benefit for animal models, what we can learn. But I think something that I've come to believe from watching n-Lorem and some other players in this space is the enormous importance, when it's safe to do so, really important caveat, the enormous importance of moving drugs into humans when it's possible, because I think we're going to sort of hit a wall with what we can learn at some point. I think other people have made that point today, but anyway, I just want to, maybe I'll stop there.
Great. So Vijay, you're kind of at the cutting edge of clinical research, kind of at the Broad, and so can you talk a little bit about your research and kind of what you're seeing and how that interfaces?
My name is Vijay Ganesh. I'm a neurologist at the Brigham and Women's Hospital, where I co-direct the neuromuscular genetics clinic. So patients either with a diagnosis or looking to obtain a diagnosis from the community in neuromuscular diseases. I run that with my division chief, Tony Amato. On the research side, I'm at the Broad Institute functioning in really two arenas. One is a project called the Rare Genomes Project, where this is a patient-initiated research study where patients apply to us who have a suspicion from their clinical team of having a rare disease, a single gene disorder, and we apply research-based advanced genomic methods, whole genome sequencing, and a slew of other methods that I can get into to get a diagnosis for that individual. So this is essentially a research study where both we're hopefully applying a service to these patients, simultaneously academically advancing what genomic methods need to do to improve the diagnostic yield across all rare disease patients. In addition to that effort, my role on the research side is to do this kind of analysis specific to neuromuscular disorders, where it's a particularly difficult problem of improving our diagnostic yield for these patients. There's many reasons for it. These are very large genes that can tolerate a lot of variation in the non-coding, the kind of silent part of the genome, where we know that causal variants exist, but how to bring our advanced methods to prove that, to put it on a platter basically for n-Lorem to say, okay, this is the cause. That is all kind of, I think, where on the academic side we can really interface with efforts such as n-Lorem to basically both build the best practices on the research side and also deliver to these patients variants with the evidence from their cells. And then have a best practices approach that these methods can be applied more broadly and expedite that process. We know it takes time, and time is essential. So broadly speaking, on how our group at the Broad, the Rare Genomes Project and some of these other consortium efforts are run by Anne O'Donnell-Luria and Heidi Ream, my mentors. There's three parts to it that I think we all see as being essential, but somewhat separate parts to it. One is the genomics methods development, applying those tools. The second piece of it is redefining these disorders. A lot of these patients are telling us that our conventional ways of making these diagnoses are increasingly antiquated and might complicate matters. So that it takes a physician effort and translating it to the genetics to sort of even, frankly, rename these syndromes. I mean, as Neil's an expert in ALS, we still diagnose ALS, or the essential part of establishing the diagnosis is hitting someone with a stick, at their reflexes, and then sort of determining if that's abnormal or not. And we can, in what I see often are neuromuscular, muscle diseases, it's defined by what people see under a microscope, but we know that many genes can cause a similar appearance. So on the research side, it's really to spearhead that effort, connect the genetics and what we know about it to redefine these syndromes and establish a more rigor about how we establish these rare diagnoses, to unify a lot of patients that are disparate, labeled with different things. And I think it'll help a lot of efforts that we can do on the academic side to help the patients going forward. And then the third piece that our group does, particularly Heidi Ream and her impressive efforts to cause improved data sharing globally, is link up patients with sort of open data sharing. Nano-rare is one thing, but it really helps to identify, both for the patients, that second individual in the world with the same disorder. It helps form a human connection for these patients. I've seen this in my own rare disease discovery work, but also from this scientific side, what are the overlaps for those patients? How do we redefine that syndrome so that now that we can see it's a shared entity? That requires a global effort and requires a lot of things that I think can only be spearheaded at the academic side. So those are the things we're sort of involved in.
Great. So Bob, you are, so you're kind of even earlier than everybody else here. You're kind of seeing what's coming through basic research in terms of technologies. So if we kind of have exome sequencing as the kind of clinical standard to an extent, so what kind of technologies are coming through the pipeline at the moment?
Do you want me to introduce myself first?
Yes, go ahead.
Okay.
Yeah.
You might know who I am, the chief editor of Nature Biotechnology. So I have taken over from Andy, which is why he knows me very well. As apart from these guys, I left the lab about 10 years ago to become an editor, and have been at Nature Biotech and leading it the last year. And so I obviously publish your research, your maybe not the clinical trials, but more of the basic science research that goes behind the clinical trials, earlier stages. So in some ways, I'm seeing the earlier stages of the research in that way. But yeah, also I'm sometimes late to the game because you guys are all doing the research, ahead of submitting your paper. But also, I have the pleasure of, I worked with Stan and Kelly on this amazing comment that they wrote earlier this summer on the, promoting n-Lorem and what they're doing here. And, just speaking about ultra rare diseases in general, what we can learn and what needs to change in the system. So, as a journal editor, I get to do, both the research side and also some of that commentary and bring some of that discussion and community to this field. I can get back to your point about technologies now. So in terms of technologies that we're seeing, I think there's a lot to be said here. I've listened to all of these talks today, and there are a lot of limitations that we're working through, whether it's in terms of diagnostics, identifying these mutations earlier, whether that's prenatal testing or moving from whole exome sequencing to whole genome sequencing and isoforms, which are, differently spliced proteins that can contribute to disease, and putting those in more context of what is actually functionally happening in the cell. And that involves a lot of different technologies. So, it's sequencing technologies, it's protein technologies, it's, and then being able to link all this back to phenotypes is just, it's difficult, and we need methods to do that. We need a lot of data to do that. So, I think that's where all of you are coming in.
So this is a really good lead into this kind of question as to, how do we get the kind of diagnosis, genetic diagnosis improved in terms of the return on, a positive, I have the pathogenic variants here. Clearly, we're further ahead in terms of understanding coding sequence than we are all of the regulatory sequences. And so does anybody want to kind of chip in, talk a little bit? Maybe VJ, do you want to?
I'll start it. Yeah, I'll start. This is a big problem. I mean, this is essentially if you look at the diagnostic yield of genetic testing, there was a huge bump from the technology, the widespread application of exome sequencing. Basically, a lot of rare diseases can now be resolved by applying exome sequencing, before we were doing single gene testing or even gene panels. So that's a big advance, but now it's sort of petered out in terms of our improvement in diagnostic yield. We need a technology advance. Genome sequencing was thought to be this advance, and it's reasonable to consider that in terms of the acquisition of the data that it actually is, but it's really the interpretation problem. We're probably capturing what are pathologic variants. We just can't say that they are. And that's a fundamental barrier for this that many people have described in very elegant ways, but is how to solve that problem is a combination of a data sharing issue. We need to just collect more phenotypes and more genome sequencing And then some model over time will resolve which are pathogenic variants and not. So there's like an in silico computational, like, big data aspect to that. And then the second part of it is methods beyond the genome to try to resolve pathogenicity. And that can be RNA sequencing, protein sequencing, and methods yet to be sort of developed. There's methylation assays, things like that. But we have each of these kind of have to be, it's a combinatorial approach. Fundamentally, that's what we do at the Broad. And so they're not necessarily competing with each other. They can work in parallel. But fundamentally for the nano-rare patients, we have to figure out not only which is the best method, but can we deploy at scale across the whole genomics community and return it in a reasonable amount of time. So this is unresolved, but I think that's the fundamental challenge.
Jeff, do you have some points to make on that?
Yeah, I was just thinking about from the n-Lorem specific perspective, the cases that we've gotten, very often we're lucky enough, not lucky enough, but we're lucky enough that it's a mutation in a potassium channel in a known residue that lines the pore. And there's physiological evidence from the literature, and it's very clear that if we could get rid of that malformed ion channel, we could probably help this kid. But then we often get, and I'm sure the clinicians are very familiar with this, but a deep intronic mutation that's kind of near a splice site, and it's like, well, maybe that's causing this patient's phenotype, but there's no publications in literature, there's no functional data, and as amazing as n-Lorem is, we're not set up. We're set up, as you saw, we're set up to make 9 INDs, but that means that there aren't people at the bench doing basic research into these mechanisms. And so I think one of the struggles for us going forward is like, how do we build partnerships with researchers who can do those proof of concept experiments to really, because we really need to be sure about the mechanism, right? ASOs are amazing, but they can't fix every kind of genetic lesion. And we're good at this because we're careful about what cases we take on, and they're ones where we think ASOs can help. And there's often a sort of a maybe like 5 or 10% gap of proof in the literature that makes it difficult to take some patients. And that's I think our future ambitions is how do we grow that and make those relationships with researchers to help close those gaps.
I feel that there's a lot more to talk about here in terms of variant analysis, but I aware of the time. I also want to kind of talk about another aspect that I know, John, that you've thought about a lot, which is kind of biospecimen kind of collection and how you use that, and how do you kind of start to use those kind of patient-derived materials. Can you talk a little bit about that and how that might apply to these kind of ultra-rare patients?
Yeah, well, one of our guiding principles at Simons has been that we want to bring really good people into the field. And we know that one of the barriers, there are a lot of really smart people out there looking for interesting problems to work on. And we know that one of the barriers is access to really clean data and to biospecimens to go along with it. So, we're a private nonprofit. We don't hold any IP. We don't have any kind of financial stake in everything, but we've basically tried to, just like you were saying, for individuals who are sequenced in these really large cohorts like Spark, we've tried to take families who have identified highly penetrant mutations and help them start to form communities around some of these gene groups. And as part of that, then because Spark is sort of a go broad, but go a little bit light, so it's sort of try to find as many as people as possible, get them into this sort of pipeline, but sort of a light amount of information. But once we sort of can identify one of these highly penetrant mutations in these individuals, we enroll them in this sort of parallel project called Simon Searchlight. And the idea there is to go much deeper if we can. And part of that deeper aspect is trying to collect blood and other biospecimens. We have a emerging effort that I think is going to be really important. I think most people would agree with that, in trying to have IPS cells that are produced in a really clean, standardized program and made available to any researchers around the world, essentially for free. So we think that's really important. Something else that goes along with it, I don't think it's been mentioned that much here, and maybe not emphasized that much during the day, but in this other program, we're also trying to really push hard on ways to bring quantitative longitudinal phenotyping to these cohorts. So it's terrific to bring people into the clinic and to do, overnight EEGs and stuff like that. But we're really pushing hard, experimenting with different ways to bring sort of wearables and different types of gadgets into the
mix as well and get data on people that's longitudinal and that's sort of in the home setting and stuff. So I think it's a really exciting area because there's so much technology being pushed into this area that I think that's going to be really exciting.
If I could just interject, sorry, I'll try to be brief, but the 2 days before this meeting, I was across town at the group meeting of the 9th IND patient talking about his imminent treatment, which is really exciting. And Anupam Gupta, who's another neurologist at MGH, showed data from longitudinal tracking of alternating an ankle monitor and a wrist monitor in this kid, and then applying these like incredible machine learning techniques to the barrage of data. And making these really amazing symptom tracks and sort of feature space of this kid's symptoms. And sure enough, he said, it looks like this got a little bit worse here. And the parents like, oh yeah, he was doing terrible that month, and it's like, it was seen in the data before they said he was not feeling well that week. And I think, that's the future. And I think with rare patients, because we can deeply phenotype them and we're intervening with this very specific on mechanism treatment, it's a really amazing chance to develop these techniques, which could then go to the broader community.
So this is really interesting to me. So we had Kelly here as one of the patient representatives, and she has some kind of amazing kind of patient derived kind of data. Yeah. But there are services like Citizen kind of coming online that are also kind of finding ways of putting EHRs together with, other types of data like that. So what do you think is the power of that technology? I mean, Jeff's just given us a really good example. Do others kind of find that really exciting?
Short answer is yes. I mean, getting electronic health records are kind of, we're living, at least in the US, in a very fractured healthcare system, and even just changing internists, like trying to figure out, how to move records over can be really challenging. But yeah, I think that's really exciting. I think there are a lot of groups and a lot of money moving into this wearable, nearable space. I think one area that's really ripe for monitoring is sleep. I think this is a really important state of the brain, especially for CNS stuff, and the technology for being able to monitor sleep structure and sleep architecture, I think, is really coming online. So I'm very excited about this.
Yeah, I am too. I'd actually emphasize some old tech aspects of it too. I mean, I drink the Kool-Aid as a neurologist that the examination is so elegant and powerful and helping us. And it was created, a couple of 100+ years ago, still has utility, but we now can apply these sort of other phenotyping tools too. What I would add though, just as a practical matter, of our system is just that the majority of cases that n-Lorem sees, and the majority of the rare disease patients that I see too, are neurologic. I think it's just our sensitivity to picking up on these phenotypes. There probably are ultra rare liver diseases. We just don't, have a way to clinically notice them, I suppose. But the patients do kind of tell you these things, and I think a little bit of increased awareness in taking care of rare disease patients to things that are outside of your domain. Like I think Stan showed a chart of some GI symptoms in patients, and that like level of awareness, I think I myself struggle with in phenotyping these patients. And I don't think like a technological solution to that problem is necessary. It's just to get increased awareness to it, that you may be seeing the primary problem be in the central nervous system, but that actually clarity of the biology of the disease itself from an academic standpoint, and also from tracking symptoms from an outcome standpoint, biomarkers, that just like this awareness, and you're taking care of ultra rare patients, that it is multi-system in a way. It would help, and I don't have a solution to that, but I would say that is the siloed idea within medicine of phenotyping, and our each field's template for how they write their examination and notes kind of is a barrier to this integration.
Bob, any thoughts?
I mean, I don't have much to add to all of those great comments, but I will add that Jeff, you talked about how these wearables are really benefiting from the use of AI, which I think is a really point that extends past the phenotypic data, and it's the integration of that phenotypic data from these devices along with the other omics and modalities that we have. And I think AI does have a lot of potential there, to just touch back on that point, because integrating all of these modalities across a large patient set, you have multiple similar phenotypes with different gene mutations and different, and, similar gene mutations with different phenotypes, and to be able to dig more into that, the use of AI, I think, is also very exciting. So that's a little off topic anyway.
Yeah, but it's really interesting to me because nano-rare, the issue is kind of linking things together,
Yeah.
And so it seems like there's no reason not to find ways of doing this in terms of, the kind of molecular data that different patients can generate, and finding ways to link those things together over international borders. But then Vijay and I were just talking last night, like a problem that kind of nano-rare has with kind of specimen research is like getting specimens, like traveling between different places is super difficult. Yeah. Vijay, do you want to talk a little bit about that?
Yeah, I mean, this gets into a little bit of the system changes that have to occur in like academic medicine to basically enable this type of ultra research. I mean, it's again a bigger problem, but data sharing is helpful. I mean, so and these networks that can link different scientists, but it still requires that be the purview of that person's job to sort of follow up on it. There aren't I mean, many patients that come to us eventually kind of say a similar thing. They had a motivated physician taking care of them, but like this ultra deep dive into a rare disease is just not practical at the community level. So we have to have that sort of structure. Somebody eventually up the chain has to be incentivized to do this and then link up globally to do that. The regulatory stuff is probably beyond my scope to know how to solve it. But just even the first contact, there are tools some of you may use, like Matchmaker Exchange, that initial contact that somebody else exists with this can, right off the bat, spearhead, all the MTA and downstream things that are necessary. What you were speaking to about, prospective collection of cell types and cell lines, I think is really helpful too. So that you don't pick them. Some things that slow up rare disease work is that, a line would be helpful here, and going back to the patient to do a skin biopsy to generate that can slow things down.
Yeah, it's almost like it seems to me all of these different kind of technological solutions are nascent at the moment, and it's going to take a few years for them to kind of kick in. Like I'm thinking of also jumping back to the kind of ability to look at things like longitudinal data, like you have all of us, which is kind of looking at healthy individuals, but that's just starting to kind of get off the ground. So in the last 10 minutes or so, also, if anybody has a question from the audience, do feel free to raise your hands. Yeah, we have a question over here.
Yeah, my question is for Vijay, and especially as Medicaid has, I think in 7 states, has started covering all exome and genome sequencing, and I'm wondering, if you might, and you touched on it a little bit, but if you could guide us in how we can enable our community. What's the difference in turnaround time from a clinical prescription for whole exome or genome sequencing and enrolling in something like the Rare Genomes Project?
Good question. Very practical one. I don't have like an exact, I can give you a framework of that answer. It is true that I think increasingly, many people, I think we're speaking to an audience that's already converted here, that genome sequencing is ultimately kind of needs to be a clinically accessible test for some patients. Maybe reasonably not first pass, but at some point, fundamentally, the answer could be obtained without a longer odyssey, without genome sequencing. It won't solve everything, of course, but that needs to happen. So the increasing coverage for that is still a practical barrier for me. Enrollment, and so turnaround time for a clinical exome is pretty reasonable. I think like 6 weeks or something, I think is what typically been my experience. On the research side, we're much slower. So the turnaround time in the Rare Genomes Project for genome sequencing, just to even run the sequencing and get the data back before we would even interpret it, is right off the bat about 4 to 6 months sometimes. And then the interpretation can really take a much longer after that. And that part of that is these are already difficult, hard to solve individuals from the first place. So that's part of the bias, but also like we're just not having like a sort of a scalable way to turn around that faster. So in general, of course, I always tell my patients, if you can obtain this test clinically, do that first before enrolling in a research-based platform. Commercial genome sequencing is still, I think, in the space of self-pay, and the standards for what needs to be returned or not, in my opinion, are not firmly established. So it's still a world that needs a little bit more clarification over time of exactly what the deliverable are, the turnaround times are. Places have shown you can do ultra rapid genome sequencing. Rady has shown this, that you can practically do genome sequencing in a day. But how to deploy that and do that is this real challenge. On the research side, we're much slower.
Sam.
I thought just a couple of comments. I think in the midst of the wearables and the AI enthusiasm, which I share, I think VJ said something that may have gone by people. That is a lesson that we've really learned at n-Lorem. And that is not AI, but intelligence. It's good to listen to patients and families. And I see constant examples of patients and families who try to tell professionals what's happening and are ignored. And for me, I always listened to the patient when I was still seeing patients, and that's where I learned. And in doing n-Lorem, I'm reminded constantly of the value of the parent observer for our patients and the problem in getting physicians to actually listen to what people want to tell them. And I hear that all the time. So yeah, let's engage in AI, but how about just some simple intelligence would be a really nice step forward, I think. And the second thing I want to talk about is greed. I think we stumble over greed about 90% of the time. And we run into greed with the office in every medical center who's responsible for getting cash. And so we spend incredible hours and days and months in trying to get a simple MTA done. And that is nothing, yes, there are some, we have to protect patient confidentiality, but most of what we deal with is greed. And it's time to get the greed out of the way and let us just help these patients. And it really annoys the hell out of me. There are a lot of things.
You seem so mellow, Stan.
Oh, they'll turn it up. I'm sorry, thank you. Well, what I'd really like to ask BJ is what we can do to get Heidi and Ann to work a little more closely with UDN and n-Lorem, but I'm not going to ask that question.
So above my pay grade, yeah.
I don't think it's actionable. You mentioned, functionalizing genetic lesions, connecting genotype and phenotype, by doing GWAS on ever larger data sets. And, we went from Nomad to UK Biobank and got Estonia and all of us you mentioned. And still, what are we going to get down to? Minor allele fractions of 0.1% maybe, by going to ever larger sets. And yet severe, if I understand correctly, I'm under the impression that severe pediatric disease is largely de novo disease, and you're going to have allele fractions near zero.
Yeah.
And so I'm wondering what if you have any inspirations for what we can do to more easily functionalize those extremely rare mutations that we only see in 30 people in the world.
Yeah, I mean, good question. I mean, I think these are like the GWAS and like large data sets, all of us, genomic, like these are different. I don't know, the metaphor that this isn't my thought, but I forgot who said it is, one is like your telescope's looking into deep space, like large, vast expanses. And the other direction in the rare disease space is the microscope in that direction. You're going to see different architecture at those different scales. And so both the technology is the same how you're looking. It's just like optics for the prior metaphor, and here it's just genomic sequencing and functional experiments. But I think what you'll learn is like a better resolution of constraint. You know, what parts of the gene are just intolerable to variation, and why is that? Like, why is that domain important? That's the academic question that I think will be resolved by that. That's very different from the population. You know, where the minor allele frequency is something like, you know, 0.1 or higher. That the ultra rare, where it's like unique, one of the, maybe you'll never see that particular variant ever again. But I think, you know, I think that's what the functional experimentation is going to be revealing to it, as like, but what like high impact intolerable variation is across the genome. Yeah.
Great. So we're running towards the end of our time. I don't want to keep anybody. It's a fantastic panel. I think kind of just I'm sure I suspect Stan's going to talk about this, but one other thing that I was hoping to get to that we didn't really get to was like how little NIH funding goes into this area. Yeah. And Stan, I presume you were going to be talking about that. All right. So if we could just thank our panel.
Yeah.
I think that's a great introduction to Stan. It is your turn.
Thank you. First of all, I have to rearrange some furniture so I don't fall. You see, we're a small organization. In my old organization, I had 10 people carry chairs for me, but now I have to do it myself. It's been a long and information-packed and wonderful day. And I know that we're all getting a bit tired, but I do want to talk about reforms. And I am going to really just hit the high points. We've encountered barriers that astonish me, of institutions unwilling to take care of patients. I don't understand that. We've encountered physicians who seem not to care about patients getting cared for. I don't understand that. And we encounter reluctance because of novelty. I thought our tertiary care institutions were to create novelty. And what we see is an unwillingness to consider novelty in many places. And so I won't spend a lot of time talking about those things because I don't have good ideas about how to solve them. They're human being problems. There are limitations in imagination and limitations in the way organizations actually work. And most of the time, we can't even figure out who makes the decision in a tertiary care institution to join us or not. We don't even know who makes those decisions. So there's a lot that we need to learn and do that have been tremendous surprises to me personally and deep disappointments in many ways. But there are some obvious things that I think cry out for us to put our shoulders to the wheel to change. And I want to preface all of this with reform is not easy. If you think about driving reform, you need to get ready to persevere. And there will be many, many solid reasons that people will raise against doing something that hasn't been done before. And then there will be a host of nonsensical reasons that are raised for all kinds of reasons. And so I do not make these recommendations lightly in any way. And I do not minimize how much effort is going to be taken to get this done. But I do say that if we don't tackle these things at the N of 1 level, they may never happen. And we cannot wait. We cannot wait to get to work on these. And so I believe strongly that the single most important step forward to enable a more rapid diagnosis of nano-rare patients and all patients is that we introduce genomic sequencing and whole genome sequencing into newborn evaluation protocols. Now, there are tremendous benefits from that. It will not even begin to solve all the problems, but it will begin to identify the problems for us in an earlier and more convenient and more actionable way. If you think about it, we have no idea what the incidence, real incidence of non-pathogenic and pathogenic mutations is. We don't know what the prevalence of any of these things are, and we will not know until programs like Genomics UK actually spread around the world, and we sequence every human being. And that's just, of course, the beginning. We don't know today which mutations are pathogenic and which are not. We don't have a reason to understand. We don't know, have even an intellectual basis to think about all this in a functional way. And until we gather some of this information, we will never get to nano-rare patients before they're already progressed well beyond the place where they need to be. So it is critical. I think it's also fascinating to think about early identification of potential environmental factors or genetic factors that are driving mutational changes in populations. And I do think that happens. And again, how will we learn that if we don't begin sequencing populations of human beings and correlating that with phenotype in a patient identifiable way? There are a lot of genotype databases, GWAS, a lot of phenotypes of various kinds. I think that's been invested enough, frankly. I think now we need to bring genotype, phenotype, and patient together in fashions that are then actionable and understandable. What are the single gene mutations that cause disease? What do those diseases look like? I would love to see a compendium of the nano-rare mutations and the syndromes associated with those. We will never have that if we don't begin to think about just this very first step. And of course, we can reduce cost. Long-term, there's no question, and I think the UDN has done a beautiful job of demonstrating that just achieving a diagnosis is a fundamental advance in terms of emotions and care, but it's also a fundamental way of reducing cost of healthcare. And so anything that will allow us to move more rapidly to a diagnosis is going to reduce healthcare costs, going to reduce journeys that are non-productive and often destructive, and going to reduce the harm that happens to our patients before they make their way to us. We need to do that. And then there will be lots of issues. What about the risk of misuse of the information? There's a lot of people who worry about labeling, their children. I think, frankly, that's getting the cart before the horse. I think if you don't have a diagnosis, you don't have a label, but you do have a problem. And so I think these are legitimate issues that we have to work through. We certainly don't want a rebirth of eugenics and all of the abuses that can take place there, but these are manageable problems. We can't spin around and worry about those things. Those are soluble problems. It is not soluble to help a patient who's progressed to the place that they're beyond help. That's an insoluble problem. And so I think good people come together and find solutions. I think that's not enough. What I'm looking forward to is coupling genomics and phenotype and standardizing phenotypic descriptions. We have multiple examples of just terrible phenotype descriptions from the physicians that we're seeing. And these are the top of the line physicians. They're taking care of patients in academic centers. And there's just tremendous variability in what gets characterized as a phenotype and what doesn't. Right now, we're digging into our data just to see how many ion channel patients actually have GI symptomatology that wasn't bothered reporting, that nobody bothered to put down on a case report. We know it exists. So we need to find ways to enhance genotype to phenotype, and we need to enhance the ways of describing phenotype and do a better job of standardizing that and assuring that every possible element of the phenotype is recorded. Just because you're not a gastroenterologist doesn't mean that the gastroenterology problems that that patient has shouldn't be recorded so that we know about it. And we see a lot of that. We see a lot of it. So the benefits, of course, I think are obvious. Again, as we think about coupling genotype to phenotype, patient confidentiality and potential abuse of that information becomes central. And I would again just say, yes, that's all true. But yes, that's a soluble problem for good people to put their heads together and find solutions that will work. It is an insoluble problem if we don't have the information. So that's number two. Number three, I think of epidemiology studies. I'm a hypothesis testing scientist. I see we're in big data and GWAS and yada, yada, yada. But in the end, that's all screening information that then generates the opportunity to test hypotheses. And I think of traditional epidemiologic studies as body counting. You know, I mean, Framingham was wonderful, but it's time to move on. And here, if we can couple genotype, phenotype, and then long-term follow-up to omics, and there are multiple omics, and the genotype is in some ways the most state of information, and what we need is kinetic information that we can get from transcriptomics, from proteomics, from metabolomics, from methylomics, and so on. And so there are some technological advances that are needed, there are cost improvements that are needed, but if we could, for example, take nano-rare patients who have a defined, well-understood pathogenic mutation and couple that to long-term follow-up and multiple omic evaluations, we could begin to conduct molecular epidemiologic studies. That would begin to teach us at the cellular and molecular and pathway level about health and disease. It's time to move beyond counting bodies, in my view. The benefits are just knowledge, just how much knowledge we're missing that could be available to us with just the methods that we have today, with just some changing in funding. And if we could encourage people to see the incredible value of a human being with a single variable as a test model for health and disease, I think we could really do some good. And again, these are the same problems, the patient confidentiality and costs and so on and so on. But I would say take $1 billion that we spend on animal models and try putting $1 billion into human models. Call them nano-rare human models. We spend how many $1 billion a year doing that, animal models that nobody likes. We have these patients who are the model of the future of health and disease. So another thing that I'd like to see happen. This is another one. We find there's an incredible amount of money for research that's long-term to find treatments. But what we were shocked at was when we said, okay, well, that's great, but we can help a patient today. We had a very difficult time getting people to get beyond the fact that no, their system is designed to support research to find a treatment. And it just seems to me that we need to think about reallocating resources. Not a lot, we don't need a lot, but if we just reallocate some of the money that's invested in more traditional approaches to thinking about these patients as an extraordinary resource, and that the treatment of these patients with specific genetic therapy provides an extraordinary opportunity to learn in reverse about the ontogeny of disease, I think we could do just a lot more. And so it is about money. but it's about thinking in new ways that are now available to us because these patients are now known to exist. We didn't know they existed until we did sequencing. Now we do. How do we take advantage of them in the most efficient way? So it would be good if we could work together to change NIH funding so that there was more money for nano-rare patient research and opportunities to help institutions with the financial costs of treating these patients in a more research-intensive environment. And that just requires a small shift in the billions of dollars of NIH money. It doesn't require a lot. It won't. And yet, it will make an enormous difference. Again, one of the big things I wanted to get out of today for all of you so that you can carry this forward is the unbelievable research opportunity these patients represent. And we need to encourage then more and more scientists in academia to be interested in these as models of health and disease. We find an astonishing lack of interest in academic centers in this remarkable resource that we're bringing to them, a patient with a single variable introduced, driving an enormous phenotype. Let's see, what else do I want to say? I'm really, I grew up in cancer, and the first 5 years of my career, I built the first broadly successful anti-cancer program in the industry, if you can believe it. There was the conventional wisdom, and when I went in the industry, is you couldn't make money in cancer. Well, I've changed that. And one of the things that cancer treatment taught us very early is that you need to think holistically about the patient. You need a team approach. And what we see in these patients is deeply disheartening, in that they're cared for by an individual physician. The places that are doing great jobs like Columbia and so on, they really bring a team together, and they recognize that these mutations affect an individual, but they affect a family, and that we have to think about these patients holistically, and we have to think about the family, and we have to bring teams together to think about the entire phenotype of that patient, that we bridge somehow the gap between a neurologist and a gastroenterologist and a hematologist and so on. That's been done. It should be done for these patients. And one of my real concerns is that sort of system weariness that I mentioned earlier in the day, where it seems like there's a tremendous amount of energy invested in the patient initially. And then when there's no answer, no solution, it seems like the system loses interest. That patient is still suffering. The family is still suffering. Where's the support? Where did that go? Why is there no one taking care of Connor Dalby for 10 years? Why is that? How can that be? How can we allow that to happen in our medical system? It's got to change. So that's a very high level list. I'm sure that there are many other opportunities for reform, and I'm sure there are lots of concerns about this and that and the other. But I believe strongly that treatment is always the tip of the spear that drives reform. And n-Lorem can be the tip of the spear. And so that I ask that all of you think about these and those reforms that you think make sense. I want you to join us to help begin drive those reforms forward. I don't ask that you agree with everything I just said, of course not. But where you do agree, then join us in helping drive those reforms in the coming years. It's a big deal, and it's a big opportunity. And to see so many of these patients suffer and die, and the opportunity to help missed and the opportunity to learn squandered is heartbreaking. I want to see it end. Make sense to everybody? Ask for your help. Thanks. I'm supposed to end it now. Oh, what am I supposed to do now, Amy? I have to have somebody tell me what to do all the time. Q&A. I hope I've stimulated some questions and comments and probably strong disagreements about some of the things I just said. Questions or comments from anyone? Sesh, how about you? You know, I listen very carefully to what Sesh says. He's a very wise person in my mind.
And I certainly applaud everything that you have suggested.
Andrew?
The mic's not working,
I think the suggestion about holistic care and listening to patients and families is critical for moving this field forward. I am somewhat more optimistic perhaps than you are that we can find incentives both for institutions and for individuals that will help do this. But we're going to need a village to be able to do this. We're going to need third-party payers. We're going to need pharmaceutical companies. We're going to need the federal government in some way, shape, or form. And we're going to need probably some enabling legislation at the federal level of some kind to sort of provide these incentives for a public-private partnership to drive the changes that you've outlined.
I couldn't agree with you more, Sesh. And I'm frustrated. I'm always frustrated. I'm always impatient, as anybody who lives with me knows. But I'm always optimistic. I mean, you wouldn't do the things I've done in my life if I weren't optimistic. And I think one of my hopes out of this meeting, and I think it's going to be realized, is to bring a group of people who are senior like you and the many CEOs we have and others together to help begin driving various types of reforms in various ways. And in the end, it's all about focus and funding and putting inducements in to do as opposed to impediments to stop, right? And I think at least today demonstrates that it's possible. And if it's possible, then I think it becomes something that we can do together and sell. I agree with you completely. Question back here.
Prioritize the things that have the biggest impact on the patient first. So I'm starting at the bottom of your list, the holistic care. I think that speaks to me as a patient dad very clearly. A brief example of this story is about how the medical system is not able to deal with the diagnosis information and process it at all. The way we found out about our diagnosis was we had taken a whole exome sequence swab months ago. We were in and out of the hospital 2 or 3, 4 times a week at Boston Children's. For some reason, our genetics appointment to tell us this diagnosis was in the middle of a workday over Zoom. And when we found out the diagnosis, there was no follow-up. There was promise that they would be there for us, that we would be able to ask follow-up questions, that they would schedule follow-up appointments with other services we weren't currently seen by, and none of that happened. We sent messages, no one responded for months. So I think all of the steps are critical, but the pieces that if you just go out and do more whole exome sequencing or whole genome sequencing to find more cases, but then patients have nowhere to go or nowhere to turn, there's no point. So I would suggest that the patient lens that you guys have very laudably pushed at n-Lorem is also an important lens to prioritize some of these things.
Oh, I really agree. And I think what you just said is really important. It's the art of the possible while we try to do the impossible. And you know, we spend a lot of time trying to help patients try to find physicians and institutions, and we keep looking for solutions that will sort of be a clearinghouse for people to where they're going to go and how to get there and that sort of thing. And then I think there are examples, you know, I think of Columbia, Rush Presby, and others of examples of holistic care systems that are working, and they need to be exported. And again, I think the more that we can do of that, the better the care will be, and that's something that can happen immediately and doesn't require a change in attitudes that are enormous. So I completely agree.
Hi. So as a daughter of a patient who's nano-rare, I just have the question of, I guess, like, as somebody who's waiting for the ASO to potentially be developed, like, how hard are the doctors working to find the right ASO? And if it doesn't work, what do you recommend families to do?
I'm sorry, I'm not sure I heard the question as well as I need to. Could you mind?
Essentially, my question is, if the ASO treatment doesn't come to fruition for whatever reason, what do you recommend families to do?
Yeah, that's a hard question. There just isn't, unfortunately, the thing we need most is industrial scale gene therapy that's cost effective. And that doesn't exist today. It's not safe enough. It's not cost effective enough. It's not generalizable enough. Because most of the patients we really can't help are null mutations. They're people who have a loss of function that we can't address. And so when we run into patients we can't help, we're today at a loss. We really like what the UDN Therapeutic Match Committee is doing, looking for existing drugs that could be repurposed, looking at antibodies that might be repurposed, thinking about genes. And I know that there are a lot of efforts in gene therapy at the NIH and so on that are designed to help. But I just think it's a long haul still. So that's one of the many questions that I don't have a good answer for. Maybe there are others in the audience who have a better answer than I do. I wish I had a better one. Anybody want to provide a better answer for that one? I'd love to have it. Okay.
My answer is kind of a question, and that's, do you think a missing part of the puzzle, Stan, for everybody is genetic counseling and the role of a genetic counselor?
Could you, I'm sorry.
How important do you think the role of a genetic counselor is in this puzzle?
I think, well, I think you have to have a genetic diagnosis, and then having a knowledgeable individual or group who can tell you what that actually means and tell you what more information is required to make it an actionable observation is the missing piece that I see a lot of. I think the UDN has worked hard at that, and there are other places that are working hard. But it's sort of beyond more general genetic counseling. It's more focused on how to interpret a genetic finding and how to be confident that the genetic finding means what you think it means, and where to go with that information that I find is most urgently needed in our patients. And that's a sort of different thing from genetic counseling more traditionally.
This will be the last question of the day.
So
you've got a lot of responsibility.
So this actually follows.
The day's gone so well. If you screw it up, I'm going to be very upset.
So to your point about the scarcity of individuals who can answer this type of complex neurogenetic, genetic and neurogenetic question, one thing that I didn't see on your list of things to do is to build the genomics infrastructure and workforce, genetic counselors, geneticists who can assess these mutations, and some of the reluctance you might be encountering at many tertiary care centers might even be the absence of somebody who feels confident and prepared to answer those types of questions. And so
one of the things that I think is very important is for mentoring and training programs to make sure that we have an adequate workforce, because when we do move to pan genomic sequencing, the numbers and complexities of genetic questions are just going to escalate. So that's another thing we need to be working on to build capacity.
I absolutely agree with you. And while the last thing I can do is counsel patients to parents and patients who have no time. But I do think it's also important to know that for everyone to understand that we're in the midst of a revolution that's just getting started. And so we're going to see continuing advances, reductions in cost, and new methods that elaborate. And just finding people who can integrate all that information and keep us at the forefront of where science is going to be an amazing challenge. And I'm no longer an educator, and I don't envy trying to train physicians today. It's even more complex than it ever was, right? There's so much more to know and to learn. So I think that's the last of the Q&A, and now my job is to bring this to an end. Okay. So we're going to come to an end here, I promise. I thought before I do that, I'd tell you the most important thing I've learned in my life. And problem with all this is I like poetry. I've always liked poetry since I can remember. If you like poetry, you learn very early on that anything worth saying has been said better by a poet, and so you end up just quoting people. But as I look at so-called intellectually evolved species and ask, what is the consistent difference between human beings and others? I really can only find one. And opposable thumb comes pretty close, but you got the possum and a few others. And that is that we have a unique ability to dream collectively. And I commend that to you. And I say that's the only difference, but if you look around you, everything that you see is a product of a collective dream of other human beings. We've changed our planet, we're changing the solar system, we may change the universe, and it will be because of our ability to dream collectively. And it works the same way every single time. There is a person who sees the world as it is, and as the world he would like to create. And that's a dream. Dreams by themselves are not worth much, but he also has a plan. It's not anywhere close to foolproof, but it's a plan that, has some plausibility, and he can galvanize others to join him. That's a collective dream. And n-Lorem is really nothing more than a collective dream. We are in the process of dreaming together and then finding ways to make that dream a reality. So I realized late in my career that really all I've ever been is a dream merchant. And I also realized that big dreams are easier to sell than small dreams, and I've never understood why anybody who dreams would want to dream small. So n-Lorem is actually a big dream. It's a big dream to change the world in little ways. And I appreciate your willingness to be a part of the n-Lorem collective dream. And now I could have said that with just these words. This is from one of our modern poets, Bruce Springsteen, and it's a title of one of his songs. Dream, baby, dream. That's my advice. Dream, baby, dream. So let me end this with thanking everybody. First, thanks to our wonderful hosts from Biogen. My God, what a wonderful job they did. Thank you very much, everybody. I also want to thank everybody at n-Lorem, Amy and Kim and all the speakers today, and Andrew and Nefiso and Catherine and Alexia back in the homestead. I think the job that everybody did today, and I'm so proud of the team and what they shared with you and how they shared it. So join me in thanking everybody for that. They tell me they're a little tired. I tell them I don't really care. Who cares? Just go to work. I want to thank all of our speakers and our panelists and moderators. I think you all did an incredible job. And I know that I learned a lot as the day went on, and I hope everyone else learned a lot as well. And so thank you all for that. Thank you to our patients and families. Those of you who made the trip here, what a wonderful experience for us to be able to share the day with you, and for you to share with us, the challenges that you face every day. We thank you for being the reason we're here today. And then thanks to all of you. I'm amazed. It's still it's the end of the day, and most of you are still here, and some of you are still awake. Although I saw Ted sleeping earlier, and I'm going to have words with him after later on. But in any event, I hope that you leave the day knowing better what we're doing, and I hope you leave the day as a cohesive, empathetic, single unit, committed to making the changes that we have to make to make life better for our patients. Thank you so much for all your efforts. I forgot to thank the AV people. Great job, folks.